Section number 1 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. Structure of the Nervous System Part 1. The nervous system is the most complicated and highly organized of the various systems which make up the human body. It is the mechanism concerned with the correlation and integration of various bodily processes and the reactions and adjustments of the organism to its environment. In addition, the cerebral cortex is concerned with conscious life. It may be divided into two parts, central and peripheral. The central nervous system consists of the encephalon or brain contained within the cranium and the medulla spinalis or spinal cord lodged in the vertebral canal. The two portions are continuous with one another at the level of the upper border of the atlas vertebra. The peripheral nervous system consists of a series of nerves by which the central nervous system is connected with the various tissues of the body. For descriptive purposes, these nerves may be arranged in two groups, cerebrospinal and sympathetic. The arrangement, however, being an arbitrary one, since the two groups are intimately connected and closely intermingled. Both the cerebrospinal and sympathetic nerves have nuclei of origin, the somatic efferent and sympathetic efferent, as well as nuclei of termination, somatic afferent and sympathetic afferent, in the central nervous system. The cerebrospinal nerves are 43 in number on either side, 12 cranial attached to the brain and 31 spinal to the medulla spinalis. They are associated with the functions of the special and general senses and with the voluntary movements of the body. The sympathetic nerves transmit the impulses which regulate the movements of the viscera, determine the caliber of the blood vessels, and control the phenomena of secretion. In relation with them are two rows of central ganglia, situated one on either side of the middle line in front of the vertebral columns. These ganglia are intimately connected with the medulla spinalis and the spinal nerves, and are also joined to each other by vertical strands of nerve fibers so as to constitute a pair of knotted cords, the sympathetic trunks, which reach from the base of the skull to the coccyx. The sympathetic nerves issuing from the ganglia form three great prevertebral plexuses which supply the thoracic, abdominal, and pelvic viscera. In relation to the walls of these viscera, intricate nerve plexuses and numerous peripheral ganglia are found. Structure of the nervous system. The nervous tissues are composed of nerve cells and their various processes, together with a supporting tissue called neuroglia, which, however, is found only in the brain and the medulla spinalis. Certain long processes of the nerve cells are of special importance, and it is convenient to consider them apart from the cells. They are known as nerve fibers. To the naked eye, a difference is obvious between certain portions of the brain and medulla spinalis, that is, the gray substance and the white substance. The gray substance is largely composed of nerve cells, while the white substance contains only their long processes, the nerve fibers. It is in the former that nervous impressions are received, stored, and transformed into efferent impulses, and by the latter that they are conducted. Hence, the gray substance forms the essential constituent of all the ganglionic centers, both those in the isolated ganglia and those aggregated in the brain and medulla spinalis, while the white substance forms the bulk of the commissural portions of the nerve centers and the peripheral nerves. Neuroglia. Neuroglia, the particular ground substance in which are embedded the true nervous constituents of the brain and medulla spinalis, consist of cells and fibers. Some of the cells are stellate in shape, with ill-defined cell body, and their fine processes become neuroglial fibers, which extend radially and unbranched among the nerve cells and fibers which they aid in supporting. Other cells give off fibers which branch repeatedly. Some of the fibers start from the epithelial cells lining the ventricles of the brain and central canal of the medulla spinalis, and pass through nervous tissue, branching repeatedly to end in slight enlargements on the pia mater. Thus, neuroglia is evidently a connective tissue in function, but is not so in development. It is ectodermal in origin, whereas all connective tissues are mesodermal. Nerve cells. Nerve cells are largely aggregated in the gray substance of the brain and medulla spinalis, but smaller collections of these cells also form the swellings called ganglia seen on many nerves. These latter are found chiefly upon the spinal and cranial nerve roots and in connection with the sympathetic nerves. The nerve cells vary in shape and size and have one or more processes. They may be divided for purposes of description into three groups according to the number of processes which they possess. 1. Unipolar cells, which are found in the spinal ganglia. The single process after a short course divides in a T-shaped manner. 2. Bipolar cells, also found in the spinal ganglia, when the cells are in an embryonic condition. They are best demonstrated in the spinal ganglia of fish. Sometimes the processes come off from opposite poles of the cell, and the cell then assumes a spindle shape. In others, both processes emerge at the same point. In some cases, where two fibers are apparently connected with a cell, one of the fibers is really derived from an adjoining nerve cell and is passing to end in a ramification around the ganglion cell. Or, again, it may be coiled spirally around the nerve process which is issuing from the cell. 3. Multipolar cells, which are pyramidal or stellate in shape and characterized by their large size and by the numerous processes which issue from them. The processes are of two kinds. One of them is termed the axis cylinder process, or axon, because it becomes the axis cylinder of a nerve fiber. The others are termed the protoplasmic processes, or dendrons. They begin to divide and subdivide as soon as they emerge from the cell, and finally end in minute twigs and become lost among the other elements of the nervous tissue. The body of the nerve cell, known as the cyton, consists of a finely fibrillated protoplasmic material of a reddish or yellowish-brown color, which occasionally presents patches of a deeper tint, caused by the aggregation of pigment granules at one side of the nucleus, as in the substantia nigra and locus coeruleus of the brain. The protoplasm also contains peculiar angular granules which stain deeply with basic dyes, such as methylene blue. These are known as Nissel's granules. They extend into the dendritic processes but not into the axis cylinder. The small clear area at the point of exit of the axon is termed the cone of origin. These granules disappear, chromatolysis, during fatigue or after prolonged stimulation of the nerve fibers connected with the cells. They are supposed to represent a store of nervous energy, and in various mental diseases are deficient or absent. The nucleus is, as a rule, a large, well-defined spherical body, often presenting an intranuclear network and containing a well-marked nucleolus. In addition to the protoplasmic network described above, each nerve may be shown to have delicate neurofibrils running through its substance. These fibrils are continuous to the fibrils of the axon and are believed to convey nerve impulses. Golgi has also described an extracellular network, which is probably a supporting structure. Nerve fibers. Nerve fibers are found universally in the peripheral nerves and in the white substance of the brain and medulla spinalis. They are of two kinds, that is, medullated, or white fibers, and non-medullated, or gray fibers. The medullated fibers form the white part of the brain and medulla spinalis, and also the greater part of every cranial and spinal nerve, and give to these structures their opaque white aspect. When perfectly fresh, they appear to be homogeneous, but soon after removal from the body, each fiber presents, when examined by transmitted light, a double outline or contour, as if consisting of two parts. The central portion is named the axis cylinder. Around this is a sheath of fatty material, staining black with osmic acid, named the white substance of Schwann or medullary sheath, which gives to the fiber its double contour, and the whole is enclosed in the delicate membrane, the neurolemma, primitive sheath, or nucleated sheath of Schwann. The axis cylinder is the essential part of the nerve fiber and is always present. The medullary sheath and the neurolemma are occasionally absent, especially at the origin and termination of the nerve fiber. The axis cylinder undergoes no interruption from its origin in the nerve center to its peripheral termination, and must be regarded as a direct prolongation of a nerve cell. It constitutes about 1/2 or 1/3 of the nerve fiber, being greater in proportion in the fibers of the central organs than in those of the nerves. It is quite transparent, and is therefore indistinguishable in a perfectly fresh and natural state of the nerve. It is made up of exceedingly fine fibrils, which stain darkly with gold chloride, and at its terminations may be seen to break up into these fibrillae. The fibrillae have been termed the primitive fibrillae of Schulze. The axis cylinder is said by some to be enveloped in a special reticular sheath, which separates it from the medullary sheath and is composed of a substance called neurokeratin. The more common opinion is that this network or reticulum is contained in the white matter of Schwann, and by some it is believed to be produced by the action of the reagents employed to show it. The medullary sheath, or white matter of Schwann, is regarded as being fatty matter in a fluid state, which insulates and protects the essential part of the nerve, the axis cylinder. It varies in thickness, in some forming a layer of extreme thinness so as to be scarcely distinguishable, in others forming about 1/2 of the nerve fiber. The variation in diameter of the nerve fibers, from 2 to 16 microns, depends mainly upon the amount of white substance, though the axis cylinder also varies within certain limits. The medullary sheath undergoes interruptions in its continuity at regular intervals, giving the fiber the appearance of constriction at these points. These are known as the nodes of Ranvier. The portion of nerve fiber between two nodes is called an internodal segment. The neurolemma, or primitive sheath, is not interrupted at the nodes, but passes over them as a continuous membrane. If the fiber be treated with silver nitrate, the reagent penetrates the neurolemma at the nodes, and on exposure to light reduction takes place, giving rise to the appearance of black crosses, Ranvier's crosses, on the axis cylinder. There may also be seen transverse lines beyond the nodes termed Frohman's lines. The significance of these is not understood. In addition to these interruptions, oblique clefts may be seen in the medullary sheath, subdividing it into irregular portions which are termed medullary segments, or segments of Lantermann. There is reason to believe that these clefts are artificially produced in the preparation of these specimens. Medullated nerve fibers, when examined in the fresh condition, frequently present a beaded or varicose appearance. This is due to manipulation and pressure causing the oily matter to collect into drops, and in consequence of the extreme delicacy of the primitive sheath, even slight pressure will cause the transudation of the fatty matter, which collects as drops of oil outside the membrane. The neurolemma, or primitive sheath, presents the appearance of a delicate structureless membrane. Here and there beneath it, and situated in depression in the white matter of Schwann, are nuclei surrounded by a small amount of protoplasm. The nuclei are oval and somewhat flattened, and bear a definite relation to the nodes of Ranvier, one nucleus generally lying in the center of each internode. The primitive sheath is not present in all medullated nerve fibers, being absent in those fibers which are found in the brain and medulla spinalis. Wallerian degeneration. When nerve fibers are cut across, the central ends of the fibers degenerate as far as the first node of Ranvier, but the peripheral ends degenerate simultaneously throughout their whole length. The axons break up into fragments and become surrounded by drops of fatty substance which are formed from the breaking down of the medullary sheath. The nuclei of the primitive sheath proliferate, and finally absorption of the axons and fatty substance occurs. If the cut ends of the nerve be sutured together, regeneration of the nerve fibers takes place by the downgrowth of axons from the central end of the nerve. At one time it was believed that the regeneration was peripheral in origin, but this has been disproved. The proliferated nuclei in the peripheral portions taking part merely in the formation of the so-called scaffolding along which the new axons pass. Non-medullated fibers. Most of the fibers in the sympathetic nervous system and some of the cerebrospinal consist of gray or gelatinous nerve fibers, fibers of Rhombic. Each of these contain an axis cylinder to which nuclei are applied at intervals. These nuclei are believed to be in connection with a delicate sheath corresponding with the neurolemma of the medullated nerve fiber. In external appearance, the non-medullated nerve fibers are semi-transparent and gray or yellowish-gray. The individual fibers vary in size, generally averaging about half the size of the medullated fibers. End of section 1. Section 2 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. Structure of the Nervous System, Part 2. Structure of the peripheral nerves and ganglia. The cerebrospinal nerves consist of numerous nerve fibers collected together and enclosed in membranous sheaths. A small bundle of fibers enclosed in a tubular sheath is called a funiculus. If the nerve is of small size, it may consist of only a single funiculus, but if large, the funiculi are collected together into larger bundles of fasciculi, which are bound together in a common membranous investment. In structure, the common membranous investment or sheath of the whole nerve, epineurium, as well as the septa given off from it to separate the fasciculi, consist of connective tissue composed of white and yellow elastic fibers, the latter existing in great abundance. The tubular sheath of the funiculi, perineurium, is a fine, smooth, transparent membrane which may be easily separated in the form of a tube from the fibers it encloses. In structure, it is made up of connective tissue, which has a distinctly lamellar arrangement. The nerve fibers are held together and supported within the funiculus by delicate connective tissue called the endoneurium. It is continuous with septae which pass inward from the innermost layer of the perineurium and shows a ground substance in which are embedded fine bundles of fibrous connective tissue running for the most part longitudinally. It serves to support capillary vessels arranged so as to form a network with elongated meshes. The cerebrospinal nerves consist almost exclusively of medullated nerve fibers, only a very small proportion of non-medullated being present. The blood vessels supplying a nerve end in a minute capillary plexus, the vessels composing which pierce the perineurium and run, for the most part, parallel with the fibers. They are connected together by short transverse vessels, forming narrow oblong meshes, similar to the capillary system of muscle. Fine, non-medullated nerve fibers, vasomotor fibers, accompany these capillary vessels, and break up into elementary fibrils, which form a network around the vessels. Horsley has demonstrated certain medullated fibers running in the epineurium and terminating in small spheroidal tactile corpuscles, or end bulbs of Krauss. These nerve fibers, which Marshall believes to be sensory, and which he has termed nervi nervorum, are considered by him to have an important bearing upon certain neuralgic pains. The nerve fibers, so far as it is present known, do not coalesce, but pursue an uninterrupted course from the center to the periphery. In separating a nerve, however, into its component funiculi, it may be seen that these do not pursue a perfectly insulated course, but occasionally join at very acute angle with other funiculi proceeding in the same direction. From this, branches are given off to join again in like manner with other funiculi. It must be distinctly understood, however, that in these communications the individual nerve fibers do not coalesce, but merely pass into the sheath of the adjacent nerve, become intermixed with nerve fibers, and again pass on to intermingle with the nerve fibers in some adjoining funiculus. Nerves, in their course, subdivide into branches, and these frequently communicate with branches of a neighboring nerve. The communications which thus take place form what is called a plexus. Sometimes a plexus is formed by the primary branches of the trunks of the nerves, as the cervical, brachial, lumbar, and sacral plexuses, and occasionally by the terminal funiculi, as in the plexus formed at the periphery of the body. In the formation of a plexus, the component nerves divide, then join, and again subdivide in such a complex manner that the individual funiculi become interlaced most intricately, so that each branch leaving a plexus may contain filaments from all the primary nerve trunks which form the plexus. In the formation also of smaller plexuses at the periphery of the body, there is a free interchange of the funiculi and primitive fibers. In each case, however, the individual fibers remain separate and distinct. It is probable that through this interchange of fibers, every branch passing off from a plexus has a more extensive connection with the spinal cord than if it had proceeded to its distribution without forming connections with other nerves. Consequently, the parts supplied by these nerves have more extended relations with the nervous centers. By this means, also, groups of muscles may be associated for combined action. The sympathetic nerves are constructed in the same manner as the cerebrospinal nerves, but consist mainly of non-medullated fibers, collected in funiculi and enclosed in sheaths of connective tissue. There is, however, in these nerves a certain admixture of medullated fibers. The number of these latter varies in different nerves and may be estimated by the color of the nerve. Those branches of the sympathetic which present a well-marked gray color are composed chiefly of non-medullated nerve fibers intermixed with a few medullated fibers, while those of a white color contain many of the latter fibers and few of the former. The cerebrospinal and sympathetic nerve fibers convey various impressions. The sensory nerves, called also centripetal or afferent nerves, transmit to the nervous centers impressions made upon the peripheral extremities of the nerves, and in this way the mind, through the medium of the brain, becomes conscious of external objects. The centrifugal or efferent nerves transmit impressions from the nervous centers to the parts to which the nerves are distributed. These impressions, either exciting muscular contraction or influencing the processes of nutrition, growth, and secretion. Origins and terminations of nerves. By the expression, quote, the termination of nerve fibers, end quote, is signified their connections with the nerve centers and with the parts they supply. The former are sometimes called their origins or central terminations, the latter their peripheral terminations. Origins of nerves. The origins in some cases is single, that is to say, the whole nerve emerges from the nervous center by a single root. In other instances, the nerve arises by two or more roots, which come off from different parts of the nerve center, sometimes widely apart from each other. And it often happens, when a nerve arises in this way by two roots, that the function of these two roots are different, as, for example, in the spinal nerves, each of which arises by two roots, the anterior of which is motor, and the posterior sensory. The point where the nerve root or roots emerge from the surface of the nervous center is named a superficial or apparent origin. But the fibers of the nerves can be traced for a certain distance into the substance of the nervous center to some portion of the gray matter, which constitutes the deep or real origin of the nerve. The centrifugal or efferent nerve fibers originate in the nerve cells of the gray substance, the axis cylinder processes of these cells being prolonged to form fibers. In the case of the centripetal or afferent nerves, the fibers grow inward either from the nerve cells in the origins of special sense, for example the retina, or from nerve cells in the ganglia. Having entered the nerve center, they branch and send their ultimate twigs among the cells, without, however, uniting with them. Peripheral terminations of nerves. Nerve fibers terminate peripherally in various ways, and these may be conveniently studied in the sensory and motor nerves respectively. The terminations of the sensory nerves are dealt with in the section on sense organs. Motor nerves can be traced into either unstriped or striped muscular fibers. In the unstriped or involuntary muscles, the nerves are derived from the sympathetic and are composed mainly of non-medullated fibers. Near their terminations, they divide into numerous branches which communicate and form intimate plexuses. At the junction of the branches, small triangular nuclear bodies, ganglion cells, are situated. From these plexuses, minute branches are given off which divide and break up into the ultimate fibrillae of which the nerves are composed. These fibrillae course between the involuntary muscle cells and, according to Elischer, terminate on the surfaces of the cells opposite the nuclei in minute swellings. In the striped or voluntary muscle, the nerves supplying the muscular fibers are derived from the cerebrospinal nerves and are composed mainly of medullated fibers. The nerve, after entering the sheath of the muscle, breaks up into fibers or bundles of fibers which form plexuses and gradually divide until, as a rule, a single nerve fiber enters a single muscular fiber. Sometimes, however, if the muscular fiber be long, more than one nerve fiber enters it. Within the muscular fiber, the nerve terminates in a special expansor called by Kuhn, who first accurately described it as a motor end plate. The nerve fiber, on approaching the muscular fiber, suddenly loses its medullary sheath. The neurolemma becomes continuous with the sarcolemma of the muscle, and only the axis cylinder enters the muscular fiber. There it at once spreads out, ramifying like the roots of a tree immediately beneath the sarcolemma, and becomes embedded in a layer of granular matter, containing a number of clear oblong nuclei, the whole constituting an end plate, from which the contractile wave of the muscular fiber is said to start. Ganglia are small aggregations of nerve cells. They are found on the posterior roots of the spinal nerve, on the sensory roots of the trigeminal, facial, glossopharyngeal, and vagus nerves, and on the acoustic nerves. They are also found in connection with the sympathetic nerves. On section, they are seen to consist of a reddish-gray substance traversed by numerous white fibers. They vary considerably in form and size. The largest are found in the cavity of the abdomen, the smallest, not visible to the naked eye, exist in considerable numbers upon the nerves distributed to the different viscera. Each ganglion is invested by a smooth and firm, closely adhering, membranous envelope consisting of dense areolar tissue. This sheath is continuous with the perineurium of the nerves and sends numerous processes into the interior to support the blood vessels supplying the substance of the ganglion. In structure, all ganglia are essentially similar, consisting of the same structural elements, that is, nerve cells and nerve fibers. Each nerve cell has a nucleated sheath which is continuous with the neurolemma of the nerve fiber with which the cell is connected. The nerve cells in the ganglia of the spinal nerves are piriform in shape and have each a single process. A short distance from the cell, and while still within the ganglion, this process divides in a T-shaped manner, one limb of the crossbar turning into the Medulla Spinalis, the other limb passing outward to the periphery. In the sympathetic ganglia, the nerve cells are multipolar and each has one axis cylinder process and several dendrons. The axon emerges from the ganglion as a non-medullated nerve fiber. Similar cells are found in the ganglia connected with the trigeminal nerve, and these ganglia are therefore regarded as the cranial portions of the sympathetic system. The sympathetic nervous system includes those portions of the nervous mechanism in which a medullated nerve fiber from the central nervous system passes to a ganglion, sympathetic or peripheral, from which fibers, usually non-medullated, are distributed to such structures, for instance blood vessels, as are not under voluntary control. The spinal and sympathetic ganglia differ somewhat in the size and disposition of the cells and in the number of nerve fibers entering and leaving them. In the spinal ganglia, the nerve cells are much larger and for the most part collected in groups near the periphery, while the fibers, which are mostly medullated, traverse the central portion of the ganglion. Whereas in the sympathetic ganglia, the cells are smaller and distributed in irregular groups throughout the whole ganglion. The fibers are also irregularly scattered. Some of the entering ones are medullated, while many of the leaving the ganglia are non-medullated. Neuron theory. The nerve cell and its processes collectively constitute what is termed a neuron. And Walden formulated the theory that the nervous system is built up of numerous neurons, quote, anatomically and genetically independent of one another, end quote. According to this theory, the neuron theory, the processes of one neuron only come into contact and are never in direct continuity with those of other neurons, while impulses are transmitted from one nerve cell to another through these points of contact, the synapses. The synapse or synaptic membranes seems to allow nervous impulses to pass in one direction only, namely from the terminals of the axis cylinder to the dendrons. This theory is based on the following facts. That is, 1. Embryonic nerve cells, or neuroblasts, are entirely distinct from one another. 2. When nervous tissues are stained by the Golgi method, no continuity is seen even between neighboring neurons. And 3. When degenerative changes occur in nervous tissue, either as the result of disease or experiment, they never spread from one neuron to another, but are limited to the individual neurons or groups of neurons primarily affected. It must, however, be added that within the past few years, the validity of the neuron theory had been called into question by certain eminent histologists, who maintain that by the employment of a more delicate histologic method, minute fibrils can be followed from one nerve cell to another. Their existence, however, in the living is open to question. Mott and Marinesco made careful examination of living cells using even the ultramicroscope and agreed that neither Nissl bodies nor neurofibrils are present in the living state. For the present, we may look upon the neurons as the units or structural elements of the nervous system. All the neurons are present at birth, which are present in the adult. Their division ceases before birth. They are not all functionally active at birth, but gradually assume functional activity. There is no indication of any regeneration after the destruction of the cell body of any individual neurons. Fasciculi tracts, or fiber systems, are groups of axons having homologous origin and homologous distribution, as regards their collaterals, subdivisions, and terminals, and are often named in accordance with their origin and termination, the name of the nucleus or the location of the cell body from which the axon or fiber arises, preceding that of the nucleus or location of its termination. A given topographical area seldom represents a pure tract, as in most cases fibers of different systems are mixed. End of section 2. Section 3 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. Development of the Nervous System, Part 1. Development of the Nervous System. The entire nervous system is of ectodermal origin, and its first rudiment is seen in the neural groove, which extends along the dorsal aspect of the embryo. By the elevation and ultimate fusion of the neural folds, the groove is converted into the neural tube. The anterior end of the neural tube becomes expanded to form the 3 primary brain vesicles. The cavity of the tube is subsequently modified to form the ventricular cavities of the brain and the central canal of the Medulla Spinalis. From the wall of the nervous system elements and the neuroglia of the brain and Medulla Spinalis are developed. The medulla spinalis. At first, the wall of the neural tube is composed of a single layer of columnar ectodermal cells. Soon the side walls become thickened, while the dorsal and ventral parts remain thin and are named the roof and floor plates. A transverse section of the tube at this stage presents an oval outline, while its lumen has the appearance of a slit. The cells which constitute the wall of the tube proliferate rapidly, lose their cell boundaries, and form a syncytium. This syncytium consists at first of dense protoplasm with closely packed nuclei, but later it opens out and forms a looser meshwork, with the cellular strands arranged in a radiating manner from the central canal. 3 layers may now be defined: an internal or ependymal, an intermediate or mantle, and an external or marginal. The ependymal layer is ultimately converted into the ependyma of the central canal. The processes of its cells pass outward towards the periphery of the medulla spinalis. The marginal layer is devoid of nuclei and later forms a supporting framework for the white funiculi of the medulla spinalis. The mantle layer represents the whole of the future gray columns in the medulla spinalis. In it, the cells are differentiated into 2 sets, that is, spongioblasts, or young neuroglial cells, and germinal cells, which are the parents of the neuroblasts, or young nerve cells. The spongioblasts are at first connected to one another by filaments of the syncytium. In these, fibrils are developed so that as the neuroglial cells become defined, they exhibit their characteristic mature appearance with multiple processes extending from each cell. The germinal cells are large, round or oval, and first make their appearance between the ependymal cells on the sides of the central canal. They increase rapidly in number, so that by the 4th week they form an almost continuous layer on each side of the tube. No germinal cells are found in the roof or floor plates. The roof plate retains, in certain regions of the brain, its epithelial character. Elsewhere, its cells become spongioblasts. By subdivision, the germinal cells give rise to the neuroblast or young nerve cells, which migrate outward from the sides of the central canal into the mantle layer and neural crest, and at the same time become pear-shaped. The tapering part of the cell undergoes still further elongation and forms the axis cylinder of the cell. The lateral walls of the medulla spinalis continue to increase in thickness, and the canal widens out near its dorsal extremity and assumes a somewhat lozenge-shaped appearance. The widest part of the canal serves to subdivide the lateral wall of the neural tube into a dorsal or alar and a ventral or basal lamina, a subdivision which extends forward into the brain. At a later stage, the ventral part of the canal widens out, while the dorsal part is first reduced to a mere slit and then becomes obliterated by the approximation and fusion of its walls. The ventral part of the canal persists and forms the central canal of the adult medulla spinalis. The caudal end of the canal exhibits a conical expansion, which is known as the terminal ventricle. The ventral part of the mantle layer becomes thickened, and on cross-section appears as a triangular patch between the marginal and ependymal layers. This thickening is the rudiment of the anterior column of gray substance and contains many neuroblasts, the axis cylinders of which pass out through the marginal layer and form the anterior roots of the spinal nerves. The thickening of the mantle layer gradually extends in a dorsal direction and forms the posterior column of gray substance. The axons of many of the neuroblasts in the alar lamina run forward and cross in the floor plate to the opposite side of the medulla spinalis. These form the rudiment of the anterior white commissure. About the end of the 4th week, nerve fibers begin to appear in the marginal layer. The first to develop are the short intersegmental fibers from the neuroblast in the mantle zone, and the fibers of the dorsal nerve roots, which grow into the medulla spinalis from the cells of the spinal ganglia. By the 6th week, these dorsal root fibers form a well-defined oval bundle in the peripheral part of the alar lamina. This bundle gradually increases in size, and spreading towards the middle line, form the rudiment of the posterior funiculus. The long intersegmental fibers begin to appear about the 3rd month, and the cerebrospinal fibers about the 5th month. All nerve fibers are at first destitute of medullary sheaths. Different groups of fibers receive their sheaths at different times. The dorsal and ventral nerve roots about the 5th month, and the cerebrospinal fibers after the 9th month. by the growth of the anterior columns of gray substance, and by the increase in size of the anterior funiculi, a furrow is formed between the lateral halves of the cord anteriorly. This gradually deepens to form the anterior median fissure. The mode of formation of the posterior septum is somewhat uncertain. Many believe that it is produced by the growing together of the walls of the posterior part of the central canal, and by the development from its ependymal cells of a septum of fibrillated tissue which separates the future funicula gracilis. Up to the 3rd month of fetal life, the medulla spinalis occupies the entire length of the vertebral canal, and the spinal nerves pass outward at right angles to the medulla spinalis. From this time onward, the vertebral column grows more rapidly than the medulla spinalis, and the latter, being fixed above through its continuity with the brain, gradually assumes a higher position within the canal. By the 6th month, its lower end reaches only as far as the upper end of the sacrum. At birth, it is on a level with the 3rd lumbar vertebra, and in the adult with the lower border of the 1st or upper border of the 2nd lumbar vertebra. A delicate filament, the filum terminale, extends from its lower end as far as the coccyx. The spinal nerves. Each spinal nerve is attached to the medulla spinalis by an anterior or ventral and a posterior or dorsal root. The fibers of the anterior roots are formed by the axons of the neuroblasts which lie in the ventral part of the mantle layer. These axons grow out through the overlying marginal layer and become grouped to form the anterior nerve root. The fibers of the posterior roots are developed from the cells of the spinal ganglia. Before the neural groove is closed to form the neural tube, a ridge of ectodermal cells, the ganglion ridge or neural crest, appears along the prominent margin of each neural fold. When the folds meet in the middle line, the two ganglion ridges fuse and form a wedge-shaped area along the line of closure of the tube. The cells of this area proliferate rapidly opposite the primitive segments and then migrate in a lateral and ventral direction to the sides of the neural tube, where they ultimately form a series of oval-shaped masses, the future spinal ganglia. These ganglia are arranged symmetrically on the two sides of the neural tube and, except in the region of the tail, are equal in number to the primitive segments. The cells of the ganglia, like the cells of the mantle layer, are of two kinds, that is, spongioblasts and neuroblasts. The spongioblasts develop into the neuroglial cells of the ganglia. The neuroblasts are at first round or oval in shape, but soon assume the form of spindles, the extremities of which gradually elongate into central and peripheral processes. The central processes grow medialward, and becoming connected with the neural tube, constitute the fibers of the posterior nerve roots, while the peripheral processes grow lateralwards to mingle with the fibers of the anterior root in the spinal nerve. As development proceeds, the original bipolar form of the cells changes. The two processes become approximated until they ultimately arise from a single stem in a T-shaped manner. Only in the ganglia of the acoustic nerve is the bipolar form retained. More recent observers hold, however, that the T form is derived from the branching of a single process which grows out from the cell. The anterior or ventral and the posterior or dorsal nerve roots join immediately beyond the spinal ganglion to form the spinal nerve, which then divides into anterior, posterior, and visceral divisions. The anterior and posterior divisions proceed directly to the areas of distribution without further association with ganglion cells. The visceral distributions are distributed to the thoracic, abdominal, and pelvic viscera, to reach which they pass through the sympathetic trunk, and many of the fibers form arborizations around the ganglion cells of this trunk. Visceral branches are not given off from all the spinal nerves. They form two groups, that is, thoracolumbar from the first or second thoracic to the second or third lumbar nerves, and pelvic from the second and third or third and fourth sacral nerves. The brain. The brain is developed from the anterior end of the neural tube, which at an early period becomes expanded into three vesicles, the primary cerebral vesicles. These are marked off from each other by intervening constrictions and are named the forebrain or prosencephalon, the midbrain or mesencephalon, and the hindbrain or rhombencephalon, the last being continuous with the medulla spinalis. As the result of unequal growth of these different parts, three flexures are formed, and the embryonic brain becomes bent on itself in a somewhat zigzag fashion. The two earliest flexures are concave ventrally and are associated with corresponding flexures of the whole head. The first flexure appears in the region of the midbrain and is named the ventral cephalic flexure. By means of it, the forebrain is bent in a ventral direction around the anterior end of the notochord and foregut, with the result that the floor of the forebrain comes to lie almost parallel with that of the hindbrain. This flexure causes the midbrain to become for a time the most prominent part of the brain, since its dorsal surface corresponds with the convexity of the curve. The second bend appears at the junction of the hindbrain and medulla spinalis. This is termed the cervical flexure, and increases from the third to the end of the fifth week, when the hindbrain forms nearly a right angle with the medulla spinalis. After the fifth week, erection of the head takes place, and the cervical flexure diminishes and disappears. The third bend is named the pontine flexure, because it is found in the region of the future pons varioli. It differs from the other two in that A, its convexity is forward, and B, it does not affect the head. The lateral walls of the brain tube, like those of the medulla spinalis, are divided by internal furrows into alar, or dorsal and basal, or ventral laminae. The hindbrain, or rhombencephalon. The cavity of the hindbrain becomes the fourth ventricle. At the time when the ventral cephalic flexure makes its appearance, the length in the hindbrain exceeds the combined length of the other two vesicles. Immediately behind the midbrain, it exhibits a marked constriction, the isthmus rhombencephali, which is best seen when the brain is viewed from the dorsal aspect. From the isthmus, the anterior medullary velum and the superior peduncle of the cerebellum are formed. It is customary to divide the rest of the hindbrain into two parts, that is, an upper called the medencephalon, and a lower, the myelencephalon. The cerebellum is developed by a thickening of the roof, and the pons by a thickening of the floor and lateral walls of the metencephalon. The floor and lateral walls of the myelencephalon are thickened to form the medulla oblongata. Its roof remains thin and, retaining to a great extent its epithelial nature, is expanded in a lateral direction. Later, by the growth and backward extension of the cerebellum, the roof is folded inward towards the cavity of the fourth ventricle. It assists in completing the dorsal wall of this cavity, and is also invaginated to form the ependymal covering of its choroid plexuses. Above it is continuous with the posterior medullary velum, below with the obex and lingulae. The development of the medulla oblongata resembles that of the medulla spinalis, but at the same time exhibits one or two interesting modifications. On transverse section, the myelencephalon at an early stage is seen to consist of two lateral walls connected across the midline by floor and roof plates. Each lateral wall consists of an alar and a basal lamina, separated by an internal furrow, the remains of which are represented in the adult brain by the sulcus limitans and the rhomboid fossa. The contained cavity is more or less triangular in outline, the base being formed by the roof plate, which is thin and greatly expanded transversely. Pear-shaped neuroblasts are developed in the alar and basal lamina, and their narrow stalks are elongated to form the axis cylinder of the nerve fibers. Opposite the furrow, or boundary between the alar and basal lamina, a bundle of nerve fibers attaches itself to the outer surface of the alar lamina. This is named the Tractus Solitarius, and is formed by the sensory fibers of the glossopharyngeal and vagus nerves. It is the homologue of the oval bundle seen in the Medulla Spinalis, and, like it, is developed by an ingrowth of fibers from the ganglia of the neural crest. At first it is applied to the outer surface of the alar lamina, but it soon becomes buried owing to the growth over it of the neighboring parts. By the fifth week, the dorsal part of the alar lamina bends in a lateral direction along its entire length to form what is termed the rhombic lip. Within a few days, this lip becomes applied to and unites with the outer surface of the main part of the alar lamina, and so covers in the Tractus Solitarius and also the spinal root of the trigeminal nerve. The nodulus and flocculus of the cerebellum are developed from the rhombic lip. Neuroblasts accumulate in the mantle layer. Those in the basal lamina correspond with the cells in the anterior gray column of the medulla spinalis, and like them, give origin to motor nerve fibers. In the medulla oblongata they are, however, arranged in groups or nuclei instead of forming a continuous column. From the alar lamina and its rhombic lip, neuroblasts migrate into the basal lamina and become aggregated to form the olivary nuclei, while many send their axis cylinders through the floor plate to the opposite side and thus constitute the rudiment of the raphe of the medulla oblongata. By means of this thickening of the ventral portion, the motor nuclei are buried deeply in the interior and, in the adult, are found close to the rhomboid fossa. This is still further accentuated, a. by the development of the pyramids, which are formed about the 4th month by the downward growth of the motor fibrils from the cerebral cortex, and b. by the fibers which pass to and from the cerebellum. On the rhomboid fossa, a series of 6 temporary furrows appear. These are termed the Rhombic Grooves. They bear a definite relationship to certain of the cranial nerves. Thus, from before backward, the 1st and 2nd grooves overlie the nucleus of the trigeminal, the 3rd the nucleus of the facial, the 4th that of the abducent, the 5th that of the glossopharyngeal, and the 6th that of the vagus. The pons is developed from the ventrolateral wall of the metencephalon by a process similar to that which has been described for the medulla oblongata. The cerebellum is developed in the roof of the anterior part of the hindbrain. The alar lamina of this region become thickened to form 2 lateral plates, which soon fuse in the middle line and produce a thick lamina which roofs the upper part of the cavity of the hindbrain vesicle. This constitutes the rudiment of the cerebellum, the outer surface of which is originally smooth and convex. The fissures of the cerebellum appear first in the vermis and floccular region, and traces of them are found during the 3rd month. The fissures on the cerebellar hemispheres do not appear until the 5th month. The primitive fissures are not developed in the order of their relative size in the adult. Thus, the horizontal sulcus in the 5th month is merely a shallow groove. The best marked of the early fissures are a. the fissura prima between the developing culmen and declive, and b. the fissura secunda between the future pyramid and uvula. The flocculus and nodule develop from the rhombic lip, and are therefore recognizable as separate portions before any of the other cerebellar lobules. The groove produced by the bending over the rhombic lip is here known as the floccular fissure. When the 2 lateral walls fuse, the right and left floccular fissures join in the middle line, and their central part becomes the post-nodular fissure. On the ventricular surface of the cerebellar lamina, a transverse furrow, the incisura fastigii, appears, and deepens to form the tent-like recess of the roof of the 4th ventricle. The rudiment of the cerebellum at first projects in a dorsal direction, but by the backward growth of the cerebrum, it is folded downward and somewhat flattened, and the thin roof plate of the 4th ventricle, originally continuous with the posterior border of the cerebellum, is projected inward towards the cavity of the ventricle. End of section 3. Section 4 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. Development of the Nervous System Part 2. The midbrain, or mesencephalon. The midbrain exists for a time as a thin-walled cavity of some size, and is separated from the isthmus rhombencephali behind and from the forebrain in front by slight constrictions. Its cavity becomes relatively reduced in diameter and forms the cerebral aqueduct of the adult brain. Its basal laminae increase in thickness to form the cerebral peduncles, which are at first of small size, but rapidly enlarge after the 4th month. The neuroblasts of these laminae are grouped in relation to the sides and floor of the cerebral aqueduct, and constitute the nuclei of the oculomotor and trochlear nerves, and of the mesencephalic root of the trigeminal nerve. By a similar thickening process, its alar laminae are developed into the quadrigeminal lamina. The dorsal part of the wall for a time undergoes expansion and presents an internal median furrow and a corresponding external ridge. These, however, disappear and the latter is replaced by a groove. Subsequently, two oblique furrows extend medialward and backward, and the thickened lamina is thus subdivided into the superior and inferior colliculi. The forebrain, or prosencephalon. A transverse section of the early forebrain shows the same parts as are displayed in similar sections of the Medulla Spinalis and Medulla Oblongata. That is, a pair of thick lateral walls connected by thin floor and roof plates. Moreover, each lateral wall exhibits a division into dorsal or alar and a ventral or basal lamina separated internally by a furrow termed the sulcus of Monro. This sulcus ends anteriorly at the medial end of the optic stalk, and in the adult brain is retained as a slight groove extending backward from the interventricular foramen to the cerebral aqueduct. At a very early period, in some animals before the closure of the cranial part of the neural tube, two lateral diverticula, the optic vesicles, appear, one on either side of the forebrain. For a time, they communicate with the cavity of the forebrain by relatively wide openings. The peripheral parts of the vesicles expand, while the proximal parts are reduced to tubular stalks, the optic stalks. The optic vesicles give rise to the retina and the epithelium on the back of the ciliary body and iris. The optic stalk is invaded by nerve fibers to form the optic nerve. The forebrain then grows forward, and from the alar lamina of this front portion, the cerebral hemispheres originate as diverticula, which rapidly expand to form two large pouches, one on either side. The cavities of these diverticula are rudiments of the lateral ventricles. They communicate with the median part of the forebrain cavity by relatively wide openings, which ultimately form the interventricular foramen. The median portion of the wall of the forebrain vesicle consists of a thin lamina, the lamina terminalis, which stretches from the interventricular foramen to the recess at the base of the optic stalk. The anterior part of the forebrain, including the rudiments of the cerebral hemispheres, is named the telencephalon, and its posterior portion is termed the diencephalon. Both of these contribute to the formation of the third ventricle. The diencephalon. From the alar lamina of the diencephalon, the thalamus, metathalamus, and epithalamus are developed. The thalamus arises as a thickening which involves the anterior two-thirds of the alar lamina. The two thalami are visible for a time on the surface of the brain, but are subsequently hidden by the cerebral hemispheres which grow backward over them. The thalami extend medialward and gradually narrow the cavity between them into a slit-like aperture, which forms the greater part of the third ventricle. Their medial surfaces ultimately adhere in part to each other, and their intermediate mass of the ventricle is developed across the area of contact. The metathalamus comprises the geniculate bodies, which originate as slight outward bulgings of the alar lamina. In the adult, the lateral geniculate body appears as an eminence on the lateral part of the posterior end of the thalamus, while the medial is situated on the lateral aspect of the midbrain. The epithalamus includes the pineal body, the posterior commissure, and the trigone habenula. The pineal body arises as an upward evagination of root plate immediately in front of the midbrain. This evagination becomes solid with the exception of its proximal part, which persists as the recessus pinealis. In lizards, the pineal evagination is elongated into a stalk, and its peripheral extremity is expanded into a vesicle in which a rudimentary lens and retina are formed. The stalk becomes solid and nerve fibers make their appearance in it, so that in these animals the pineal body forms a rudimentary eye. The posterior commissure is formed by the ingrowth of fibers into the depression behind and below the pineal evagination, and the trigonum habenulae is developed in front of the pineal recess. From the basal laminae of the diencephalon, the pars mammillaris hypothalami is developed. This comprises the corpora mammillaria and the posterior part of the tuber cinerum. The corpora mammillaria arise as a single thickening, which comes divided into two by a median furrow during the third month. The roof plate of the diencephalon, in front of the pineal body, remains thin and epithelial in character, and is subsequently invaginated by the choroid plexus of the third ventricle. The telencephalon. This consists of a median portion and two lateral diverticula. The median portion forms the anterior part of the cavity of the third ventricle, and is closed below and in front by the lamina terminalis. The lateral diverticula consist of outward pouchings of the alar laminae. The cavities represent the lateral ventricles, and their walls become thickened to form the nervous matter of the cerebral hemispheres. The roof plate of the telencephalon remains thin, and is continuous in front with the lamina terminalis and behind with the roof plate of the diencephalon. In the basal lamina and the foreplate, the pars optica hypothalami is developed. This comprises the anterior part of the tuberous cinerum, the infundibulum, and the posterior lobe of the hypophysis and the optic chiasma. The anterior part of the tuberous cinerum is developed from the posterior part of the floor of the telencephalon. The infundibulum and posterior lobe of the hypophysis arises as a downward diverticulum from the floor. The most dependent part of the diverticulum becomes solid and forms the posterior lobe of the hypophysis. The anterior lobe of the hypophysis is developed from a diverticulum of the ectodermal lining of the stomadeum. The optic chiasma is formed by the meeting and partial decussation of the optic nerves, which subsequently grow backward as the optic tracts and end in the diencephalon. The cerebral hemispheres arise as diverticula of the alar lamina of the telencephalon. They increase rapidly in size and ultimately overlap the structures developed from the mid and hindbrains. This great expansion of the hemispheres is a characteristic feature of the brains of mammals and attains its maximum development in the brain of man. Elliott Smith divides each cerebral hemisphere into three fundamental parts, that is, the rhinencephalon, the corpus striatum, and the neopallium. The rhinencephalon represents the oldest part of the telencephalon and forms almost the whole of the hemisphere in fishes, amphibians, and reptiles. In man, it is feebly developed in comparison with the rest of the hemisphere, and comprises the following parts. That is, the olfactory lobe, consisting of the olfactory tract and bulb and trigonum olfactorium, the anterior perforated substance, the septum pellucidum, the subcallosal, supracallosal, and dentate gyri, the fornix, the hippocampus, and the uncus. The rhinencephalon appears as longitudinal elevation with a corresponding internal furrow on the undersurface of the hemisphere close to the lamina terminalis. It is separated from the lateral surface of the hemisphere by a furrow, the external rhinal fissure, and is continuous behind with that part of the hemisphere which will ultimately form the anterior end of the temporal lobe. The elevation becomes divided by a groove into an anterior and a posterior part. The anterior grows forward as a hollow stalk, the lumen of which is continuous with the anterior part of the ventricular cavity. During the third month, the stalk becomes solid and forms the rudiment of the olfactory bulb and tract. A strand of gelatinous tissue in the interior of the bulb indicates the position of the original cavity. From the posterior part, the anterior perforated substance and the piriform lobe are developed. At the beginning of the fourth month, the latter forms a curved elevation continuous behind with the medial surface of the temporal lobe and consisting from before backwards of the gyrus olfactorius lateralis, gyrus ambiens, and gyrus semilunaris, parts which in the adult brain are represented by the lateral root of the olfactory tract and the uncus. The position and connections of the remaining portions of the rhinencephalon are described with the anatomy of the brain. The corpus striatum appears in the fourth week as a triangular thickening of the floor of the telencephalon between the optic recess and the interventricular foramen, and continues behind with the thalamic part of the diencephalon. It increases in size, and by the second month is seen as a swelling in the floor of the furtio lateral ventricle. This swelling reaches as far as the posterior end of the primitive hemisphere, and when this part of the hemisphere grows backward and downward to form the temporal lobe, the posterior part of the corpus striatum is carried into the roof of the inferior horn of the ventricle, where it is seen as the tail of the caudate nucleus in the adult brain. During the 4th and 5th month, the corpus striatum becomes incompletely subdivided by the fibers of the internal capsule into two masses, an inner, the caudate nucleus, and an outer, the lentiform nucleus. In front, the corpus striatum is continuous with the anterior perforated substance. Laterally, it is confluent for a time with that portion of the wall of the vesicle which is developed into the insula, but this continuity is subsequently interrupted by the fibers of the external capsule. The neopallium forms the remaining and by far the greater part of the cerebral hemisphere. It consists as an early stage of a relatively large, more or less hemispherical cavity, the primitive lateral ventricle, enclosed by a thin wall from which the cortex of the hemisphere is developed. The vesicle expands in all directions, but more especially upward and backward, so that by the 3rd month, the hemispheres cover the diencephalon, by the 6th they overlap the midbrain, and by the 8th the hindbrain. The median lamina uniting the two hemispheres does not share in their expansion, and thus the hemispheres are separated by a deep cleft, the forerunner of the longitudinal fissure, and this cleft is occupied by a septum of mesodermal tissue which constitutes the primitive falx cerebri. Coincidentally with the expansion of the vesicle, its cavity is drawn out into 3 prolongations which represent the horn of the future lateral ventricle. The hinder end of the vesicle is carried downward and forward and forms the inferior horn. The posterior horn is produced somewhat later and associated with the backward growth of the occipital lobe of the hemisphere. The roof plate of the primitive forebrain remains thin and of an epithelial character. It is invaginated into the lateral ventricle along the median wall of the hemisphere. This invagination constitutes the choroidal fissure and extends from the interventricular foramen to the posterior end of the vesicle. Mesodermal tissue, continuous with that of the primitive falx cerebri and carrying blood vessels with it, spreads between the two layers of the invaginated fold and forms the rudiment of the tela choroidea. The margins of the tela become highly vascular and form the choroid plexuses, which for some months almost completely fill the ventricular cavities. The tela at the same time invaginates the epithelial roof of the diencephalon to form the choroid plexuses of the 3rd ventricle. By the downward and forward growth of the posterior end of the vesicle to form the temporal lobe, the choroidal fissure finally reaches from the interventricular foramen to the extremity of the inferior horn of the ventricle. Parallel with, but above and in front of the choroidal fissure, the median wall of the cerebral vesicle becomes folded outward and gives rise to the hippocampal fissure on the median surface and to a corresponding elevation, the hippocampus within the ventricular cavity. The gray or ganglionic covering of the wall of the vesicle ends at the inferior margin of the fissure, is a thickened edge. Beneath this, the marginal reticular layer, future white substance, is exposed, and its lower thinned edge is continuous, with the epithelial invagination covering the choroid plexus. As a result of the later downward and forward growth of the temporal lobe, the hippocampal fissure and the parts associated with it extend from the interventricular foramen to the end of the inferior horn of the ventricle. The thickened edge of gray substance becomes the gyrus dentatus, the fasciola cinerea, and the supra and subcallosal gyri, while the free edge of the white substance forms the fimbria hippocampi and the body and crus of the fornix. The corpus callosum is developed within the arch of the hippocampal fissure, and the upper part of the fissure forms in the adult brain the callosal fissure on the medial surface of the hemisphere. The commissures. The development of the posterior commissure has already been referred to. The great commissures of the hemisphere, that is, the corpus callosum, the fornix, and the anterior commissures, arise from the lamina terminalis. About the 4th month, a small thickening appears in this lamina immediately in front of the interventricular foramen. The lower part of this thickening is soon constricted off, and fibers appear in it to form the anterior commissure. The upper part continues to grow with the hemispheres and is evaded by 2 sets of fibers. Transverse fibers, extending between the hemispheres, pass into its dorsal part, which is now differentiated as the corpus callosum. In rare cases, the corpus callosum is not developed. Into the ventral part, longitudinal fibers from the hippocampus pass to the lamina terminalis and through that structure to the corpora mammillaria. These fibers constitute the fornix. A small portion lying anterior-inferiorly between the corpus callosum and the fornix is not invaded by the commissural fibers. It remains thin, and later a cavity, the cavity of the septum pellucidum, forms in its interior. Fissures and sulci. The outer surface of the cerebral hemispheres is at first smooth, but later it exhibits a number of elevations or convolutions, separated from each other by fissures and sulci, most of which make their appearance during the 6th or 7th month of fetal life. The term fissure is applied to such grooves as involve the entire thickness of the cerebral wall, and thus produce corresponding eminences in the ventricular cavity, while the sulci affect only the superficial part of the wall, and therefore leave no impression in the ventricle. The fissures comprise the choroidal and hippocampal already described, and two others, that is, the calcarine and collateral, which produce the swellings known respectively as the calcar avus and the collateral eminence in the ventricular cavity. Of the sulci, the following may be referred to, that is, the central sulcus, fissure of Rolando, which is developed in two parts, the intraparietal sulcus in 4 parts, and the cingulate sulcus in 2 or 3 parts. The lateral cerebral or sylvian fissures differs from all the other fissures in its mode of development. It appears about the 3rd month as a depression, the sylvian fossa, on the lateral surface of the hemisphere. This fossa corresponds with the position of the corpus striatum, and its floor is molded to form the insula. The intimate connection which exists between the cortex of the insula and the subjacent corpus striatum prevents this part of the hemisphere wall from expanding at the same rate as the portions which surround it. The neighboring parts of the hemisphere therefore gradually grow over and cover in the insula, and constitute the temporal, parietal, frontal, and orbital opercula of the adult brain. The frontal and orbital opercula are the last to form, but by the end of the 1st year after birth, the insula is completely submerged by the approximation of the opercula. The fissures separating the opposed margins of the opercula constitute the composite lateral cerebral fissure. If a section across the wall of the hemisphere about the 6th week be examined microscopically, it will be found to consist of a thin marginal or reticular layer, a thick ependymal layer, and a thin intervening mantle layer. Neuroblasts from the ependymal and mantle layers migrate into the deep part of the marginal layer and form the cells of the cerebral cortex. The nerve fibers, which form the underlying white substance of the hemispheres, consist at first of outgrowth from the cells of the corpora striata and the thalami. Later, the fibers from the cells of the cortex are added. Medullation of these fibers begins about the time of birth and is continuous until puberty. A summary of these parts, derived from the brain vesicles, is given in the following table. The hindbrain or rhombencephalon give rise to the myelencephalon, medencephalon, and isthmus rhombencephali. The myelencephalon gives rise to the medulla oblongata and lower part of the 4th ventricle, the medencephalon to the pons, cerebellum, and intermediate part of the 4th ventricle, and the isthmus rhombencephali, the anterior medullary velum, brachium conjunctiva cerebelli, and upper part of the 4th ventricle. The midbrain, or mesencephalon, gives rise to the cerebral peduncles, lamina quadrigemina, and cerebral aqueduct. The forebrain, or prosencephalon, gives rise to the diencephalon and telencephalon. The diencephalon gives rise to the thalamus, metathalamus, epithalamus, pars mammillaris hypothalami, and the posterior part of the 3rd ventricle. The telencephalon gives rise to the anterior part of the 3rd ventricle, pars optica hypothalami, cerebral hemisphere's lateral ventricles, and interventricular foramina. The cranial nerves. With the exception of the olfactory, optic, and acoustic nerves, which will be especially considered, the cranial nerves are developed in a similar manner to the spinal nerves. The sensory or afferent nerves are derived from the cells of the ganglion rudiments of the neural crest. The central processes of these cells grow into the brain and form the roots of the nerves, while the peripheral processes extend outward and constitute their fibers of distribution. It has been seen, in considering the development of the medulla oblongata, that the Tractus Solitarius, derived from the fibers which grow inward from the ganglion rudiments of the glossopharyngeal and vagus nerves, is the homologue of the oval bundle in the cord, which had its origin in the posterior nerve roots. The motor or efferent nerves arise as outgrowths of the neuroblasts situated in the basal lamina of the mid and hindbrain. While, however, the spinal motor nerve roots arise in one series from the basal lamina, the cranial motor nerves are grouped into two sets, according as they spring from the medial or lateral parts of the basal lamina. To the former set belong the ocular motor, trochlear, abducent, and hypoglossal nerves. To the latter, the accessory and motor fibers of the trigeminal, facial, glossopharyngeal, vagus nerves. End of section 4. Section 5 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. The Spinal Cord, Part 1. The Medulla Spinalis, or spinal cord. The medulla spinalis, or spinal cord, forms the elongated, nearly cylindrical part of the central nervous system which occupies the upper two-thirds of the vertebral canal. Its average length in the male is about 45 centimeters, in the female from 42 to 43 centimeters, while its weight amounts to about 30 grams. It extends from the level of the upper border of the atlas to that of the lower border of the first or upper border of the second lumbar vertebra. Above, it is continuous with the brain. Below, it ends in a conical extremity, the conus medullaris, from the apex of which a delicate filament, the filum terminale, descends as far as the first segment of the coccyx. The position of the medulla spinalis varies with the movements of the vertebral column, its lower extremity being drawn slightly upward when the column is flexed. It also varies at different periods of life. Up to the 3rd month of fetal life, the medulla spinalis is as long as the vertebral canal, but from this stage onward, the vertebral column elongates more rapidly than the medulla spinalis, so that by the end of the 5th month, the medulla spinalis terminates at the base of the sacrum, and at birth, about the 3rd lumbar vertebra. The medulla spinalis does not fill the part of the vertebral canal in which it lies. It is ensheathed by 3 protective membranes, separated from each other by 2 concentric spaces. The 3 membranes are named from without inward, the Dura Mater, the arachnoid, and the Pia Mater. The Dura Mater is a strong fibrous membrane which forms a wide tubular sheath. This sheath extends below the termination of the medulla spinalis and ends at a point cul-de-sac at the level of the lower border of the second sacral vertebra. The Dura Mater is separated from the wall of the vertebral canal by the epidural cavity, which contains a quantity of loose areolar tissue and a plexus of veins. Between the Dura Mater and the subjacent arachnoid is a capillary interval, the subdural cavity, which contains a small quantity of fluid, probably of the nature of lymph. The arachnoid is a thin, transparent sheath separated from the Pia Mater by a comparatively wide interval, the subarachnoid cavity, which is filled with cerebrospinal fluid. The Pia Mater closely invests the medulla spinalis and sends delicate septae into its substance. A narrow band, the ligamentum denticulatum, extends along each of the lateral surfaces and is attached by a series of pointed processes to the inner surfaces of the Dura Mater. 31 pairs of spinal nerves spring from medulla spinalis, each nerve having an anterior or ventral and a posterior or dorsal root, the latter being distinguished by the presence of an oval swelling, the spinal ganglion, which contains numerous nerve cells. Each root consists of several bundles of nerve fibers, and at its attachment extends for some distance along the side of the medulla spinalis. The pairs of spinal nerves are grouped as follows: cervical 8, thoracic 12, lumbar 5, sacral 5, coccygeal 1, and for convenience of description, the medulla spinalis is divided into cervical, thoracic, lumbar, and sacral regions, corresponding with the attachments of the different groups of nerves. Although no trace of transverse segmentation is visible on the surface of the medulla spinalis, it is convenient to regard it as being built up in a series of superimposed spinal segments, or neuromeres, each of which has a length equivalent to the extent of attachment of a pair of spinal nerves. Since the extent of attachment of the successive pairs of nerves varies in different parts, it follows that the spinal segments are of varying lengths. Thus, in the cervical region, they average about 13 millimeters, in the mid-thoracic region about 26 millimeters, while in the lumbar and sacral region they diminish rapidly from about 15 millimeters at the level of the first pair of lumbar nerves to about 4 millimeters opposite the attachment of the lower sacral nerves. As a consequence of the relevant inequality in the rates of growth of the medulla spinalis and vertebral column, the nerve roots, which in the early embryo pass transversely outward to reach their respective intervertebral foramina, become more and more oblique in direction from above downward, so that the lumbar and sacral nerves descend almost vertically to reach their points of exit. From the appearance these nerves present at the attachment to the medulla spinalis, and from their great length, they are collectively termed the cauda equina. The filum terminale is the delicate filament, about 20 centimeters in length, prolonged downward from the apex of the conus medullaris. It consists of two parts, an upper and a lower. The upper part, or filum terminale internum, measures about 15 centimeters in length and reaches as far as the lower border of the second sacral vertebra. It is contained within the tubular sheath of Dura Mater and is surrounded by the nerves forming the cauda equina, from which it can be readily recognized by its bluish-white color. The lower part, or filum terminale externum, is closely invested by and is adherent to the Dura Mater. It extends downward from the apex of the tubular sheath and is attached to the back of the first segment of the coccyx. The filum terminale consists mainly of fibrous tissue, continuous above with that of the pia mater. Adhering to its outer surface, however, are a few strands of nerve fibers which probably represent rudimentary second and third coccygeal nerves. Further, the central canal of the medulla spinalis extends downward into it for 5 or 6 centimeters. Enlargements. The medulla spinalis is not quite cylindrical, being slightly flattened from before backward. It also presents two swellings or enlargements, an upper or cervical and a lower or lumbar. The cervical enlargement is the more pronounced and corresponds with the attachment of the large nerves which supply the upper limbs. It extends from about the third cervical to the second thoracic vertebra, its maximal circumference, about 38 millimeters, being on a level with the attachment of the sixth pair of cervical nerves. The lumbar enlargement gives attachment to the nerves which supply the lower limbs. It commences about the level of the ninth thoracic vertebra and reaches its maximum circumference of about 33 millimeters opposite the last thoracic vertebra, below which it tapers rapidly into the conus medullaris. Fissures and sulci. An anterior median fissure and a posterior median sulcus incompletely divide the medulla spinalis into two symmetrical parts, which are joined across the midline by a commissural band of nervous matter. The anterior median fissure, fissura mediana anterior, has an average depth of about 3 millimeters, but this is increased in the lower part of the medulla spinalis. It contains a double fold of pia mater, and its floor is formed by a transverse band of white substance, the anterior white commissure, which is perforated by blood vessels on their way to or from the central part of the medulla spinalis. The posterior median sulcus, sulcus medianus posterior, is very shallow. From it, a septum of neuroglia reaches rather more than halfway into the substance of the medulla spinalis. This septum varies in depth from 4 to 6 millimeters, but diminishes considerably in the lower part of the medulla spinalis. On either side of the posterior median sulcus, and at a short distance from it, the posterior nerve roots are attached along a vertical furrow named the posterior lateral sulcus. The portion of the medulla spinalis which lies between this and the posterior median sulcus is named the posterior funiculus. In the cervical and upper thoracic regions, this funiculus presents a longitudinal furrow, the posterior intermediate sulcus. This marks the position of a septum which extends into the posterior funiculus and subdivides it into two fasciculi, a medial, named the fasciculus gracilis, tract of Gall, and a lateral, the fasciculus cuneatus, tract of Burdach. The portion of the medulla spinalis which lies in front of the posterior lateral sulcus is termed the anterior lateral region. The anterior nerve roots, unlike the posterior, are not attached in linear series, and their position of exit is not marked by a sulcus. They arise by separate bundles which spring from the anterior column of the gray substance and, passing forward through the white substance, emerge over an area of some slight width. The most lateral of these bundles is generally taken as a dividing line which separates the anterior lateral region into two parts. That is, an anterior funiculus between the anterior median fissure and the most lateral of the anterior nerve roots, and a lateral funiculus between the exit of these roots and the posterior lateral sulcus. In the upper part of the cervical region, a series of nerve roots passes outward through the lateral funiculus of the medulla spinalis. These unite to form the spinal portion of the accessory nerve, which runs upward and enters the cranial cavity through the foramen magnum. The internal structure of the medulla spinalis. On examining a transverse section of the medulla spinalis, it is seen to consist of gray and white nervous substance, the former being enclosed within the latter. Gray substance. Substantia grisea centralis. The gray substance consists of two symmetrical portions, one in each half of the medulla spinalis. These are joined across the middle line by a transverse commissure of gray substance, through which runs a minute canal, the central canal, just visible to the naked eye. In a transverse section, each half of the gray substance is shaped like a comma or crescent, the concavity of which is directed laterally, and these, together with the intervening gray commissure, present the appearance of the letter H. An imaginary coronal plane through the central canal serves to divide each crescent into an anterior or ventral and a posterior or dorsal column. The anterior column, columna anterior, anterior cornu, directed forward is broad and of a rounded or quadrangular shape. Its posterior part is termed the base and its anterior part the head, but these are not differentiated from each other by any well-defined constriction. It is separated from the surface of the medulla spinalis by a layer of white substance, which is traversed by the bundles of the anterior nerve roots. In the thoracic region, the posterior lateral part of the anterior column projects lateralwards as a triangular field, which is named the lateral column. Columna lateralis, the lateral cornu. The posterior column, columna posterior, posterior cornu, is long and slender and is directed backwards and lateralwards. It reaches almost as far as the posterior lateral sulcus, from which it is separated by a thin layer of white substance, the tract of Lissauer. It consists of a base, directly continuous with the base of the anterior horn, and a neck or slightly constricted portion, which is succeeded by an oval or fusiform area termed the head, of which the apex approaches the posterior lateral sulcus. The apex is capped by a V-shaped or crescentic mass of translucent gelatinous neuroglia, termed the substantia gelatinosa of Rolando, which contains both neuroglial cells and small nerve cells. Between the anterior and posterior columns, the gray substance extends as a series of processes into the lateral funiculus to form a network called the formatio reticularis. The quantity of gray substance, as well as the form which it presents on transverse section, varies markedly at different levels. In the thoracic region, it is small, not only in amount, but relatively to the surrounding white substance. In the cervical and lumbar enlargements, it is greatly increased. In the latter, and especially in the conus medullaris, its proportion to the white substance is greatest. In the cervical region, its posterior column is comparatively narrow, while its anterior is broad and expanded. In the thoracic region, both columns are attenuated, and the lateral column is evident. In the conus medullaris, the gray substance assumes the form of two oval masses, one in each half of the cord, connected together by a broad gray commissure. The central canal, Canalus centralis, runs throughout the entire length of medulla spinalis. The portion of gray substance in front of the canal is named the anterior gray commissure, that behind it, the posterior gray commissure. The former is thin and is in contact anteriorly with the anterior white commissure. It contains a couple of longitudinal veins, one on either side of the middle line. The posterior gray commissure reaches from the central canal to the posterior median septum and is thinnest in the thoracic region and thickest in the conus medullaris. The central canal is continued upward through the lower part of the medulla oblongata and opens into the fourth ventricle of the brain. Below, it reaches for a short distance into the filum terminale. In the lower part of the conus medullaris, it exhibits a fusiform dilatation, the terminal ventricle. This has a vertical measurement of from 8 to 10 millimeters, is triangular on cross-section with its base directed forward, and tends to undergo obliteration after the age of 40 years. Throughout the cervical and thoracic regions, the central canal is situated in the anterior third of the medulla spinalis. In the lumbar enlargement, it is near the middle, and in the conus medullaris, it approaches the posterior surface. It is filled with cerebrospinal fluid and lined by ciliated columnar epithelium, outside of which is an encircling band of gelatinous substance, the substantia gelatinosa centralis. This gelatinous substance consists mainly of neuroglia, but contains a few nerve cells and fibers. It is traversed by processes from the deep ends of the columnar ciliated cells which line the central canal. End of section 5. Section number 6 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Spinal Cord Part 2. Structure of the gray substance. The gray substance consists of numerous nerve cells and nerve fibers held together by neuroglia. Throughout the greater part of the gray substance, the neuroglia presents the appearance of a sponge-like network, but around the central canal and on the apices of the posterior columns, it consists of the gelatinous substance already referred to. The nerve cells are multipolar and vary greatly in size and shape. They consist of 1. Motor cells of large size, which are situated in the anterior horn, and are especially numerous in the cervical and lumbar enlargements. The axons of most of these cells pass out from the anterior nerve roots, but before leaving the white substance, they frequently give off collaterals, which re-enter and ramify in the gray substance. 2. Cells of small or medium size, whose axons pass into the white matter, where some pursue an ascending and others a descending course, but most of them divide in a T-shaped manner into descending and ascending processes. They give off collaterals which enter and ramify into the gray substance, and the terminations of the axons behave in a similar manner. The lengths of these axons vary greatly. Some are short and pass only between the joining spinal segments, while others are longer and connect more distant segments. These cells and their processes constitute a series of association or intersegmental neurons, which link together the different parts of the medulla spinalis. The axons of most of these cells are confined to the side of the medulla spinalis in which the nerve cells are situated, but some cross to the opposite side through the anterior commissures and are termed crossed commissural fibers. Some of these latter end directly in the gray substance, while others enter the white substance and ascend or descend in it for varying distances, before finally terminating in the gray substance. 3. Cells of the type 2 of Golgi, limited for the most part to the posterior column, are found also in the substantia gelatinosa of Rolando. Their axons are short and entirely confined to the gray substance, in which they break up into numerous fine filaments. Most of the nerve cells are arranged in longitudinal columns and appear as groups on transverse section. Nerve cells in the anterior column. The nerve cells in the anterior column are arranged in columns of varying length. The longest occupies the medial part of the anterior column and is named the anteromedial column. It is well marked in C4, C5, again from C8 to L4. It disappears in L5 and S1, but is well marked in S2, S3, and S4. Open parens, Bruce, close parens, see footnote. Footnote, Topical Atlas of the Spinal Cord, 1901. End footnote. Behind it is a dorsomedial column of small cells which is not represented in L5, S1, S2, nor below S4. Its axons probably pass into the dorsal rami of the spinal nerves to supply the dorsal musculature of the spinal column. In the cervical and lumbar enlargements, where the anterior column is expanded in a lateral direction, the following additional columns are present, that is, A. Anterolateral, which consists of two groups, one in C4, C5, C6, the other in C6, C7, C8 in the cervical enlargement, and of a group from L2 to S2 in the lumbosacral enlargement. B. Posterior lateral in the lower 5 cervical, lower 4 lumbar, and upper 3 sacral segments. C. Posterior lateral in the last cervical, 1st thoracic, and upper 3 sacral segments. And D. Acentral in the lower 4 lumbar and upper 2 sacral segments. These cell groups are evidently related to the nerve roots of the brachial and sacral plexuses and supply fibers to the muscles of the arm and leg. Throughout the base of the anterior column are scattered solitary cells, the axons of some of which form cross-commissural fibers, while others constitute the motor fibers of the posterior nerve roots. Footnote: Lenhasek and Cahal found that in the chick embryo, the axons of a few of these nerve cells passed backwards through the posterior column and emerged as the motor fibers of the posterior nerve roots. These fibers are said to control the peristaltic movement of the intestine. Their presence in man has not yet been determined. End footnote. Nerve cells in the lateral column. These form a column which is best marked where the lateral gray column is differentiated, that is, in the thoracic region, but it can be traced throughout the entire length of Medulla Spinalis in the form of groups of small cells which are situated in the anterior part of the formatio reticularis. In the upper part of the cervical region and lower part of the Medulla Oblongata, as well as in the 3rd and 4th sacral segments, this column is again differentiated. In the medulla, it is known as the lateral nucleus. The cells of this column are fusiform or star-shaped and of a medium size. The axons of some of them pass into the anterior nerve roots by which they are carried to the sympathetic nerve. They constitute the white rami and are sympathetic or visceral efferent fibers. They are also known as preganglionic fibers of the sympathetic system. The axons of others pass into the anterior and lateral funiculi, where they become longitudinal. Nerve cells in the posterior column. 1. The dorsal nucleus. Nucleus dorsalis, column of Clark. Occupies the medial part of the base of the posterior column and appears on the transverse section as a well-defined oval area. It begins below at the level of the 2nd or 3rd lumbar nerve and reaches its maximum size opposite the 12th thoracic nerve. Above the level of the 9th thoracic nerve, its size diminishes and the column ends opposite the last cervical or 1st thoracic nerve. It represented, however, in the other regions by scattered cells which become aggregated to form a cervical nucleus opposite the 3rd cervical nerve and a sacral nucleus in the middle and lower part of the sacral region. Its cells are of medium size and of an oval or piriform shape. Their axons pass into the peripheral part of the lateral funiculus of the same side and there ascend, probably in the dorsospinocerebellar, direct cerebellar, fasciculus. 2. The nerve cells in the substantia gelatinosa of Rolando are arranged in 3 zones: a posterior or marginal of large angular or fusiform cells, an intermediate of small fusiform cells, and an anterior of star-shaped cells. The axons of these cells pass into the lateral and posterior funiculi and there assume a vertical course. In the anterior zone, some Golgi cells are found whose short axons ramify in the gray substance. 3. Solitary cells of varying forms and size are scattered throughout the posterior column. Some of these are grouped to form the posterior basal column in the base of the posterior column, lateral to the dorsal nucleus. The posterior basal column is well marked in the gorilla, Waldeyer, but is ill-defined in man. The axons of its cells pass partly to the posterior and lateral funiculi of the same side, and partly through the anterior white commissure to the lateral funiculus of the opposite side. Golgi cells, type 2, located in this region, send axons to the lateral and ventral columns. A few star-shaped or fusiform nerve cells of varying size are found in the substantia gelatinosa centralis. Their axons pass into the lateral funiculus of the same or of the opposite side. The nerve fibers in the gray substance form a dense interlacement of minute fibrils among the nerve cells. This interlacement is formed partly of axons which pass from the cells in the gray substance to enter the white funiculi or nerve roots, partly of the axons of Golgi cells, which ramify only in the gray substance, and partly of collaterals from the nerve fibers in the white funiculi which, as already stated, enter the gray substance and ramify within it. White substance, substantia alba. The white substance of the medulla spinalis consists of medullated nerve fibers embedded in a sponge-like network of neuroglia, and is arranged in 3 funiculi, anterior, lateral, and posterior. The anterior funiculus lies between the anterior median fissure and the most lateral of the anterior nerve roots. The lateral funiculus between these nerve roots and the posterior lateral sulcus, and the posterior funiculus between the posterior lateral and the posterior median sulci. The fibers vary greatly in thickness, the smaller being found in the fasciculus gracilis, the tract of Lissauer, and inner part of the lateral funiculus, while the largest are situated in the anterior funiculus and in the peripheral part of the lateral funiculus. Some of the nerve fibers assume a more or less transverse direction, as, for example, those which cross from side to side in the anterior white commissure, but the majority pursue a longitudinal course and are divisible into 1, those connecting the medulla spinalis with the brain and conveying impulses to or from the latter, and 2, those which are confined to the medulla spinalis and link together its different segments, that is, intersegmental or association fibers. Nerve fasciculi. The longitudinal fibers are grouped into more or less definite bundles or fasciculi. These are not recognizable from each other in the normal state, and their existence has been determined by the following methods. 1. A. Waller discovered that if a bundle of nerve fibers be cut, the portions of the fibers which are separated from their cells rapidly degenerate and become atrophied, while the cells and the parts of the fibers connected with them undergo little alteration. See footnote. Somewhat later, a change, termed chromatolysis, takes place in the nerve cells, and consists of a breaking down and an ultimate disappearance of the Nissl bodies. Further, the body of the cell is swollen, the nucleus displaced towards the periphery, and the part of the axon still attached to the altered cell is diminished in size and somewhat atrophied. Under favorable conditions, the cell is capable of reassuming its normal appearance, and its axon may grow again. End footnote. This is known as Wallerian degeneration. Similarly, if a group of nerve cells be destroyed, the fibers arising from them undergo degeneration. Thus, if the motor cells of the cerebral cortex be destroyed, or if the fibers arising from these cells be severed, a descending degeneration from the seat of injury takes place in the fibers. In the same manner, if a spinal ganglion be destroyed, or the fibers which pass from it into the medulla spinalis be cut, an ascending degeneration will extend along these fibers. 2. Pathological changes, especially in man, have given important information by causing ascending and descending degeneration. 3. By tracing the development of the nervous system, it has been observed that at first the nerve fibers are merely naked axis cylinders, and that they do not all acquire their medullary sheaths at the same time. Hence the fibers can be grouped into different bundles according to the dates at which they receive their medullary sheaths. 4. Various methods of staining nervous tissue are of great value in tracing the course and mode of termination of the axis cylinder processes. Fasciculi in the anterior funiculus. Descending fasciculi. The anterior cerebrospinal, Fasciculus cerebrospinalis anterior, direct pyramidal tract, which is usually small but varies inversely in size with the lateral cerebrospinal funiculus. It lies close to the anterior median fissure and is present only in the upper part of the medulla spinalis, gradually diminishing in size as it descends. It ends about the middle of the thoracic region. It consists of descending fibers which arise from cells in the motor area of the cerebral hemisphere of the same side, and which, as they run downward in the medulla spinalis, cross in succession through the anterior white commissure to the opposite side, where they end, either directly or indirectly, by arborizing around the motor cells in the anterior column. A few of its fibers are said to pass to lateral columns of the same side and to the gray matter at the base of the posterior column. They conduct voluntary motor impulses from the precentral gyrus to the motor centers of the cord. The Vestibulospinal Fasciculus, situated chiefly in the marginal part of the funiculus and mainly derived from the cells of Dieter's nucleus, of the same and opposite side, that is, the chief terminal nucleus of the vestibular nerve. Fibers are also contributed to this fasciculus from scattered cells of the articular formation of the Medulla Oblongata, the pons, and the midbrain, tegmentum. The other terminal nuclei of the vestibular nerve also contributes fibers. In the brainstem, these fibers form part of the median longitudinal bundle. The fasciculus can be traced to the sacral region. Its terminals and collaterals end either directly or indirectly among the motor cells of the anterior column. This fasciculus is probably concerned with equilibratory reflexes. The Tectospinal Fasciculus, situated partly in the anterior and partly in the lateral funiculus, is mainly derived from the opposite superior colliculus of the midbrain. The fibers from the superior colliculus cross the median raphe and the fountain decussation of Meinert and descend as the ventral longitudinal bundle in the reticular formation of the brainstem. Its collaterals and terminals end either directly or indirectly among the motor cells of the anterior column of the same side. Since the superior colliculus is an important visual reflex center, the tectospinal fasciculus is probably concerned with visual reflexes. Ascending fasciculi. The ventral spinothalamic fasciculus, situated in the marginal part of the funiculus and intermingled more or less with the vestibulospinal fasciculus, is derived from cells in the posterior column or intermediate gray matter of the opposite side. Their axons cross in the anterior commissure. This is a somewhat doubtful fasciculus, and its fibers are supposed to end in the thalamus and to conduct certain of the touch impulses. The remaining fibers of the anterior funiculus constitute what is termed the anterior proper fasciculus, fasciculus anterior proprius, anterior basis bundle. It consists of a. Longitudinal intersegmental fibers which arise from cells in the gray substance, more especially from those of the medial group of the anterior column, and, after a longer or shorter course, re-enter the gray substance. B. Fibers which cross in the anterior white commissure from the gray substance of the opposite side. End of section 6. Section 7 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information as a volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Spinal Cord Part 3. Fasciculi in the lateral funiculus. 1. Descending fasciculi. A. The lateral cerebrospinal fasciculus. Fasciculus cerebrospinus lateralis, cross-pyramidal tract. Extends throughout the entire length of Medulla Spinalis, and, on transverse section, appears as an oval area in front of the posterior column and medial to the cerebellospinal. Its fibers arise from the cells in the motor area of the cerebral hemisphere of the opposite side. They pass downward, in company with that of the anterior cerebrospinal fasciculus, through the same side of the brain as that from which they originate, but they cross to the opposite side in the Medulla Oblongata and descend in the lateral funiculus of the Medulla Spinalis. It is probable that the fibers of the anterior and lateral corticospinal fasciculus are not related in this direct manner with the cells of the anterior column, but terminate by arborizing around the cells at the base of the posterior column and the cells of Clark's column, which in turn link them to the motor cells in the anterior column, usually of several segments of the cord. In consequence of these interposed neurons, the fibers of the cerebrospinal fasciculi correspond not to individual muscles, but to associated groups of muscles. The anterior and lateral cerebrospinal fasciculi constitute the motor fasciculi of the Medulla Spinalis and have their origins in the motor cells of the cerebral cortex. They descend through the internal capsule of the cerebrum, traverse the cerebral peduncles and pons, and enter the pyramid of the Medulla Oblongata. In the lower part of the latter, about 2/3 of them cross in the middle line and run downward in the lateral funiculus as the lateral cerebrospinal fasciculus, while the remaining fibers do not cross in the middle line, but are continued into the same side in the Medulla Spinalis, where they form the anterior cerebrospinal fasciculus. The fibers of the latter, however, cross the midline in the anterior white commissure, and thus all the motor fibers from one side of the brain ultimately reach the opposite side of the Medulla Spinalis. The proportion of fibers which cross in the Medulla Oblongata is not a constant one, and thus the anterior and lateral cerebrospinal fasciculi vary inversely in size. Sometimes the former is absent, and in such cases it may be presumed that the decussation in the motor fibers in the Medulla Oblongata has been complete. The fibers in these two fasciculi do not acquire their medullary sheaths until after birth. In some animals, the motor fibers are situated in the posterior funiculus. B. The rubrospinal fasciculus, Monaco, prepyramidal tract, lies on the ventral aspect of the lateral cerebrospinal fasciculus and, on transverse section, appears at a somewhat triangular area. Its fibers descend from the midbrain, where they have their origin in the red nucleus of the tegmentum of the opposite side. Its terminals and collaterals end either directly or indirectly in relationship with the motor cells of the anterior column. The rubrospinal fasciculus is supposed to be concerned with cerebellar reflexes, since its afferent fibers, which pass through the superior cerebellar peduncle, send many collaterals and terminals to the red nucleus. C. The olivospinal fasciculus, Hellwig, arises in the vicinity of the inferior olivary nucleus in the Medulla Oblongata, and is seen only in the cervical region of the Medulla Spinalis, where it forms a small triangular area at the periphery, close to the most lateral of the anterior nerve roots. Its exact origin and its mode of ending have not yet been definitely made out. 2. Ascending fasciculi. A. The dorsospinocerebellar fasciculus, Fasciculus cerebellospinalis, direct cerebellar tract of Flechsig, is situated at the periphery of the posterior part of the lateral funiculus, and on transverse section appears as a flattened band reaching as far forward as a line drawn transversely through the central canal. Medially, it is in contact with the lateral cerebrospinal fasciculus, behind with the fasciculus of Lissauer. It begins about the level of the second or third lumbar nerve, and increasing in size as it ascends, passes to the vermis of the cerebellum through the inferior peduncle. Its fibers are generally regarded as being formed by the axons of the cells of the dorsal nucleus, Clark's column. They receive their medullary seeds about the 6th or 7th month of fetal life. Its fibers are supposed to conduct impulses of unconscious muscle sense. The superficial anterior lateral fasciculus, tract of Gowers, consists of 4 fasciculi: the ventral spinocerebellar, the lateral spinothalamic, the spinotectal, and the ventral spinothalamic. B. The ventral spinocerebellar fasciculus, Gowers, skirts the periphery of the lateral funiculus in front of the dorsal spinocerebellar fasciculus. In transverse section, it is shaped somewhat like a comma, the expanded end of which lies in front of the dorsal spinocerebellar fasciculus, while the tail reaches forward into the anterior funiculus. Its fibers come from the same but mostly from the opposite side of the medulla spinalis and cross both in the anterior white commissure and in the gray commissure. They are probably derived from the cells of the dorsal nucleus and from other cells in the posterior column and the intermediate portion of the gray matter. The ventral spinocerebellar fasciculus begins about the level of the third pair of lumbar nerves and can be followed into the medulla oblongata and pons almost to the level of the inferior colliculus, where it crosses over the superior peduncle and then passes backward along its medial border to reach the vermis of the cerebellum. In the pons, it lies along the lateral edge of the lateral lemniscus. Some of its fibers join the dorsospinocerebellar fasciculus at the level of the inferior peduncle and pass with them into the cerebellum. Other fibers are said to continue upward in the dorsolateral part of the tegmentum of the midbrain, probably as far as the thalamus. C. The lateral spinothalamic fasciculus is supposed to come from cells in the dorsal column and the intermediate gray matter whose axons cross in the anterior commissure to the opposite lateral funiculus, where they pass upward on the medial side of the ventral spinocerebellar fasciculus. On reaching the medulla oblongata, They continue in the formatio reticularis near the median fillet and probably terminate in the ventrolateral region of the thalamus. It is supposed to conduct impulses of pain and temperature. The lateral and ventral spinal thalamic fasciculi are sometimes termed the secondary sensory fasciculus or spinal lemniscus. D. The spinotectal fasciculus is supposed to arise in the dorsal column and terminate in the possibly inferior and superior colliculi. It is situated ventral to the lateral spinothalamic fasciculus, but its fibers are more or less intermingled with it. It is also known as the spino-quadrigeminal system of Mott. In the brainstem, the fibers run lateral from the inferior olive, ventrolateral from the superior olives, then ventromedial from the spinal tract of the trigeminal. The fibers come to lie in the medial portion of the lateral lemniscus. E. The fasciculus of Lissauer is a small strand situated in relation to the tip of the posterior column close to the entrance of the posterior nerve roots. It consists of fine fibers which do not receive their medullary sheaths until towards the close of fetal life. It is usually regarded as being formed by some of the fibers of the posterior nerve roots, which ascend for a short distance in the tract and then enter the posterior column. But since its fibers are myelinated later than those of the posterior nerve roots and do not undergo degeneration in locomotor ataxia, they are probably intersegmental in character. In addition, the fasciculus or tract of Lissauer contains great numbers of fine, non-medullated fibers derived mostly from the dorsal roots, but partly endogenous in origin. These fibers are intimately related to the substantia gelatinosa, which is probably the terminal nucleus. The non-medullated fibers ascend or descend for short distances not exceeding 1 or 2 segments, but most of them enter the substantia gelatinosa at or near the level of their origin. Ransom suggests that these non-medullated fibers and the substantia gelatinosa are concerned with the reflexes associated with pain impulses. F. The lateral proper fasciculus, Fasciculus lateralis proprius, lateral basis bundle, constitutes the remainder of the lateral column, and is continuous in front with the anterior proper fasciculus. It consists chiefly of intersegmental fibers which arise from cells in the gray substance, and, after a longer or shorter course, re-enter the gray substance and ramify in it. Some of its fibers are, however, continued upward into the brain under the name of the medial longitudinal fasciculus. Fasciculi in the posterior funiculus. This funiculus comprises 2 main fasciculi, that is, the fasciculus gracilis and the fasciculus cuneatus. These are separated from each other in the cervical and upper thoracic regions by the posterior intermediate septum and consist mostly of ascending fibers derived from posterior nerve roots. The fasciculus gracilis, tract of Gall, is wedge-shaped on transverse section and lies next to the posterior median septum, its base being at the surface of the medulla spinalis, and its apex directed towards the posterior gray commissure. It increases in size from below upward and consists of long, thin fibers which are derived from the posterior nerve roots and ascend as far as the medulla oblongata, where they end in the nucleus gracilis. The fasciculus cuneatus, tract of Burdach, is a triangular on transverse section and lies between the fasciculus gracilis and the posterior column, its base corresponding with the surface of the medulla spinalis. Its fibers, larger than those of the fasciculus gracilis, are mostly derived from the same source, that is, the posterior nerve roots. Some may ascend only for a short distance in the tract and, entering the gray matter, come in close relationship with the cells of the dorsal nucleus, while others can be traced as far as the medulla oblongata, where they end in the gracil and cuneate nuclei. The fasciculus gracilis and fasciculus cuneatus conduct 1. Impulses of conscious muscle sense. Neurons of the second order, from the nucleus gracilis and nucleus cuneatus, pass in the median lemniscus to the thalamus, and neurons of the third order, from the thalamus to the cerebral cortex. 2. Impulses of unconscious muscle sense, via neurons of the second order from the nucleus gracilis and nucleus cuneatus, pass in the internal and external arcuate fibers of the medulla oblongata to the inferior peduncle and through it to the cerebellum. 3. Impulses of tactile discrimination, via neurons of the second order from the nucleus cuneatus and nucleus gracilis, pass in the median lemniscus to the thalamus. Neurons of the third order pass from the thalamus to the cortex. The posterior proper fasciculus, posterior grand bundle, posterior basis bundle, arises from cells in the posterior column. Their axons bifurcate into ascending and descending branches, which occupy the ventral part of the funiculus close to the gray column. They are intersegmental and run for varying distances, sending off collaterals and terminals to the gray matter. Some descending fibers occupy different parts at different levels. In the cervical and upper thoracic regions, they appear as a comma-shaped fasciculus in the lateral part of the fasciculus cuneatus, the blunt end of the comma being directed towards the posterior gray commissure. In the lower thoracic region, they form a dorsal peripheral band on the posterior surface of the funiculus. In the lumbar region, they are situated by the side of the posterior median septum and appear on section as a semi-elliptical bundle, which, together with the corresponding bundle of the opposite side, forms the oval area of Flechsig, while in the conus medullaris they assume the form of a triangular strand in the posterior medial part of the fasciculus gracilis. These descending fibers are mainly intersegmental in character and derived from the cells in the posterior columns, but some consist of the descending branches of posterior nerve roots. The comma-shaped fasciculus was supposed to belong to the second category, but against this view is the fact that it does not undergo descending degeneration when the posterior nerve roots are destroyed. As already stated, each spinal nerve possesses two roots, an anterior and a posterior, which are attached to the surface of the medulla spinalis opposite the corresponding column of gray substance. Their fibers become medullated about the 5th month of fetal life. The anterior nerve, radix anterior, consists of efferent fibers, which are the axons of the nerve cells in the ventral portion of the anterior and lateral columns. A short distance from their origins, these axons are invested by medullary seeds and, passing forward, emerge in 2 or 3 irregular rows over an area which measures about 3 millimeters in width. The posterior root, radix posterior, comprises some 6 or 8 fasciculi, attached in linear series along the posterior lateral sulcus. It consists of afferent fibers which arise from the nerve cells in a spinal ganglion. Each ganglion cell gives off a single fiber which divides in a T-shaped manner into 2 processes, medial and lateral. The lateral processes extend to the sensory end organs of the skin, muscle, tendons, joints, etc., somatic receptors, and to the sensory end organs of the viscera, visceral receptors. The medial processes of the ganglion cells grow into the medulla spinalis as the posterior roots of the spinal nerves. The posterior nerve root enters the medulla spinalis in 3 chief bundles: medial, intermediate, and lateral. The medial strand passes directly into fasciculus cuneatus. It consists of coarse fibers, which acquire their medullary sheaths about the 5th month of intrauterine life. The intermediate strand consists of coarse fibers which enter the gelatinous substance of Rolando. The lateral is composed of fine fibers, which assume a longitudinal direction in the tract of Lissauer and do not acquire their medullary sheaths until after birth. In addition to these medullated fibers, there are great numbers of non-medullated fibers which enter with the lateral bundle. They are more numerous than the myelinated fibers. They arise from the small cells of the spinal ganglia by T-shaped axons similar to the myelinated. They are distributed with the peripheral nerves chiefly to the skin, and only a few are found in the nerves to the muscles. Having entered the medulla spinalis, all the fibers of the posterior nerve roots divide to ascending and descending branches, and these in their turn give off collaterals which enter the gray substance. The descending fibers are short and soon enter the gray substance. The ascending fibers are grouped into long, short, and intermediate. The long fibers ascend in the fasciculus cuneatus and fasciculus gracilis as far as the medulla oblongata, where they end by arborizing around cells of the cuneate and gracile nuclei. The short fibers run upward for a distance of only 5 or 6 millimeters and enter the gray substance, while the intermediate fibers, after a somewhat longer course, have a similar destination. All fibers entering the gray substance end by arborizing around its nerve cells or the dendrites of cells, those of intermediate length being especially associated with the cells of the dorsal nucleus. The long fibers of the posterior nerve roots pursue an oblique course upward, being situated at first in the lateral part of the fasciculus cuneatus. Higher up, they occupy the middle of this fasciculus, having been displaced by the accession of other entering fibers. While still higher, they ascend in the fasciculus gracilis. The upper cervical fibers do not reach this fasciculus, but are entirely confined to the fasciculus cuneatus. The localization of these fibers is very precise. The sacral nerves lie in the medial part of the fasciculus gracilis and near its periphery, the lumbar nerves lateral to them, the thoracic nerves still more laterally, while the cervical nerves are confined to the fasciculus cuneatus. End of section 7. Section 8 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4, by Henry Gray. The Hindbrain, Part 1. The encephalon, or brain. General considerations and divisions. The brain is contained within the cranium and constitutes the upper, greatly expanded part of the central nervous system. In its early embryonic condition, it consists of 3 hollow vesicles termed the hindbrain, or rhombencephalon, the midbrain, or mesencephalon, and the forebrain, or prosencephalon. And the parts derived from each of these can be recognized in the adult. Thus, in the process of development, the wall of the hindbrain undergoes modification to form the medulla oblongata, the pons and cerebellum, while its cavity is expanded to form the fourth ventricle. The midbrain forms only a small part of the adult brain. Its cavity becomes the cerebral aqueduct, aqueduct of Sylvius, which serves as a tubular communication between the third and fourth ventricles, while its walls are thickened to form the corpora quadrigemina and cerebral peduncles. The forebrain undergoes great modifications. Its anterior part, or telencephalon, expands laterally in the form of 2 hollow vesicles, the cavities of which become the lateral ventricles, while the surrounding walls form the cerebral hemispheres and their commissures. The cavity of the posterior part, or diencephalon, forms the greater part of the third ventricle, and from its walls are developed most of the structures which bound that cavity. The hindbrain, or rhombencephalon. The hindbrain, or rhombencephalon, occupies the posterior fossa of the cranial cavity, and lies below a fold of Dura Mater, the tentorium cerebelli. It consists of A, the myelencephalon, comprising the medulla oblongata and the lower part of the fourth ventricle, B, the metencephalon, consisting of the pons, cerebellum, and the intermediate part of the fourth ventricle, and C, the isthmus rhombencephali, a constricted portion immediately adjoining the midbrain, and including the superior peduncles of the cerebellum, the anterior medullary velum, and the upper part of the fourth ventricle. The medulla oblongata, spinal bulb. The medulla oblongata extends from the lower margin of the pons to a plane passing transversely below the pyramidal decussation and above the first pair of cervical nerves. This plane corresponds with the upper border of the atlas behind and the middle of the odontoid process of the axis in front. At this level, the medulla oblongata is continuous with the medulla spinalis. Its anterior surface is separated from the basilar part of the occipital bone and the upper part of the odontoid process by the membranes of the brain and the occipital axial ligaments. Its posterior surface is received into the fossa between the hemispheres of the cerebellum, and the upper portion of it forms the lower part of the floor of the fourth ventricle. The medulla oblongata is pyramidal in shape, its broad extremity being directed upwards towards the pons, while its narrow lower end is continuous with the medulla spinalis. It measures about 3 centimeters in length, about 2 centimeters in breadth at its widest part, and about 1.25 centimeters in thickness. The central canal of the medulla spinalis is prolonged into its lower half and then opens into the cavity of the fourth ventricle. The medulla oblongata may therefore be divided into a lower closed part containing the central canal and an upper open part corresponding with the lower portion of the fourth ventricle. The anterior median fissure, fissura mediana anterior, ventral or ventromedian fissure. contains a fold of pia mater and extends along the entire length of the medulla oblongata. It ends at the lower border of the pons in a small triangular expansion termed the foramen cecum. Its lower part is interrupted by bundles of fibers which cross obliquely from one side to the other and constitute the pyramidal decussation. Some fibers, termed the anterior external arcuate fibers, emerge from the fissure above this decussation and curve lateralward and upward over the surface of the medulla oblongata to join the inferior peduncle. The posterior median fissure, fissura mediana posterior, dorsal or dorsal-medial fissure, is a narrow groove and exists only in the closed part of the medulla oblongata. It becomes gradually shallower from below upward and finally ends about the middle of the medulla oblongata, where the central canal expands into the cavity of the fourth ventricle. These two fissures divide the closed part of the medulla oblongata into symmetrical halves, each presenting elongated eminences which, on the surface view, are continuous with the funiculi of the medulla spinalis. In the open part, the halves are separated by the anterior median fissure and by a median raphe, which extends from the bottom of the fissure to the floor of the fourth ventricle. Further, certain of the cranial nerves pass through the substance of the medulla oblongata and are attached to its surface in series with the roots of the spinal nerves. Thus, the fibers of the hypoglossal nerve represent the upward continuation of the anterior nerve roots and emerge in linear series from a furrow termed the anterior lateral sulcus. Similarly, the accessory, vagus, and glossopharyngeal nerves correspond with the posterior nerve roots and are attached to the bottom of a sulcus named the posterior lateral sulcus. Advantage is taken of this arrangement to subdivide each half of the medulla oblongata into three districts: anterior, middle, and posterior. Although these three districts appear to be directly continuous with the corresponding funiculi of the medulla spinalis, they do not necessarily contain the same fibers, since some of the fasciculi of the medulla spinalis end in the medulla oblongata, while others alter their course in passing through it. The anterior district is named the pyramid, Pyramis medulla oblongata, and lies between the anterior median fissure and the anterior lateral sulcus. Its upper end is slightly constricted, and between it and the pons the fibers of the abducent nerve emerge. A little below the pons it becomes enlarged and prominent, and finally tapers into the anterior funiculus of the medulla spinalis, with which, at first sight, it appears to be directly continuous. The two pyramids contain the motor fibers which pass from the brain to the medulla oblongata and medulla spinalis, corticobulbar and corticospinal fibers. When these pyramidal fibers are traced downward, it is found that some 2/3 or more of them leave the pyramids in successive bundles and decussate in the anterior median fissure, forming what is termed the pyramidal decussation. Having crossed the middle line, they pass down in the posterior part of the lateral funiculus as the lateral cerebrospinal fasciculus. The remaining fibers, that is, those which occupy the lateral part of the pyramid, do not cross the middle line, but are carried downward as the anterior cerebrospinal fasciculus into the anterior funiculus of the same side. The greater part of the anterior proper fasciculus of the medulla spinalis is continued upward through the medulla oblongata under the name of the medial-longitudinal fasciculus. The lateral district is limited in front by the anterior lateral sulcus and the roots of the hypoglossal nerve, and behind by the posterior lateral sulcus and the roots of the accessory, vagus, and glossopharyngeal nerves. Its upper part consists of a prominent oval mass which is named the olive, while its lower part is of the same width as the lateral funiculus of the medulla spinalis and appears on the surface to be a direct continuation of it. As a matter of fact, only a portion of the lateral funiculus is continued upward into this district, for the lateral cerebrospinal fasciculus passes into the pyramid of the opposite side, and the dorsal spinocerebellar fasciculus is carried into the inferior peduncle in the posterior district. The ventral spinocerebellar fasciculus is continued upward on the lateral surface of the medulla oblongata in the same relative position it occupies in the spinal cord, until it passes under cover of the external arcuate fibers. It passes beneath these fibers, just dorsal to the olive and ventral to the roots of the vagus and glossopharyngeal nerves. It continues upward through the pons along the dorsolateral edge of the lateral lemniscus. The remainder of the lateral funiculus consists chiefly of the lateral proper fasciculus. Most of these fibers dip beneath the olive and disappear from the surface, but a small strand remains superficial to the olive. In a depression at the upper end of this strand is the acoustic nerve. The olive, oliva, olivary body, is situated lateral to the pyramid, from which it is separated by the anterior lateral sulcus and the fibers of the hypoglossal nerve. Behind, it is separated from the posterior lateral sulcus by the ventral spinocerebellar fasciculus. In the depression between the upper end of the olive and the pons lies the acoustic nerve. It measures about 1.25 centimeters in length, and between its upper end and the pons there is a slight depression to which the roots of the facial nerve are attached. The external arcuate fibers wind across the lower part of the pyramid and olive and enter the inferior peduncle. The posterior district lies behind the posterior lateral sulcus and the roots of the accessory vagus and glossopharyngeal nerves, and, like the lateral district, is divisible into a lower and an upper portion. The lower part is limited behind by the posterior median fissure and consists of the fasciculus gracilis and the fasciculus cuneatus. The fasciculus gracilis is placed parallel to and along the side of the posterior median fissure, and separated from the fasciculus cuneatus by the posterior intermediate sulcus and septum. The gracile and cuneate fasciculi are at first vertical in direction, but at the lower part of the rhomboid fossa they diverge from the middle line in a V-shaped manner, and each presents an elongated swelling. That on the fasciculus gracilis is named the clava, and is produced by its subjacent nucleus of gray matter, the nucleus gracilis. That on the fasciculus cuneatus is termed the cuneate tubercle, and is likewise caused by a gray nucleus named the nucleus cuneatus. The fibers of these fasciculi terminate by arborizing around the cells in their respective nuclei. A third elevation, produced by the substantia gelatinosus of Rolando, is present in the lower part of the posterior district of the medulla oblongata. It lies on the lateral aspect of the fasciculus cuneatus and is separated from the surface of the medulla oblongata by a band of nerve fibers which form the spinal tract, spinal root, of the trigeminal nerve. Narrow below, this elevation gradually expands above and ends about 1.25 centimeters below the pons in a tubercle, the tubercle of Rolando, tuber cinereum. The upper part of the posterior district of the medulla oblongata is occupied by the inferior peduncle, a thick rope-like strand situated between the lower part of the fourth ventricle and the roots of the glossopharyngeal and vagus nerves. The inferior peduncles connect the medulla spinalis and medulla oblongata with the cerebellum and are sometimes named the restiform bodies. As they pass upward, they diverge from each other and assist in forming the lower part of the lateral boundaries of the fourth ventricle. Higher up, they are directed backwards, each passing to the corresponding cerebellar hemisphere. Near their entrance into the cerebellum, they are crossed by several strands of fibers which run to the median sulcus of the rhomboid fossa and are named the stria medullaris. The inferior peduncle appears to be the upward continuation of the fasciculus gracilis and fasciculus cuneatus. This, however, is not so, as the fibers of these fasciculi end in the gracile and cuneate nuclei. The constitution of the inferior peduncle will be subsequently discussed. Caudal to the stria medullaris, the inferior peduncle is partly covered by the corpus pontobulbar, a thin mass of cells and fibers extending from the pons between the origin of the 7th and 8th cranial nerves. Internal structure of the medulla oblongata. Although the external form of the medulla oblongata bears a certain resemblance to that of the upper part of the medulla spinalis, its internal structure differs widely from that of the latter, and this for the following principal reasons. 1. Certain fasciculi, which extend from the medulla spinalis to the brain and vice versa, undergo a rearrangement in their passage through the medulla oblongata. 2. Others, which exist in the medulla spinalis, end in the medulla oblongata. 3. New fasciculi originate in the gray substance of the medulla oblongata and pass to different parts of the brain. 4. The gray substance, which in the medulla spinalis forms a continuous H-shaped column, becomes greatly modified and subdivided in the medulla oblongata, where also new masses of gray substance are added. 5. On account of the opening out of the central canal of the medulla spinalis, certain parts of the gray substance, which in the medulla spinalis were more or less centrally situated, are displaced in the rhomboid fossa. 6. The medulla oblongata is intimately associated with many of the cranial nerves, some arising from and others ending in nuclei within its substance. The cerebrospinal fasciculi. The downward course of these fasciculi from the pyramids of the medulla oblongata and their partial decussation have already been described. In crossing to reach the lateral funiculus of the opposite side, the fibers of the lateral cerebrospinal fasciculi extend backwards through the anterior columns and separate the head of each of these columns from its base. The base retains its position in relation to the ventral aspect of the central canal, and when the latter opens into the fourth ventricle, appears in the rhomboid fossa close to the middle line, where it forms the nuclei of the hypoglossal and abducent nerves, while above the level of the ventricle, it exists as the nuclei of the trochlear and oculomotor nerves in relation to the floor of the cerebral aqueduct. The head of the column is pushed lateralward and forms the nucleus ambiguus, which give origin from below upward to the cranial part of the accessory and the motor fibers of the vagus and glossopharyngeal, and still higher to the motor fibers of the facial and trigeminal nerves. The fasciculus gracilis and fasciculus cuneatus constitute the posterior sensory fasciculi of the medulla spinalis. They are prolonged upward into the lower part of the medulla oblongata, where they end respectively in the nucleus gracilis and nucleus cuneatus. These two nuclei are continuous with the central gray substance of the medulla spinalis and may be regarded as dorsal projections of this, each being covered superficially by the fibers of the corresponding fasciculus. On transverse section, the nucleus gracilis appears as a single, more or less quadrangular mass, while the nucleus cuneatus consists of two parts, a larger, somewhat triangular medial nucleus composed of small or medium-sized cells, and a small lateral nucleus containing large cells. The fibers of the fasciculus gracilis and fasciculus cuneatus end by arborizing around the cells of these nuclei. From the cells of the nuclei, new fibers arise. Some of these are continued as the posterior external arcuate fibers into the inferior peduncle and through it to the cerebellum, but most of them pass forward through the neck of the posterior column, thus cutting off its head from its base. Curving forward, they decussate in the middle line with the corresponding fibers of the opposite side and run upward immediately behind the cerebrospinal fibers as a flattened band named the lemniscus or fillet. The decussation of these sensory fibers is situated above that of the motor fibers and is named the decussation of the lemniscus or sensory decussation. The lemniscus is joined by the spinothalamic fasciculus, the fibers of which are derived from the cells of the gray substance of the opposite side of the medulla spinalis. The base of the posterior column at first lies on the dorsal aspect of the central canal, but when the latter opens into the fourth ventricle, it appears in the lateral part of the rhomboid fossa. It forms the terminal nuclei of the sensory fibers of the vagus and glossopharyngeal nerves and is associated with the vestibular part of the acoustic nerve and the sensory root of the facial nerve. Still higher, it forms a mass of pigmented cells, the locus ceruleus, in which some of the sensory fibers of the trigeminal nerve appear to end. The head of the posterior column forms a long nucleus, in which the fibers of the spinal tract of the trigeminal nerve largely end. The dorso-spinocerebellar fasciculus, Fasciculus cerebellospinalis, direct cerebellar tract, leaves the lateral district of the medulla oblongata. Most of its fibers are carried backward into the inferior peduncle of the same side, and through it are conveyed to the cerebellum. But some run upward with the fibers of the lemniscus and, reaching the inferior colliculus, undergo decussation and are carried to the cerebellum through the superior peduncle. The proper fasciculi, basis bundles, of the anterior and lateral funiculi largely consist of intersegmental fibers, which link together the different segments of the medulla spinalis. They assist in the production of the phaematio reticularis in the medulla oblongata, and many of them are accumulated into a fasciculus which runs up close to the median raphe between the lemniscus and the rhomboid fossa. This strand is named the medial longitudinal fasciculus and will be again referred to. End of section 8. Section 9 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Hindbrain Part 2. Gray substance of the medulla oblongata. In addition to the gracile and cuneate nuclei, there are several other nuclei to be considered. Some of these are traceable from the gray substance of a medulla spinalis, while others are unrepresented in it. 1. The hypoglossal nucleus is derived from the base of the anterior column. In the lower closed part of the medulla oblongata, it is situated on the ventrolateral aspect of the central canal, but in the upper part, it approaches the rhomboid fossa, where it lies close to the middle line under an eminence named the trigonum hypoglossae. Numerous fibers connect the two nuclei. Both nuclei send long dendrons across the midline to the opposite nucleus. Commissure fibers also connect them. The nucleus measures about 2 centimeters in length and consists of large multipolar nerve cells similar to those in the anterior column of the spinal cord, whose axons constitute the roots of the hypoglossal nerve. These nerve roots leave the ventral side of the nucleus, pass forward between the white reticular formation and the gray reticular formation, some between the inferior olivary nucleus and the medial accessory olivary nucleus, and emerge from the anterolateral sulcus. 2. The nucleus ambiguus, the somatic motor nucleus of the glossopharyngeal, vagus, and cranial portion of the accessory nerves, is the continuation into the medulla oblongata of the dorsolateral cell group of the anterior column of the spinal cord. Its large, multipolar cells are like those in the anterior column of the cord. They form a slender column in the deep part of the formatio reticularis grisae, about midway between the dorsal accessory olive and the nucleus of the spinal tract of the trigeminal. It extends from the level of the decussation of the median fillet to the upper end of the medulla oblongata. Its fibers first pass backward towards the floor of the fourth ventricle, and then curve rather abruptly lateralward and ventrally to join the fibers from the dorsal nucleus. The dorsal nucleus, nucleus alicinerea, often called the sensory nucleus or the trigeminal nucleus of the sensory fibers of the glossopharyngeal and vagus nerves, is probably a mixed nucleus and contains not only the terminations of the sympathetic afferent or sensory fibers and the cells connected with them, but contains also cells which give rise to the sympathetic efferent or preganglionic fibers. These preganglionic fibers terminate in sympathetic ganglia, from which the impulses are carried by other neurons. The cells of the dorsal nucleus are spindle-shaped like those of the posterior column of the spinal cord, and the nucleus is usually considered as representing the base of the posterior column. It measures about 2 centimeters in length, and in the lower, closed part of the medulla oblongata, is situated behind the hypoglossal nucleus, whereas in the upper open part, it lies lateral to that nucleus and corresponds to an eminence named the alicinerea, trigonum vagi, in the rhomboid fossa. 4. The nuclei of the cochlear and vestibular nerves are described later. 5. The olivary nuclei are 3 in number on either side of the midline, that is, the inferior olivary nucleus and the medial and dorsal accessory olivary nuclei. They consist of small round yellowish cells and numerous fine nerve fibers. A. The inferior olivary nucleus is the largest and is situated within the olive. It consists of a gray folded lamina arranged in the form of an incomplete capsule, open medially by an aperture called the hilum. Emerging from the hilum are numerous fibers which collectively constitute the peduncle of the olive. The axons, olivocerebellar fibers, which leave the olivary nucleus, pass out through the hilum and decussate with those from the opposite olive in the raphe. Then, as internal arcuate fibers, they pass partly through and partly around the opposite olive and enter the inferior peduncle to be distributed to the cerebellar hemisphere of the opposite side from which they arise. The fibers are smaller than the internal arcuate fibers connected with the medial lemniscus. Fibers passing in the opposite direction from the cerebellum to the olivary nucleus are often described, but their existence is doubtful. Much uncertainty also exists in regard to the connections of the olive and the spinal cord. Important connections between the cerebellum and the olive of the same side exist, but the exact pathway is unknown. Many collaterals from the reticular formation and from the pyramids enter the inferior olivary nucleus. Removal of one cerebellar hemisphere is followed by atrophy of the opposite olivary nucleus. B. The medial accessory olivary nucleus lies between the inferior olivary nucleus and the pyramid and forms a curved lamina, the concavity of which is directed laterally. The fibers of the hypoglossal nerve, as they traverse the medulla, pass between the medial accessory and the inferior olivary nuclei. C. The dorsal accessory olivary nucleus is the smallest and appears on transverse section as the curved lamina behind the inferior olivary nucleus. 6. The nucleus arcuatus is described below with the anterior external arcuate fibers. Inferior peduncle, restiform body. The position of the inferior peduncles has already been described. Each comprises 1. Fibers from the dorsal spinocerebellar fasciculus, which ascends from the lateral funiculus of the medulla spinalis. 2. The olivocerebellar fibers from the opposite olivary nucleus. 3. Internal arcuate fibers from the gracillin cuneate nuclei of the opposite side. These fibers form the deeper and larger part of the inferior peduncle. 4. The anterior external arcuate fibers vary as to the prominence in different cases. In some, they form an almost continuous layer covering the pyramid and olive, while in others they are barely visible on the surface. They arise from the cells of the gracillin cuneate nuclei and, passing forward through the formatio reticularis, decussate in the middle line. Most of them reach the surface by way of the anterior median fissure and arch backward over the pyramid. Reinforced by others which emerge between the pyramid and olive, they pass backwards over the olive and lateral district of the medulla oblongata and enter the inferior peduncle. Thus, they connect the cerebellum with the gracillin cuneate nuclei of the opposite side. As the fibers arch across the pyramid, they enclose a small nucleus which lies in front of and medial to the pyramid. This is named the nucleus arcuatus, and is serially continuous above with the nucleus pontus and the pons. It contains small fusiform cells around which some of the arcuate fibers end and from which others arise. 5. The posterior external arcuate fibers also take origin in the gracillin cuneate nuclei. They pass to the inferior peduncle of the same side. It is uncertain whether fibers are continued directly from the gracillin cuneate fasciculi into the inferior peduncle. 6. Fibers from the terminal sensory nuclei of the cranial nerves, especially the vestibular. Some of the fibers of the vestibular nerve are thought to continue directly into the cerebellum. 7. Fibers from the ventral spinocerebellar fasciculus. 8. The existence of fibers from the cerebellum, cerebellobulbar, cerebellar-olivary, and cerebellospinal, to the medulla and spinal cord is very uncertain. Formatio reticularis. This term is applied to the coarse reticulum which occupies the anterior and lateral districts of the medulla oblongata. It is situated behind the pyramid and olive, extending laterally as far as the inferior peduncles, and dorsally to within a short distance of the rhomboid fossa. The reticulum is caused by the intersection of bundles of fibers running at right angles to each other, some being longitudinal, others more or less transverse in direction. The formatio reticularis presents a different appearance in the anterior district from what it does in the lateral. In the former, there is an almost entire absence of nerve cells, and hence this part is known as the reticularis alba, whereas in the lateral district nerve cells are numerous, and as a consequence it presents a gray appearance and is termed the reticularis grisea. In the substance of the formatio reticularis are two small nuclei of gray matter, one, the inferior central nucleus, nucleus of Roller, near the dorsal aspect of the hilus of the inferior olivary nucleus, the other, the nucleus lateralis, between the olive and the spinal tract of the trigeminal nerve. In the reticularis alba, the longitudinal fibers form two well-defined fasciculi, that is, 1, the lemniscus, which lies close to the raphe immediately behind the fibers of the pyramid, and 2, the medial longitudinal fasciculus, which is continued upward from the anterior and lateral proper fasciculi of the medulla spinalis, and, in the upper part of the medulla oblongata, lies between the lemniscus and the gray substance of the rhomboid fossa. The longitudinal fibers in the reticularis grisea are derived from the lateral funiculus of the medulla spinalis after the lateral cerebrospinal fasciculus has passed over to the opposite side, and the dorsal spinocerebellar fasciculus has entered the inferior peduncle. They form indeterminate fibers, with the exception of a bundle named the Fasciculus solitarius, which is made up of descending fibers of the vagus and glossopharyngeal nerves. The transverse fibers of the formatio reticularis are the arcuate fibers already described. The pons, Pons varioli. The pons, or forepart of the hindbrain, is situated in front of the cerebellum. From its superior surface, the cerebral peduncles emerge, one on either side of the middle line. Curving around each peduncle, close to the upper surface of the pons, a thin white band, the tinea pontis, is frequently seen. It enters the cerebellum between the middle and superior peduncles. Behind and below, the pons is continuous with the medulla oblongata, but is separated from it in front by a furrow in which the abducent, facial, and acoustic nerves appear. Its ventral or anterior surface, pars basilaris pontis, is very prominent, markedly convex from side to side, less so from above downward. It consists of transverse fibers arched like a bridge across the middle line and gathered on either side into a compact mass which forms the middle peduncle. It rests upon the clivus of the sphenoidal bone and is limited above and below by well-defined borders. In the middle line is the sulcus basilaris for the lodgment of the basilar artery. This sulcus is bounded on either side by an eminence caused by the descent of the cerebrospinal fibers through the substance of the pons. Outside these eminences, near the upper border of the pons, the trigeminal nerves make their exit, each consisting of a smaller mediomotor root and a larger lateral sensory root. Vertical lines drawn immediately beyond the trigeminal nerves may be taken as the boundaries between the ventral surface of the pons and the middle cerebellar peduncle. Its dorsal or posterior surface, pars dorsalis pontis, triangular in shape, is hidden by the cerebellum and is bounded laterally by the superior peduncle. It forms the upper part of the rhomboid fossa, with which it will be described. Structure. Transverse sections of the pons show it to be composed of two parts, which differ in appearance and structure. Thus, the basilar or ventral portion consists for the most part of fibers arranged in transverse and longitudinal bundles, together with a small amount of gray substance, while the dorsal tegmental portion is a continuation of the reticular formation of the medulla oblongata, and most of its constituents are continued into the tegmenta of the cerebral peduncles. The basilar part of the pons consists of A. Superficial and deep transverse fibers, B. Longitudinal fasciculi, and C. Some small nuclei of gray substance, termed the nuclei pontis, which give rise to the transverse fibers. The superficial transverse fibers, fibrae pontis superficialis, constitute a rather thick layer on the ventral surface of the pons and are collected into a large rounded bundle on either side of the middle line. This bundle, with the addition of some transverse fibers from the deeper part of the pons, forms the greater part of the brachium pontis. The deep transverse fibers, fibrae pontis profundae, partly intersect and partly lie in the dorsal aspect of the cerebrospinal fibers. They course to the lateral border of the pons and form part of the middle peduncle. A further connections of this brachium will be discussed with the anatomy of the cerebellum. The longitudinal fasciculi, Fasciculi longitudinalis, are derived from the cerebral peduncles and enter the upper surface of the pons. They stream downward on either side of the middle line in larger or smaller bundles separated from each other by the deep transverse fibers. These longitudinal bundles cause a forward projection of the superficial transverse fibers and thus give rise to the eminences on the anterior surface. Some of these fibers end in or give off collateral to the nuclei pontis. An important pathway is thus formed between the cerebral cortex and the cerebellum, the first neuron having its cell body in the cortex and sending its axon through the internal capsule and cerebral peduncle to form synapses either by terminals or collaterals with cell bodies situated in the nuclei pontis. Axons from these cells form the transverse fiber which pass through the middle peduncle into the cerebellum. Others, after decussating, terminate either directly or indirectly in the motor nuclei of the trigeminal, abducent, facial, and hypoglossal nerves, but most of them are carried through the pons and at the lower surface are collected into the pyramids of the medulla. The fibers which end in the motor nuclei of the cranial nerves are derived from the cells of the cerebral cortex and bear the same relation to the motor cells of the cranial nerves that the cerebrospinal fibers bear to the motor cells in the anterior column of the medulla spinalis. Probably none of the collaterals or terminals of the cerebrospinal and cerebrobulbar fibers end directly in the motor nuclei of the spinal and cranial nerves. One or more association neurons are probably interpolated in the pathway. The nuclei pontis are serially continuous with the arcuate nuclei in the medulla and consist of small groups of multipolar nerve cells which are scattered between the bundles of the transverse fibers. The dorsal or tegmental part of the pons is chiefly composed of an upward continuation of the reticular formation and gray substance of the medulla oblongata. It consists of transverse and longitudinal fibers and also contains important gray nuclei and is subdivided by a median raphe which, however, does not extend into the basilar part, being obliterated by the transverse fibers. The transverse fibers in the lower part of the pons are collected into a distinct strand named the trapezoid body. This consists of fibers which arise from cells of the cochlear nucleus and will be referred to in connection with the cochlear division of the acoustic nerve. In the substance of the trapezoid body is a collection of nerve cells which constitutes the trapezoid nucleus. The longitudinal fibers, which are continuous with those of the medulla oblongata, are mostly collected into two fasciculi on each side. One of these lies between the trapezoid body and the reticular formation and forms the upward prolongation of the lemniscus. The second is situated near the floor of the fourth ventricle and is the medial longitudinal fasciculus. Other longitudinal fibers, more diffusely distributed, arise from the cells of the gray substance of the pons. The rest of the dorsal part of the pons is a continuation upward of the formatio reticularis of the medulla oblongata, and like it, presents the appearance of a network, in the meshes of which are numerous nerve cells. Besides these scattered nerve cells, there are some larger masses of gray substance, that is, the superior olivary nucleus and the nuclei of the trigeminal, abducent, facial, and acoustic nerves. 1. The superior olivary nucleus, nucleus olivaris superior, is a small mass of gray substance situated on the dorsal surface of the lateral part of the trapezoid body. Rudimentary in man, but well developed in certain animals, it exhibits the same structure as the inferior olivary nucleus and is situated immediately above it. Some of the fibers of the trapezoid body end by arborizing around the cells of this nucleus, while others arise from these cells. 2. The nuclei of the trigeminal nerve. Nuclei and trigemini. In the pons are two in number, a motor and a sensory. The motor nucleus is situated in the upper part of the pons, close to its posterior surface and along the line of the lateral margin of the fourth ventricle. It is serially homologous with the nucleus ambiguus and the dorsolateral cell group of the anterior column of the spinal cord. The axis cylinder processes of its cells form a portion of the motor root of the trigeminal nerve. The remaining fibers of the motor root of this nerve consist of a fasciculus, which arises from the gray substance of the floor of the cerebral aqueduct, and hence is named the mesencephalic root. It is not altogether clear whether the mesencephalic root is motor or sensory. The sensory nucleus is lateral to the motor one and beneath the superior peduncle. Some of the sensory fibers of the trigeminal nerve end in this nucleus, but the greater number descend under the name of the spinal tract of the trigeminal nerve to end in the substantia gelatinosa of Rolando. The roots, motor and sensory, of the trigeminal nerve pass through the substance of the pons and emerge near the upper margin of its anterior surface. 3. The nucleus of the abducent nerve, nucleus N. abducentus, is a circular mass of gray substance situated close to the floor of the fourth ventricle, above the stria medullaris and subjacent to the medial eminence. It lies a little lateral to the ascending part of the facial nerve. The fibers of the abducent nerve pass forward through the entire thickness of the pons on the medial side of the superior olivary nucleus and between the lateral fasciculi of the cerebrospinal fibers and emerge in the furrow between the lower border of the pons and the pyramid of the medulla oblongata. The nucleus of the facial nerve, nucleus N. facialis, is situated deeply in the reticular formation of the pons, on the dorsal aspect of the superior olivary nucleus, and the roots of the nerve derived from it pursue a remarkably tortuous course in the substance of the pons. At first they pass backward and medialward until they reach the rhomboid fossa close to the median sulcus, where they are collected into a round bundle. This passes upward and forward, producing an elevation, the colliculus facialis, in the rhomboid fossa, and then takes a sharp bend and arches lateralward through the substance of the pons to emerge at its lower border in the interval between the olive and the inferior peduncle of the medulla oblongata. 5. The nucleus of the cochlear nerve consists of a. The laterocochlear nucleus, corresponding to the tuberculum acousticum on the dorsolateral surface of the inferior peduncle, and b. The ventral or exsensory cochlear nucleus, placed between the two divisions of the nerve on the ventral aspect of the inferior peduncle. The nuclei of the vestibular nerve. A. The medial, dorsal or chief vestibular nucleus, corresponding to the lower part of the area acoustica in the rhomboid fossa. The caudal end of this nucleus is sometimes termed the descending or spinal vestibular nucleus. B. The lateral or nucleus of Dieters, consisting of large cells and situated in the lateral angle of the rhomboid fossa. The dorsolateral part of this nucleus is sometimes termed the nucleus of Bechtereau. The fibers of the vestibular nerve enter the medulla oblongata on the medial side of those of the cochlear and pass between the inferior peduncle and the spinal tract of the trigeminal. They then divide into ascending and descending fibers. The latter end by arborizing around the cells of the medial nucleus, which is situated in the area acoustica of the rhomboid fossa. The ascending fibers either end in the same manner or in the lateral nucleus, which is situated lateral to the area acoustica and farther from the ventricular floor. Some of the axons of the cells of the lateral nucleus, and possibly also the medial nucleus, are continued upward through the inferior peduncle to the roof nuclei of the opposite side of the cerebellum, to which also other fibers of the vestibular root are prolonged without interruption in the nuclei of the medulla oblongata. A second set of fibers from the medial and lateral nuclei, and partly in the tegmentum, while the remaining ascend in the medial longitudinal fasciculus to arborize around the cells of the nuclei of the ocular motor nerve. End of section 9. Section 10 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Hindbrain Part 3. The cerebellum constitutes the largest part of the hindbrain. It lies behind the pons and medulla oblongata. Between its central portion and these structures is the cavity of the fourth ventricle. It rests on the inferior occipital fossae, while above it is the tentorium cerebellae, a fold of Dura Mater which separates it from the tentorial surface of the cerebrum. It is somewhat oval in form, but constricted medially and flattened from above downward, its greatest diameter being from side to side. Its surface is not convoluted like that of the cerebrum, but is traversed by numerous curved furrows or sulci, which vary in depth at different parts, and separate the laminae of which it is composed. Its average weight in the male is about 150 grams. In the adult, the proportion between the cerebellum and cerebrum is about 1 to 8. In the infant, about 1 to 20. Lobes of the cerebellum. The cerebellum consists of 3 parts, a median and 2 lateral, which are continuous with each other and are substantially the same in structure. The median portion is constricted and is called the vermis, from its annulated appearance which it owes to the transverse ridges and furrows upon it. The lateral expanded portions are named the hemispheres. On the upper surface of the cerebellum, the vermis is elevated above the level of the hemisphere, but on the undersurface, it is sunk almost out of sight in the bottom of a deep depression between them. This depression is called the vallecula cerebellae and lodges the posterior part of the medulla oblongata. The part of the vermis on the upper surface of the cerebellum is named the superior vermis, that on the lower surface the inferior vermis. The hemispheres are separated below and behind by a deep notch, the posterior cerebellar notch, and in front by a broader, shallower notch, the anterior cerebellar notch. The anterior notch lies close to the pons and upper part of the medulla, and its superior edge encircles the inferior colliculi and the superior cerebellar peduncle. The posterior notch contains the upper part of the falx cerebelli, a fold of Dura Mater. The cerebellum is characterized by a laminated or foliated appearance. It is marked by deep, somewhat curved fissures, which extend for a considerable distance into its substance and divide it into a series of layers or leaves. The largest and deepest fissure is named the horizontal sulcus. It commences in front of the pons and passes horizontally around the free margin of the hemisphere to the middle line behind and divides the cerebellum into an upper and a lower portion. Several secondary but deep fissures separate the cerebellum into lobes, and these are further subdivided by shallower sulci, which separate the individual folia or laminae from each other. Sections across the laminae show that the folia, though differing in appearance from the convolutions of the cerebrum, are analogous to them inasmuch as they consist of central white substance covered by gray substance. The cerebellum is connected to the cerebrum, pons, and medulla oblongata, to the cerebrum by the superior peduncle, to the pons by the middle peduncle, and to the medulla oblongata by the inferior peduncles. The upper surface of the cerebellum is elevated in the middle and sloped towards the circumference, the hemispheres being connected together by a superior vermis, which assumes the form of a raised median ridge, most prominent in front, but not sharply defined from the hemispheres. The superior vermis is subdivided from before backwards into the lingula, the lobus centralis, the monticulus, and the folium vermis, and each of these, with the exception of the lingula, is continuous with the corresponding parts of the hemispheres, the lobulus centralis with the ali, the monticulus with the quadrigeminal lobules, and the folium vermis with the superior semilunar lobules. The lingula, lingula cerebelli, is a small tongue-shaped process consisting of 4 or 5 folia. It lies in front of the lobulus centralis and is concealed by it. Anteriorly, it rests on the dorsal surface of the anterior medullary velum, and its white substance is continuous with that of the velum. The lobulus centralis and ala. The lobulus centralis is a small square lobule situated in the anterior cerebellar notch. It overlaps the lingula, from which it is separated by the precentral fissure. Laterally, it extends along the upper and anterior part of each hemisphere, where it forms a wing-like prolongation, the ala lobula centralis. The monticulus and quadrangular lobules. The monticulus is the largest part of the superior vermis. Anteriorly, it overlaps the lobulus centralis, from which it is separated by the postcentral fissure. Laterally, it is continuous with the quadrangular lobule in the hemispheres. It is divided by the preclival fissure into an anterior raised part, the culmen or summit, and a posterior slope part, the clivus. The quadrangular lobule is similarly divided. The culmen and the anterior parts of the quadrangular lobules form the lobus culminis, the clivus and the posterior parts the lobus clevi. The folium vermis and superior semilunar lobule. The folium vermis, folium cacuminis, cacuminal lobe, is a short, narrow, concealed band at the posterior extremity of the vermis, consisting apparently of a single folium, but in reality marked on its upper and under surfaces by secondary fissures. Laterally, it expands in either hemisphere into a considerable lobule, the superior semilunar lobule, lobulus semilunaris superior, posterior superior lobules, which occupy the posterior third of the upper surface of the hemispheres, and is bounded below by the horizontal sulcus. The superior semilunar lobules and the folium vermis form the lobus semilunaris. The undersurface of the cerebellum presents in the middle line the inferior vermis, buried in the vallecula and separated from the hemisphere on either side by a deep groove, the sulcus valleculae. Here, as on the upper surface, there are deep fissures dividing it into separate segments or lobules, but the arrangement is more complicated, and the relation of the segments to the vermis to those of the hemispheres is less clearly marked. The inferior vermis is subdivided from below backwards into 1. The nodule, 2. the uvula, 3 the pyramid, and 4 the tuber vermis. The corresponding parts on the hemisphere are 1. The flocculus, 2. The tonsillus cerebelli, 3. The biventral lobule, and 4. The inferior semilunar lobule. The 3 main fissures are 1. The postnodular fissure, which runs transversely across the vermis between the nodule and the uvula. In the hemisphere, this fissure passes in front of the tonsil, crosses between the flocculus in front and the biventral lobule behind, and joins the anterior end of the horizontal sulcus. 2. The prepyramidal fissure crosses the vermis between the uvula in front and the pyramid behind, then curves forward between the tonsil and the biventral lobe to join the postnodular fissure. 3. The postpyramidal fissure passes across the vermis between the pyramid and the tuber vermis and, in the hemispheres, courses behind the tonsil and biventral lobules, and then along the lateral border of the biventral lobule to the postnodular sulcus. In the hemisphere, it forms the anterior boundary of the inferior semilunar lobule. The nodule and flocculus. The nodule, nodulus vermis, nodular lobe, or anterior end of the inferior vermis, abuts against the roof of the fourth ventricle, and can only be distinctly seen after the cerebellum has been separated from the medulla oblongata and pons. On either side of the nodule is a thin layer of white substance named the posterior medullary velum. It is semilunar in form, its convex border being continuous with the white substance of the cerebellum. It extends on either side as far as the flocculus. The flocculus is a prominent irregular lobule situated in front of the biventral lobule, between it and the midcerebellar peduncle. It is subdivided into a few small laminae and is connected to the inferior medullary velum by its central white core. The flocculi, together with the posterior medullary velum and nodule, constitute the lobus noduli. The uvula and tonsilla. The uvula, uvula vermis, uvular lobe, forms a considerable portion of the inferior vermis. It is separated on either side from the tonsil by the sulculus folliculae, at the bottom of which it is connected to the tonsil by a ridge of gray matter, indented on its surface by shallow furrows, and hence called the furrowed band. The tonsilla, tonsilla cerebelli, amygdaline nucleus, is a rounded mass situated in the hemispheres. Each lies in a deep fossa termed the bird's nest, nidus avis, between the uvula and the biventral lobule. The uvula and tonsillae form the lobus uvulae. The pyramid and biventral lobules constitute the lobus pyramidus. The pyramid is a conical portion forming the largest prominence of the inferior vermis. It is separated from the hemispheres by the sulcus folliculae, across which it is connected to the biventral lobule by an indistinct gray band, analogous to the furrowed band already described. The biventral lobular is triangular in shape, its apex points backwards, and is joined by the gray band to the pyramid. The lateral border is separated from the inferior semilunar lobule by the postpyramidal fissure. The base is directed forward and is on a line with the anterior border of the tonsil, and is separated from the flocculus by the postnodular fissure. The tuber vermis, tuber valvulae, and the inferior semilunar lobule, lobulus semilunaris inferior, posterior superior lobule, collectively form the lobus tuberus, tuberae lobe. The tuber vermis, the most posterior division of the interior vermis, is of small size and laterally spreads out into the large inferior semilunar lobules, which comprise at least 2/3 of the inferior surface of the hemisphere. Internal structure of the cerebellum. The cerebellum consists of white and gray substance. White substance. If a sagittal section be made through either hemisphere, the interior will be found to consist of a central stem of white substance, in the middle of which is a gray mass, the dentate nucleus. From the surface of the central white stem, a series of plates is prolonged. These are covered with gray substance and form the laminae. In consequence of the main branches from the central stem dividing and subdividing, a characteristic appearance named the arbor vitae is presented. If the sagittal section be made through the middle of the vermis, it will be found that the central stem divides into a vertical and horizontal branch. The vertical branch passes upwards to the culmen monticuli, where it subdivides freely, one of its ramifications passing forward and upward to the central lobule. The horizontal branch passes backwards to the folium vermis, greatly diminished in size in consequence of having given off large secondary branches. One, from its upper surface, ascends to the clivus monticuli, the others descend and enter the lobes of the inferior vermis, that is, the tuber vermis, the pyramid, the uvula, and the nodule. The white substance of the cerebellum include two sets of nerve fibers. 1. Projection fibers. 2. Fibrae propriae. Projection fibers. The cerebellum is connected to other parts of the brain by 3 large bundles of projection fibers, that is, to the cerebrum by the superior peduncle, to the pons by the middle peduncle, and to the medulla oblongata by the inferior peduncles. The superior cerebellar peduncles, brachia conjunctiva, 2 in number, emerge from the upper and medial portions of the white substance of the hemispheres and are placed under cover of the upper part of the cerebellum. They are joined to each other across the middle line by the anterior medullary velum and can be followed upward as far as the inferior colliculi, under which they disappear. Below, they form the upper lateral boundaries of the fourth ventricle, but as they ascend, they converge on the dorsal aspect of the ventricle and thus assist in roofing it in. The fibers of the superior peduncle are mainly derived from the cells of the dentate nucleus of the cerebellum and emerge from the hilus of this nucleus. A few arise from the cells of the smaller gray nuclei in the cerebellar white substance, and others from the cells of the cerebellar cortex. They are continued upward beneath the corpora quadrigemina, and the fibers of the 2 peduncles undergo a complete decussation ventral to the sylvian aqueduct. Having crossed the middle line, they divide into ascending and descending groups of fibers, the former ending in the red nucleus, the thalamus, and the nucleus of the ocular motor nerve, while the descending fibers can be traced as far as the dorsal part of the pons. Cajal believes them to be continued into the anterior funiculus of the medulla spinalis. As already stated, the majority of the fibers of the ventral spinocerebellar fasciculus of the medulla spinalis pass to the cerebellum, which they reach by way of the superior peduncle. The middle cerebellar peduncles, brachia pontis, are composed entirely of centripetal fibers which arise from the cells of the nuclei pontis of the opposite side and end in the cerebellar cortex. The fibers are arranged in 3 fasciculi, superior, inferior, and deep. The superior fasciculus, the most superficial, is derived from the upper transverse fibers of the pons. It is directed backward and lateralward superficial to the other 2 fasciculi, and is distributed mainly to the lobules on the inferior surface of the cerebellar hemisphere and to the parts of the superior surface adjoining the posterior and lateral margins. The inferior fasciculus is formed by the lowest transverse fibers of the pons. It passes under cover of the superior fasciculus and continued downward and backward more or less parallel with it to be distributed to the folia on the undersurface close to the vermis. The deep fasciculus comprises most of the deep transverse fibers of the pons. It is at first covered by the superior and inferior fasciculi, but crosses obliquely and appears on the medial side of the superior, from which it receives a bundle. Its fibers spread out and pass to the upper anterior cerebellar folia. The fibers of this fasciculus cover those of the restiform body. The inferior cerebellar peduncles, restiform bodies, pass at first upward and lateralward, forming part of the lateral walls of the fourth ventricle, and then bend abruptly backwards to enter the cerebellum between the superior and middle peduncles. Each contains the following fasciculi. 1. The dorsal spinocerebellar fasciculus of the medulla spinalis, which ends mainly in the superior vermis. 2. fibers from the gracillin cuneate nuclei of the same side and of the opposite sides. 3. Fibers from the opposite olivary nuclei. 4. Crossed and uncrossed fibers from the reticular formation of the medulla oblongata. 5. Vestibular fibers, derived partly from the vestibular division of the acoustic nerve and partly from the nuclei in which this division ends. These fibers occupy the medial segment of the inferior peduncle and divide into ascending and descending groups of fibers. The ascending fibers partly end in the roof nucleus on the opposite side of the cerebellum. 6. Cerebellobulbar fibers, which come from the opposite roof nucleus and probably from the dentate nucleus, and are said to end in the nucleus of Dieters and in the formatio reticularis of the medulla oblongata. 7. Some fibers from the ventral spinocerebellar fasciculus are said to join the dorsal spinocerebellar fasciculus. The anterior medullary velum. Velum medullare anterius, valve of Vincennes, superior medullary velum, is a thin, transparent lamina of white substance which stretches between the superior peduncle on the dorsal surface of its lower half. The folia and lingula are prolonged. It forms, together with the superior peduncle, the roof of the upper part of the fourth ventricle. It is narrow above, where it passes beneath the inferior colliculi, and broader below, where it is continuous with the white substance of the superior vermis. A slightly elevated ridge, the frenulum veli, descends upon its upper part from between the inferior colliculi, and on either side of this the trochlear nerve emerges. The posterior medullary velum, velum medullare posterius, inferior medullary velum, is a thin layer of white substance prolonged from the white center of the cerebellum above and on either side of the nodule. It forms a part of the roof of the fourth ventricle. Somewhat semilunar in shape, its convex edge is continuous with the white substance of the cerebellum, while its thin concave margin is apparently free. In reality, however, it is continuous with the epithelium of the ventricle, which is prolonged downward from the posterior medullary velum to the ligulae. The two medullary vela are in contact with each other along their line of emergence from the white substance of the cerebellum, and this line of contact forms the summit of the roof of the fourth ventricle, which, in a vertical section through the cavity, appears as a pointed angle. The fibrae propriae of the cerebellum are of two kinds. 1. Commissural fibers, which cross the middle lines at the anterior and posterior parts of the vermis and connect the opposite halves of the cerebellum. 2. Arcuate or association fibers, which connect adjacent laminae with each other. Gray substance. The gray substance of the cerebellum is found in two situations. 1. On the surface forming the cortex. 2. As independent masses in the anterior. 1. The gray substance of the cortex presents a characteristic foliated appearance due to the series of laminae which are given off from the central white substance. These, in turn, give off secondary laminae which are covered by gray substance. Externally, the cortex is covered by pia mater. Internally is the medullary center, consisting mainly of nerve fibers. End of section 10. Section 11 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Recording by M.L. Cohen. Anatomy of the Human Body, Part 4 by Henry Gray. The Hindbrain, Part 4. Microscopic appearance of the cortex. The cortex consists of two layers, that is, an external gray molecular layer and an internal rust-colored nuclear layer. Between these is an incomplete stratum of cells which are characteristic of the cerebellum, that is, the cells of Purkinje. The external gray or molecular layer consists of fibers and cells. The nerve fibers are delicate fibrillae. and are derived from the following sources. A. The dendrites and axon collaterals of Purkinje cells. B. Fibers from the cells of the nuclear layer. C. Fibers from the central white substance of the cerebellum. D. Fibers derived from cells in the molecular layer itself. In addition to these are other fibers, which have a vertical direction and are the processes of the large neuroglial cells situated in the nuclear layer. They pass outward to the periphery of the gray matter, where they expand into little conical enlargements, which form a sort of limiting membrane beneath the pia mater, analogous to the membrana limitans interna in the retina formed by the sustentacular fibers of Mueller. The cells of the molecular layer are small and are arranged in two striata, an outer and an inner. They all possess branched axons. Those of the inner layer are termed basket cells. They run for some distance parallel with the surface of the folium, giving off collaterals which pass in a vertical direction towards the bodies of the Purkinje cells, around which they become enlarged and form basket-like networks. The cells of Purkinje form a single stratum of large flask-like shells at the junction of the molecular and nuclear layers, their bases resting against the latter. In fishes and reptiles, they are arranged in several layers. The cells are flattened in a direction transverse to the long axis of the folium, and thus appear broad in sections carried across the folium, and fusiform in sections parallel to the long axis of the folium. From the neck of the flask, one or more dendrites arise and pass into the molecular layer, where they subdivide and form an extremely rich arborescence, the various subdivisions of the dendrites being covered by lateral spine-like processes. The arborescence is not circular, but, like the cell, is flattened at right angles to the long axis of the folium. In other words, it does not resemble a round bush, but has been aptly compared by Obersteiner to the branches of a fruit tree trained against a trellis or a wall. Hence, in sections carried across the folium, the arborescence is broad and expanded, where in those which are parallel to the long axis of the folium, the arborescence, like the cell itself, is seen in profile and is limited to a narrow area. From the bottom of the flask-shaped cell, the axon arises. This passes through the nuclear layer and, becoming medullated, is continued as a nerve fiber in the subjacent white substance. As this axon traverses the granular layer, it gives off fine collaterals, some of which run back into the molecular layer. The internal, rust-colored or nuclear layer is characterized by containing numerous small nerve cells of a reddish-brown color, together with many nerve fibrils. Most of the cells are nearly spherical and provided with short dendrites which spread out in a spider-like manner in the nuclear layer. Their axons pass outward into the molecular layer and, bifurcating at right angles, run for some distance parallel with the surface. In the outer part of the nuclear layer are some larger cells of the type 2 of Golgi. Their axons undergo frequent division as soon as they leave the nerve cells and pass into the nuclear layer, while their dendrites ramify chiefly in the molecular layer. Finally, in the gray substance of the cerebellar cortex, there are fibers which come from the white center and penetrate the cortex. The cell origin of these fibers is unknown, though it is believed that it is probably in the gray substance of the Medulla Spinalis. Some of these fibers end in the nuclear layer by dividing into numerous branches, on which are to be seen peculiar moss-like appendages. Hence, they have been termed by Raymond y Cajal the moss fibers. They form an arborescence around the cells of the nuclear layer and are said to come from fibers in the inferior peduncle. Other fibers, the clinging or tendril fibers, derived from the medullary center, can be traced into the molecular layer, where their branches cling around the dendrites of Purkinje cells. They are said to come from fibers of the middle peduncle. 2. The independent centers of gray substance in the cerebellum are 4 in number on either side. One is of large size and is known as the nucleus dentatus. The other 3, much smaller, are situated near the middle of the cerebellum and are known as the nucleus emboliformis, nucleus globosus, and nucleus fastigii. The nucleus dentatus is situated a little to the medial side of the center of the stem of the white substance of the hemisphere. It consists of an irregularly folded lamina of grayish-yellow color, containing white fibers, and presenting on its anterior medial aspect an opening, the hilus, from which most of the fibers of the superior peduncle emerge. The nucleus emboliformis lies immediately to the medial side of the nucleus dentatus and partly covering its hilus. The nucleus globosus is an elongated mass directed anterior-posteriorly and placed medial to the preceding. The nucleus fastigii is somewhat larger than the other two, and is situated close to the middle line at the anterior end of the superior vermis, and immediately over the roof of the fourth ventricle, from which it is separated by a thin layer of white substance. The cerebellum is concerned with the coordination of movements necessary in equilibration, locomotion, and prehension. In it terminate pathways conducting impulses of muscle sense, tendon sense, joint sense, and equilibratory disturbances. With the exception of the ventral spinocerebellar fasciculus, these impulses enter through the inferior peduncle. The reflex arc is completed by fibers in the superior peduncle, which pass to the red nucleus and the thalamus, and thence, by additional neurons, cerebrospinal tract, to the motor centers. The exact function of its different parts are still quite uncertain, owing to the contradictory nature of the evidence furnished by 1. Ablation experiments upon animals, and 2. Clinical observation in man of the effects produced by abscesses or tumors affecting different portions of the organ. The fourth ventricle, ventriculus courtis. The fourth ventricle, or cavity of the hindbrain, is situated in front of the cerebellum and behind the pons and upper half of the medulla oblongata. Developmentally considered, the fourth ventricle consists of three parts: a superior belonging to the isthmus rhombencephalon, an intermediate to the metencephalon, and an inferior to the myelencephalon. It is lined by ciliated epithelium and is continuous below with the central canal of the medulla oblongata. Above, it communicates by means of a passage termed the cerebral aqueduct with the cavity of the third ventricle. It presents four angles and possesses a roof or dorsal wall, a floor or ventral wall, and lateral boundary. Angles. The superior angle is on a level with the upper border of the pons and is continuous with the lower end of the cerebral aqueduct. The inferior angle is on a level with the lower end of the olive and opens into the central canal of the medulla oblongata. Each lateral angle corresponds with the point of meeting of the brachia and inferior peduncle. A little below the lateral angles, on a level with the striae medullares, the ventricular cavity is prolonged outward in the form of two narrow lateral recesses, one on either side. These are situated between the inferior peduncles and the flocculi, and reach as far as the detachments of the glossopharyngeal and vagus nerves. Lateral boundaries. The lower part of each lateral boundary is constituted by the clava, the fasciculus cuneatus, and the inferior peduncle, the upper part by the middle and the superior peduncle. Roof or dorsal wall. The upper portion of the roof is formed by the superior peduncle and the anterior medullary velum, the lower portion by the posterior medullary velum, the epithelial lining of the ventricle covered by the telechoroidea inferior, the tanae of the fourth ventricle, and the obex. The superior peduncle, on emerging from the central white substance of the cerebellum, pass upward and forward, forming at first the lateral boundaries of the upper part of the cavity. On approaching the inferior colliculi, they converge and their medial portions overlap the cavity and form part of its roof. The anterior medullary velum fills the angular interval between the superior peduncle and is continuous behind with the central white substance of the cerebellum. It is covered on its dorsal surface by the lingula of the superior vermis. The posterior medullary velum is continued downward and forward from the central white substance of the cerebellum in front of the nodule and tonsils, and ends inferiorly in a thin, concave, somewhat ragged margin. Below this margin, the roof is devoid of nervous matter, except in the immediate vicinity of the lower lateral boundaries of the ventricle, where two narrow white bands, the tanae of the fourth ventricle, ligulae, appear. These bands meet over the inferior angle of the ventricle in a thin triangular lamina, the obex. The non-nervous part of the roof is formed by the epithelial lining of the ventricle, which is prolonged downward as a thin membrane, from the deep surface of the posterior medullary velum to the corresponding surface of the obex and tanae, and thence on to the floor of the ventricular cavity. It is covered and strengthened by a portion of the pia mater, which is named the telechoroidea of the fourth ventricle. The tanae of the fourth ventricle, tinae ventriculi corto, ligula, are two narrow bands of white matter, one on either side, which complete the lower part of the roof of the cavity. Each consists of a vertical and a horizontal part. The vertical part is continuous below the obex with the clava, to which it is adherent by a lateral border. The horizontal portion extends transversely across the inferior peduncle, below the striae medullares, and roofs in the lower and posterior part of the lateral recess. It is attached by its lower margin to the inferior peduncle and partly encloses the choroid plexus, which, however, projects beyond it like a cluster of grapes, and hence this part of the tenea has been termed the cornucopia, Bochdelic. The obex is a thin triangular gray lamina which roofs in the lower angle of the ventricle and is attached by its lateral margins to the clavae. The telechoroidea of the fourth ventricle is the name applied to the triangular fold of Pia Mater, which is carried upward between the cerebellum and the medulla oblongata. It consists of two layers which are continuous with each other in front and are more or less adherent throughout. The posterior layer covers the anterior-inferior surface of the cerebellum, while the anterior is applied to the structures which form the lower part of the roof of the ventricle, and is continued inferiorly with the Pia Mater on the inferior peduncles and closed part of the medulla. Choroid plexuses. These consist of two highly vascular inflections of the telechoroidea, which invaginate the lower part of the roof of the ventricle and are everywhere covered by the epithelial lining of the cavity. Each consists of a vertical and a horizontal portion. The former lies close to the middle line, and the latter passes into the lateral recess and projects beyond its apex. The vertical parts of the plexuses are distinct from each other, but the horizontal portions are joined in the middle line, and hence the entire structure presents the form of the letter T, the vertical limb of which, however, is double. Openings in the roof. In the roof of the fourth ventricle, there are three openings, a medial and two lateral. The medial aperture, foramen majandi, is situated immediately above the inferior angle of the ventricle. The lateral apertures, foramina ovlushka, are found at the extremities of the lateral recesses. By means of these three openings, the ventricle communicates with the subarachnoid cavity, and the cerebral spinal fluid can circulate from the one to the other. Rhomboid fossa. Fossa rhomboidea, floor of the fourth ventricle. The anterior part of the fourth ventricle is named, from its shape, the rhomboid fossa, and its anterior wall, formed by the back of the pons and medulla oblongata, constitutes the floor of the fourth ventricle. It is covered by a thin layer of gray substance, continuous with that of the medulla spinalis. Superficial to this is a thin lamina of neuroglia which constitutes the ependema of the ventricle and supports a layer of ciliated epithelium. The fossa consists of three parts, superior, intermediate, and inferior. The superior part is triangular in shape and limited laterally by the superior cerebellar peduncle. Its apex, directed upward, is continuous with the cerebral aqueduct. Its base is represented by an imaginary line at the level of the upper ends of the superior foveae. The intermediate part extends from this level to that of the horizontal portions of the tinea of the ventricle. It is narrow above, where it is limited laterally by the medial peduncle, but widens below and is prolonged into the lateral recesses of the ventricle. The inferior part is triangular, and its downwardly directed apex, named the calamus scriptorius, is continuous with the central canal of the closed port of the medulla oblongata. The rhomboid fossa is divided into symmetrical halves by a median sulcus, which reaches from the upper to the lower angles of the fossa and is deeper below than above. On either side of this sulcus is an elevation, the medial eminence, bounded laterally by a sulcus, the sulcus limitans. In the superior part of the fossa, the medial eminence has a width equal to that of the corresponding half of the fossa, but opposite the superior fovea, it forms an elongated swelling, the colliculus fascialis, which overlies the nucleus of the abducent nerve, and is, in part at least, produced by the ascending portion of the root of the facial nerve. In the inferior part of the fossa, the medial eminence assumes the form of a triangular area, the trigonum hypoglossai. When examined under water with a lens, this trigone is seen to consist of a medial and a lateral area separated by a series of oblique furrows. The medial area corresponds with the upper part of the nucleus of the hypoglossal nerve, the lateral, with a small nucleus, the nucleus intercalatus. The sulcus limitans forms the lateral boundary of the medial eminence. In the superior part of the rhomboid fossa, it corresponds with the lateral limit of the fossa and presents a bluish-gray area, the locus ceruleus, which owes its color to an underlying patch of deeply pigmented nerve cells termed the substantia ferruginea. At the level of the colliculus facialis, the sulcus limitans widens into a flattened depression, the superior fovea, and in the inferior part of the fossa appears as a distinct dimple, the inferior fovea. Lateral to the foveae is a rounded elevation named the area acoustica, which extends into the lateral recess and there forms a feebly marked swelling, the tuberculum acousticum. Winding around the inferior peduncle and crossing the area acoustica and the medial eminence are a number of white strands, the striae medullares, which form a portion of the cochlear division of the acoustic nerve and disappear into the median sulcus. Below the inferior fovea and between the trigonum hypoglossae and the lower part of the area acoustica is a triangular dark field, the alis cinerea, which corresponds to the sensory nucleus of the vagus and glossopharyngeal nerves. The lower end of the alis cinerea is crossed by a narrow translucent ridge, the funiculus separans, and between this funiculus and the clava is a small tongue-shaped area, the area postrema. On section, it is seen that the funiculus separans is formed by a strip of thickened ependema, and the area postrema by loose, highly vascular neuroglial tissue containing nerve cells of moderate size. End of section 11. Section 12 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The midbrain. The midbrain or mesencephalon. The midbrain or mesencephalon is the short constricted portion which connects the pons and cerebellum with the thalencephalon and cerebral hemispheres. It is directed upward and forward and consists of 1. A ventrolateral portion composed of a pair of cylindrical bodies named as cerebral peduncles. 2. A dorsal portion consisting of 4 rounded eminences named the corpora quadrigemina. And 3. An intervening passage or tunnel, the cerebral aqueduct, which represents the original cavity of the midbrain and connects the 3rd with the 4th ventricles. The cerebral peduncles, pedunculus cerebri, crus cerebri, are 2 cylindrical masses situated at the base of the brain and largely hidden by the temporal lobes of the cerebrum, which must be drawn aside or removed in order to expose them. They emerge from the upper surface of the pons, one on either side of the middle line, and, divergent as they pass upward and forward, disappear into the substance of the cerebral hemispheres. The depressed area between the crura is termed the interpeduncular fossa and consists of a layer of grayish substance, the posterior perforated substance, which is pierced by small apertures for the transmission of blood vessels. Its lower part lies on the ventral aspect of the medial portions of the tegmenta and contains a nucleus named the interpeduncular ganglion. Its upper part assists in forming the floor of the 3rd ventricle. The ventral surface of each peduncle is crossed from the medial to the lateral side by the superior cerebellar and posterior cerebral arteries. Its lateral surface is in relation to the gyrus hippocampi of the cerebral hemisphere and is crossed from behind forward by the trochlear nerve. Close to the point of disappearance of the peduncle into the cerebral hemisphere, the optic tract winds forward around its ventrolateral surface. The medial surface of the peduncle forms the lateral boundary of the interpeduncular fossa and is marked by a longitudinal furrow, the oculomotor sulcus, from which the roots of the oculomotor nerve emerge. On the lateral surface of each peduncle, there is a second longitudinal furrow, termed the lateral sulcus. The fibers of the lateral lemniscus come to the surface in this sulcus and pass backward and upward to disappear under the inferior colliculus. Structure of the cerebral peduncles. On transverse section, each peduncle is seen to consist of a dorsal and a ventral part, separated by a deeply pigmented lamina of gray substance termed the substantia nigra. The dorsal part is named the tegmentum, the ventral the base or crustum. The 2 bases are separated from each other but the tegmenta are joined in the median plane by a forward prolongation of the raphe of the pons. Laterally, the tegmenta are free. Dorsally, they blend with the corpora quadrigemina. The base, basis pedunculi, crusta or pes, is semilunar on transverse section and consists almost entirely of longitudinal bundles of efferent fibers, which arise from the cells of the cerebral cortex and are grouped into three principal sets, that is, cerebrospinal, frontopontine, and temporopontine. The cerebrospinal fibers, derived from the cells of the motor area of the cerebral cortex, occupy the middle 3/5 of the base. They are continued partly to the nuclei of the motor cranial nerves, but mainly into the pyramids of the medulla oblongata. The frontopontine fibers are situated in the medial 1/5 of the base. They arise from the cells of the frontal lobe and end in the nuclei of the pons. The temporopontine fibers are lateral to the cerebrospinal fibers. They originate in the temporal lobe and end in the nuclei pontis. The substantia nigra, intercalatum, is a layer of gray substance containing numerous deeply pigmented multipolar nerve cells. It is semilunar on transverse section, its concavity being directed towards the tegmentum. From its convexity, prolongations extend between the fibers of the base of the peduncle. Thicker medially than laterally, it reaches from the ocular motor sulcus to the lateral sulcus, and extends from the upper surface of the pons to the subthalamic region. Its medial part is traversed by the fibers of the oculomotor nerve as these stream forward to reach the oculomotor sulcus. The connections of the cells of the substantia nigra have not been definitively established. It receives collaterals from the medial lemniscus and the pyramidal bundles. Bechtereau is of the opinion that the fibers from the motor area of the cerebral cortex form synapses with the cells whose axons pass to the motor nucleus of the trigeminal nerve and serve for the coordination of the muscles of mastication. The tegmentum is continuous below with the reticular formation of the pons and, like it, consists of longitudinal and transverse fibers together with a considerable amount of gray substance. The principal gray masses of the tegmentum are the red nucleus and the interpeduncular ganglion. Of its fibers, the chief longitudinal tracts are the superior peduncle, the medial longitudinal fasciculus, and the lemniscus. Gray substance. The red nucleus is situated in the anterior part of the tegmentum and is continued upward into the posterior part of the subthalamic regions. In sections at the level of the superior colliculus, it appears as a circular mass which is traversed by the fibers of the oculomotor nerve. It receives many terminals and collaterals from the superior cerebellar peduncle, also collaterals from the ventral longitudinal bundle, from Gudin's bundle, and the median lemniscus. The axons of its larger cells cross the middle line and are continued downward into the lateral funiculus of the Medulla Spinalis as the rubrospinal tract, those of its smaller cells and mainly in the thalamus. The rubrospinal tract forms an important part of the pathway from the cerebellum to the lower motor centers. The interpeduncular ganglion is a median collection of nerve cells situated in the ventral part of the tegmentum. The fibers of the fasciculus retroflexus of Meinert, which have their origin in the cells of the ganglion habenula, end in it. Besides the two nuclei I mentioned, there are small collections of cells which form the dorsal and ventral nuclei, and the central nucleus or nucleus of the raphe. White substance. 1. The origin and course of the superior peduncle have already been described. 2. The medial posterior longitudinal fasciculus is continuous below with the proper fasciculi of the anterior and lateral funiculi of the medulla spinalis. In the medulla oblongata and pons, it runs close to the middle line near the floor of the fourth ventricle. In the midbrain, it is situated on the ventral aspect of the cerebral aqueduct, below the nuclei of the oculomotor and trochlear nerves. Its connections are imperfectly known, but it consists largely of ascending and descending intersegmental or association fibers, which connect in the nuclei of the hindbrain and midbrain to each other. Many of the fibers arise in Dieter's nucleus, lateral vestibular nucleus, and divide into ascending and descending branches which send terminals and collaterals to the motor nuclei of the cranial and spinal nerves. Its spinal portion is located in the anterior funiculus and is known as the Vestibulospinal Fasciculus. Other fibers pass to the median longitudinal bundle from cells in the reticular formation of the medulla, pons, and midbrain, and also from certain large cells in the terminal nucleus of the trigeminal nerve. According to Edinger, it extends to the so-called nucleus of the posterior longitudinal bundle in the hypothalamic region. But this is uncertain, and the fibers above the nucleus of the oculomotor are smaller in diameter than the rest of the bundle. According to Held, fibers from the posterior commissure can be traced into the posterior longitudinal bundle, and according to the same author, many of the descending fibers arise in the superior colliculus, and, after decussating in the middle line, end in the motor nuclei of the pons and medulla oblongata. These fibers from the superior colliculus probably pass into the ventral longitudinal bundle. Fibers are said to pass through the medial longitudinal fasciculus from the nucleus of the abducent into the oculomotor nerve of the opposite side, and through this nerve to the rectus medialis oculi. Fraser, however, denies the existence of such fibers. Again, fibers are said to be prolonged through this fasciculus from the nucleus of the oculomotor nerve into the facial nerve, and are distributed to the orbicularis oculi, the corrugator, and the frontalis. The ventral longitudinal bundle consists for the most part of the Tectospinal Fasciculus, and arises from the superior colliculus. The fibers arch ventrally around the central gray matter and cross the midline in the fountain decussation of Monert. They then descend in the tegmentum, part of them passing through the red nucleus ventral to the medial longitudinal bundle. In the medulla oblongata and spinal cord, its fibers are more or less intermingled with the medial longitudinal bundle and the rubrospinal tract. It descends in the adjoining region of the ventral and lateral funiculi. Collaterals and terminals are given off to the red nucleus, and probably other nuclei of the brainstem, and to the anterior column of the spinal cord. It is probably concerned in optic reflexes. The medial lemniscus, or medial fillet. The fibers of the medial lemniscus take origin in the gracile and cuneate nuclei of the medulla oblongata, and, as internal arcuate fibers, they cross to the opposite side in the sensory decussation. They then pass into interolivary stratum upward through the medulla oblongata, in which they are situated behind the cerebrospinal fibers and between the olives. In the pons and lower part of the midbrain, it occupies the ventral part of the reticular formation and tegmentum close to the raphe, while above it gradually shifts to the dorsolateral part of the tegmentum in the angle between the red nucleus and the substantia nigra. In the pons, it assumes a flattened ribbon-like appearance and is placed dorsal to the trapezium. As the lemniscus ascends, it receives additional fibers from the terminal sensory nuclei of the cranial nerves of the opposite side. Many of the fibers which arise from the terminal sensory nuclei of the cranial nerves pass upward in the formatio reticularis as a separate bundle, known as the central tract of the cranial nerves, to the thalamus. Many fibers either terminate in or send off collaterals to the gray matter of the medulla, the pons, and the midbrain. Large numbers of fibers pass to or from the substantia nigra. Many collaterals enter the red nucleus, and other fibers are said to run to the superior colliculus. The great bulk of the fibers, however, enter the ventrolateral portion of the thalamus, give off collaterals to the posterior semilunar nucleus, and then terminate in the principal central nucleus of the thalamus. In the cerebral peduncle, a few of its fibers pass upward in the lateral part of the base of the peduncle on the dorsal aspect of the temporopontine fibers, and reach the lentiform nucleus and the insula. The greater part of the medial lemniscus, on the other hand, is prolonged through the tegmentum, and most of its fibers end in the thalamus. Probably some are continued directly through the occipital part of the internal capsule to the cerebral cortex. From the cells of the thalamus, a relay of fibers is prolonged to the cerebral cortex. The medial lemniscus may be considered as the upward continuation of the posterior funiculus of the spinal cord, and to convey conscious impulses of muscle sense and tactile discrimination. The central or thalamic tract of the cranial nerves is closely associated with the medial lemniscus. The fibers of the spinothalamic fasciculi are continued from the spinal cord into this tract, which passes upward in the reticular formation and the tegmentum to the thalamus, along the dorsal side of the median lemniscus. It receives fibers from the opposite terminal sensory nuclei of the vagus, glossopharyngeal, facial, trigeminal, and probably the vestibular nerves. Many of the secondary sensory fibers of the trigeminal cross the raphe from its terminal nucleus and pass upward to the thalamus by a more or less separate but closely associated pathway known as the central tract of the trigeminal nerve, which also lies on the dorsal aspect of the lemniscus. These two tracts give off collaterals to the posterior semilunar nucleus of the thalamus and terminate in the anterior semilunar nucleus of the ventrolateral region of the thalamus, sending collaterals into the zona incerta. The fibers of the rubrospinal tract, bundle of Monaco, arise in the red nucleus, cross the midline in the decussation of Forel, and pass downward in the formatio reticularis of the brainstem to the lateral funiculus of the spinal cord, ventral to the crossed pyramidal tract. The lateral lemniscus, lemniscus lateralis, comes to the surface of the midbrain along its lateral sulcus and disappears under the inferior colliculus. It consists of fibers from the terminal nuclei of the cochlear division of the acoustic nerve, together with others from the superior olivary and trapezoid nuclei. Most of these fibers are crossed, but some are uncrossed. Many of them pass to the inferior colliculus of the same or opposite side, but others are prolonged to the thalamus and thence through the occipital part of the internal capsule to the middle and superior temporal gyri. The corpora quadrigemina are four rounded eminences which form the dorsal part of the midbrain. They are situated above and in front of the anterior medullary velum and superior peduncle, and below and behind the third ventricle and posterior commissure. They are covered by the splenium of the corpus callosum and are partly overlapped on either side by the medial angle or pulvinar of the posterior end of the thalamus. On the lateral aspect, under cover of the pulvinar, is an oval eminence named the medial geniculate body. The corpora quadrigemina are arranged in pairs, superior and inferior colliculi, and are separated from one another by a crucial sinus. The longitudinal part of this sulcus expands superiorly to form a slight depression which supports the pineal body, a cone-like structure which projects backwards from the thalamoencephalon and partly obscures the superior colliculi. From the inferior end of the longitudinal sulcus, a white band, termed the frenulum veli, is prolonged downward to the anterior medullary velum. On either side of this band, the trochlear nerve emerges and passes forward on the lateral aspect of the cerebral peduncle to reach the base of the brain. The superior colliculi are larger and darker in color than the inferior, and are oval in shape. The inferior colliculi are hemispherical and somewhat more prominent than the superior. The superior colliculi are associated with the sense of sight, the inferior with that of hearing. From the lateral aspect of each colliculus, a white band, termed the brachium, is prolonged upward and forward. The superior brachium extends lateralward from the superior colliculus, and, passing between the pulvinar and the medial geniculate body, is partly continued into an eminence called the lateral geniculate body, and partly into the optic tract. The inferior brachium passes forward and upward from the inferior colliculus and disappears under cover of the medial geniculate body. In close relationship with the corpora quadrigemina are the superior peduncles, which emerge from the upper and medial parts of the cerebellar hemispheres. They run upward and forward and, passing under the inferior colliculi, enter the tegmenta as already described. Structure of the corpora quadrigemina. The inferior colliculus, colliculus inferior, inferior quadrigeminal body, postgemina, consists of a compact nucleus of gray substance containing large and small multipolar nerve cells, and more or less completely surrounded by white fibers derived from the lateral lemniscus. Most of these fibers end in the gray nucleus of the same side, but some cross the middle line and end in that of the opposite side. From the cells of the gray nucleus, fibers are prolonged through the inferior brachium into the tegmentum of the cerebral peduncle, and are carried to the thalamus and the cortex of the temporal lobe. Other fibers cross the middle line and end in the opposite colliculus. The superior colliculus, colliculus superior, superior quadrigeminal body, pregemina, is covered by a thin stratum, stratum zonale, of white fibers, the majority of which are derived from the optic tract. Beneath this is the stratum cinereum, a cap-like layer of gray substance, thicker in the center than at the circumference, and consisting of numerous small multipolar nerve cells embedded in a fine network of nerve fibers. Still deeper is the stratum opticum, containing large multipolar nerve cells separated by numerous fine nerve fibers. Finally, there is the stratum lemnisci, consisting of fibers derived partly from the lemniscus and partly from the cells of the stratum opticum. Interspersed among these fibers are many large multipolar nerve cells. The two last named strata are sometimes termed the gray-white layers, from the fact they consist of both gray and white substance. Of the afferent fibers which reach the superior colliculus, some are derived from the lemniscus, but the majority have their origins in the retina and are conveyed to it through the superior brachium. All of them end by arborizing around the cells of the gray substance. Of the efferent fibers, some cross the middle line to the opposite colliculus, many ascend through the superior brachium, and finally reach the cortex of the occipital lobe of the cerebrum, while others, after undergoing decussation, fountain decussation of Meinert, form the Tectospinal Fasciculus, which descends through the formatio reticularis of the midbrain, pons, and Medulla Oblongata into the Medulla Spinalis, where it is found partly in the anterior funiculus and partly intermingled with the fibers of the rubrospinal tract. The corpora quadrigemina are larger in the lower animals than in man. In fishes, reptiles, and birds they are hollow, and only 2 in number. Corpora bigemina. They represent the superior colliculi of mammals, and are frequently termed the optic lobes, because of their intimate connection with the optic tracts. The cerebral aqueduct, Aqueductus cerebri, aqueduct of Sylvius, is a narrow canal, about 15 millimeters long, situated between the corpora quadrigemina and tegmenta, and connecting the third with the fourth ventricle. Its shape, as seen in transverse section, varies at different levels, being T-shaped, triangular above, and oval in the middle. The central part is slightly dilated, and was named by Retzius the ventricle of the midbrain. It is lined by ciliated columnar epithelium, and is surrounded by a layer of gray substance named the central gray stratum. This is continuous below with the gray substance in the rhomboid fossa, and above with that of the third ventricle. Dorsally, it is partly separated from the gray substance of the quadrigeminal bodies by the fibers of the lemniscus. Ventral to it are the medial longitudinal fasciculus and the formatio reticularis of the tegmentum. Scattered throughout the central gray stratum are numerous nerve cells of various sizes, interlaced by a network of fine fibers. Besides these scattered cells, it contains 3 groups which constitute the nuclei of the oculomotor and trochlear nerves and the nucleus of the mesencephalic root of the trigeminal nerve. The nucleus of the trigeminal nerve extends along the entire length of the aqueduct and occupies the lateral part of the gray stratum, while the nuclei of the oculomotor and trochlear nerves are situated in its ventral part. The nucleus of the oculomotor nerve is about 10 millimeters long and lies under the superior colliculus, beyond which, however, it extends for a short distance into the gray substance of the third ventricle. The nucleus of the trochlear nerve is small and nearly circular, and is on a level with the plane carried transversely through the upper part of the inferior colliculus. End of section 12. Section 13 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 1. The forebrain or prosencephalon. The forebrain or prosencephalon consists of 1. The diencephalon, corresponding in a large measure to the third ventricle and the structures which bound it, and 2. The telencephalon, comprising the largest part of the brain, that is, the cerebral hemispheres. These hemispheres are intimately connected with each other across the middle line, and each contains a large cavity named the lateral ventricle. The lateral ventricles communicate through the intervertebral foramen with the third ventricle, but are separated from each other by a medial septum, the septum pellucidum. This contains a slit-like cavity which does not communicate with the ventricles. The diencephalon. The diencephalon is connected above and in front with the cerebral hemispheres, behind with the midbrain. Its upper surface is concealed by the corpus callosum and is covered by a fold of pia mater named the telechoroidea of the third ventricle. Inferiorly, it reaches to the base of the brain. The diencephalon comprises 1. The thalamencephalon, 2. The pars mammillaris hypothalami, and 3. The posterior part of the third ventricle. For descriptive purposes, however, it is more convenient to consider the whole of the third ventricle and its boundaries together. This necessitates the inclusion under this heading of the pars optica hypothalami and the corresponding part of the third ventricle, structures which properly belong to the telencephalon. The thalamencephalon. The thalamencephalon comprises 1. The thalamus, 2. The metathalamus or corpora geniculata, and 3. The epithalamus, consisting of the trigone habenulae, the pineal body, and the posterior commissure. The thalami, optic thalamus, are 2 large ovoid masses situated one on either side of the third ventricle and reaching for some distance behind that cavity. Each measures about 4 centimeters in length and presents 2 extremities, an anterior and a posterior, and 4 surfaces, superior, inferior, medial, and lateral. The anterior extremity is narrow. It lies close to the middle line and forms the posterior boundary of the interventricular foramen. The posterior extremity is expanded, directed backward and lateralward, and overlaps the superior colliculus. Medially, it presents an angular prominence to pulvinar, which is continued laterally into an oval swelling, the lateral geniculate body, while beneath the pulvinar, but separated from it by the superior brachium, is a second oval swelling, the medial geniculate body. The superior surface is free, slightly convex, and covered by a layer of white substance, termed a stratum zonale. It is separated laterally from the caudate nucleus by a white band, the stria terminalis, and by the terminal vein. It is divided into a medial and a lateral portion by an oblique shallow furrow, which runs from behind forward and medialward and corresponds with the lateral margin of the fornix. The lateral part forms a portion of the floor of the lateral ventricle and is covered by the epithelial lining of this cavity. The medial part is covered by the telechoroidea of the third ventricle and is destitute of an epithelial covering. In front, the superior is separated from the medial surface by a salient margin, the tinea thalami, along which the epithelial lining of the third ventricle is reflected onto the undersurface of the telechoroidea. Behind, it is limited medially by a groove, the sulcus habenulae, which intervenes between it and a small triangular area termed the trigrome habenulae. The inferior surface rests upon and is continuous with the upward prolongation of the tegmentum, subthalamic tegmental region, in front of which it is related to substantia innominata of Meinert. The medial surface constitutes the upper part of the lateral wall of the third ventricle and is connected to the corresponding surface of the opposite thalamus by a flattened gray band, the massa intermedia, middle or gray commissure. This mass averages about 1 centimeter in its anterior-posterior diameter. It sometimes consists of 2 parts and occasionally is absent. It contains nerve bundles and nerve fibers. A few of the latter may cross the middle line, but most of them pass towards the middle line and then curve lateralward on the same side. The lateral surface is in contact with a thick band of white substance which forms the occipital part of the internal capsule and separates the thalamus from the lentiform nucleus of the corpus striatum. Structure. The thalamus consists chiefly of gray substance, but its upper surface is covered by a layer of white substance termed a stratum zonale, and its lateral surface by a similar layer termed the lateromedullary lamina. Its gray substance is incompletely subdivided into 3 parts, anterior, medial, and lateral, by a white layer, the medial medullary lamina. The anterior part comprises the anterior tubercle. The medial part lies next to the lateral wall of the third ventricle, while the lateral and largest part is interposed between the medullary laminae and includes the pulvinar. The lateral part is traversed by numerous fibers which radiate from the thalamus into the internal capsule and pass through the latter into the cerebral cortex. These 3 parts are built up of numerous nuclei, the connections of many of which are imperfectly known. Connections. The thalamus may be regarded as a large ganglionic mass in which the ascending tracts of the tegmentum and a considerable portion of the fibers of the optic tract end, and from the cells in which numerous fibers, thalamocortical, take origin and radiate to almost every part of the cerebral cortex. The lemniscus, together with other longitudinal strands of the tegmentum, enters its ventral part, the thalamo-mammary fasciculus, bundle of Vic d'Azur, from the corpus mammillare, enters in its anterior tubercle, while many of its fibers of the optic tract terminate in its posterior ends. The thalamus also receives numerous fibers, corticothalamic, from the cells of the cerebral cortex. The fibers that arise from the cells of the thalamus form 4 principal groups or stalks. A. Those of the anterior stalk pass through the frontal part of the internal capsule to the frontal lobe. B. The fibers of the posterior stalk, optic radiations, arise in the pulvinar and are conveyed through the occipital part of the internal capsule to the occipital lobe. C. The fibers of the inferior stalk leave the undermedial surface of the thalamus and pass beneath the lentiform nucleus to the temporal lobe and insula. D. Those of the parietal stalk pass from the lateral nucleus of the thalamus to the parietal lobe. Fibers also extend from the thalamus into the corpus striatum. Those destined for the caudate nucleus leave the lateral surface, and those from the lentiform nucleus, the inferior surface of the thalamus. The metathalamus comprises the geniculate bodies, which are two in number, a medial and a lateral on each side. The medial geniculate body, corpus geniculatum mediale, internal geniculate body, post geniculatum, lies under cover of the pulvinar of the thalamus and on the lateral aspect of the corpora quadrigeminum. Oval in shape, with its long axis directed forward and lateralward, it is lighter in color and smaller in size than the lateral. The inferior brachium from the inferior colliculus disappears under cover of it, while from its lateral extremity, a strand of fibers passes to join the optic tract. Entering it are many acoustic fibers from the lateral lemniscus. The medial geniculate bodies are connected with one another by the commissure of Gudin, which passes through the posterior part of the optic chiasma. The lateral geniculate body, corpus geniculatum laterale, external geniculate body, pregeniculatum, is an oval elevation on the lateral part of the posterior end of the thalamus, and is connected with the superior colliculus by the superior brachium. It is of a dark color, and presents a laminated arrangement consisting of alternating layers of gray and white substance. It receives numerous fibers from the optic tract, while other fibers of this tract pass over or through it into the pulvinar. Its cells are large and pigmented. Their axons pass to the visual area in the occipital part of the cerebral cortex. The superior colliculus, the pulvinar, and the lateral geniculate body receive many fibers from the optic tracts, and are therefore intimately connected with sight, constituting what are termed the lower visual centers. Extirpation of the eyes in the newly born animal entails an arrest to the development of these centers, but has no effect on the medial geniculate bodies or on the inferior colliculi. Moreover, the latter are well developed in the mole, an animal in which the superior colliculi are rudimentary. The epithalamus comprises the trigonum habenulae, the pineal body, and the posterior commissure. The trigonum habenulae is a small depressed triangular area situated in front of the superior colliculus and on the lateral aspect of the posterior part of the tinea thalami. It contains a group of nerve cells termed the ganglion habenulae. Fibers enter it from the stalk of the pineal body, and others forming what is termed the habenula commissure pass across the middle line to the corresponding ganglion of the opposite side. Most of its fibers are, however, directed downward and form a bundle, the fasciculus retroflexus of Meinert, which passes medial to the red nucleus and, after decussating with the corresponding fasciculus of the opposite side, ends in the interpeduncular ganglion. The pineal body, corpus pineale epiphysis, is a small, conical, reddish-gray body which lies in the depression between the superior colliculi. It is placed beneath the splenia of the corpus callosum, but is separated from this by the telechoroidea of the third ventricle, the lower layer of which envelops it. It measures about 8 millimeters in length, and its base, directed forward, is attached by a stalk or peduncle of white substance. The stalk of the pineal body divides anteriorly into two laminae, a dorsal and a ventral, separated from one another by the pineal recess of the third ventricle. The ventral lamina is continuous with the posterior commissure. The dorsal lamina is continuous with the habenular commissure and divides into two strands, the medullary striae, which run forward, one on either side, along the junction of the medial and upper surface of the thalamus to blend in front with the columns of the fornix. The posterior commissure is a rounded band of white fibers crossing the middle line on the dorsal aspect of the upper end of the cerebral aqueduct. Its fibers acquire their medullary sheaths early, but their connections have not been definitively determined. Most of them have their origin in a nucleus, the nucleus of the posterior commissure, nucleus of Darkschewitz, which lies in the central gray substance of the upper end of the cerebral aqueduct, in front of the nucleus of the oculomotor nerve. Some are probably derived from the posterior part of the thalamus and from the superior colliculus, while others are believed to be continued downward into the medial longitudinal fasciculus. The hypothalamus includes the subthalamic tegmental region and the structures forming the greater part of the floor of the third ventricle, that is, the corpora mammillaria, tuber cinereum, infundibulum hypophysis, and optic chiasma. The subthalamic tegmental region consists of the upward continuation of the tegmentum. It lies on the ventrolateral aspect of the thalamus and separates it from the fibers of the internal capsule. The red nucleus and the substantia nigra are prolonged into its lower part. In front, it is continuous with the substantia nominata of Meinert, medially with the gray substance of the floor of the third ventricle. It consists from above downward of three strata. 1. Stratum dorsale, directly applied to the undersurface of the thalamus and consisting of fine longitudinal fibers. 2. Zona incerta, a continuation forward of the formatio reticularis of the tegmentum. And 3. The corpus subthalamicum, nucleus of Lewis, a brownish mass presenting a lenticular shape on transverse section and situated on the dorsal aspect of the fibers of the base of the cerebral peduncle. It is encapsulated by a lamina of nerve fibers and contains numerous medium-sized nerve cells, the connections of which are not as yet fully determined. The corpora mammillaria, corpus albicontia, are two round white masses, each about the size of a small pea, placed side by side below the gray substance of the floor of the third ventricle, in front of the posterior perforated substance. They consist of white substance externally and gray substance internally, the cells of the latter forming two nuclei, a medial of smaller and a lateral of larger cells. The white substance is mainly formed by the fibers of the columns of the fornix, which descend to the base of the brain and end partly in the corpora mammillaria. From the cells of the gray substance of each of the mammillary bodies, two fasciculi arise. One, the thalamo-mammillary fasciculus, bundle of Vic d'Azur, passes upward into the anterior nucleus of the thalamus. The other is directed downward into the tegmentum. Afferent fibers are believed to reach the corpus mammillaria from the medial lemniscus and from the tegmentum. The tuber cinereum is a hollow eminence of gray substance situated between the corpora mammillaria behind and the optic chiasma in front. Laterally, it is continued with the anterior perforated substance, and anteriorly with a thin lamina, the lamina terminalis. From the undersurface of the tuber cinereum, a hollow conical process, the infundibulum, projects downward and forward and is attached to the posterior lobe of the hypophysis. In the lateral part of the tuber cinereum is a nucleus of nerve cells, the basal optic nucleus of Meinert, while close to the cavity of the third ventricle are three additional nuclei. Between the tuber cinereum and the corpora mammillaria, a small elevation with a corresponding depression in the third ventricle is sometimes seen. Retzius has named it the eminentia sacularis and regards it as a representative of the saccus vasculosus found in this situation in some of the lower vertebrates. The hypophysis, pituitary body, is a reddish-gray, somewhat oval mass measuring about 12.5 mm in its transverse and about 8 mm in its anterior-posterior diameter. It is attached to the end of the infundibulum and is situated in the fossa hypophyseos of the sphenoidal bone, where it is retained by a circular fold of Dura Mater, the diaphragma sella. This fold almost completely roofs in the fossa, leaving only a small central aperture through which the infundibulum passes. Optic chiasma. Chiasma opticum, optic commissure. The optic chiasma is a flattened and somewhat quadrilateral band of fibers situated at the junction of the floor and anterior wall of the third ventricle. Most of its fibers have their origins in the retina and reach the chiasma through the optic nerves, which are continuous with its anterior lateral angles. In the chiasma, they undergo a partial decussation. The fibers from the nasal half of the retina decussate and enter the optic tract of the opposite side, while the fibers from the temporal half of the retina do not undergo decussation, but pass back into the optic tract of the same side. Occupying the posterior part of the commissure, however, is a strand of fibers, the commissure of Guden, which is not derived from the optic nerves. It forms a connecting link between the medial geniculate bodies. Optic tracts. The optic tracts are continued backward and lateralward from the posterior lateral angles of the optic chiasma. Each passes between the anterior perforated substance and the tuber cinereum, and, winding around the ventrolateral aspect of the cerebral peduncle, divides into a medial and a lateral root. The former comprises the fibers of Guden's commissure. The lateral root consists mainly of afferent fibers which arise in the retina and undergo partial decussation in the optic chiasma as described. But it also contains a few fine efferent fibers which have their origin in the brain and their terminations in the retina. When traced backward, the afferent fibers of the lateral root are found to end in the lateral geniculate body and pulvinar of the thalamus and in the superior colliculus, and these three structures constitute the lower visual centers. Fibers arise from the nerve cells in these centers and pass through the occipital part of the internal capsule under the name of the optic radiations to the cortex of the occipital lobe of the cerebrum, where the higher or cortical visual center is situated. Some of the fibers of the optic radiations take an opposite course, arising from the cells of the occipital cortex and passing to the lower visual centers. Some fibers are detached from the optic tract and pass through the cerebral peduncle to the nucleus of the ocular motor nerve. These may be regarded as the afferent branches for the sphincter pupillae and ciliaris muscles. Other fibers have been described as reaching the cerebrum through the superior peduncle, while others again are lost in the pons. The third ventricle, ventriculus tertius. The third ventricle is a median cleft between the two thalami. Behind it, it communicates with the fourth ventricle through the cerebral aqueduct, and in front with the lateral ventricles through the interventricular foramen. Somewhat triangular in shape, with the apex directed backwards, it has a roof, a floor, an anterior and a posterior boundary, and a pair of lateral walls. The roof is formed by a layer of epithelium which stretches between the upper edges of the lateral wall of the cavity and is continuous with the epithelial lining of the ventricle. It is covered by and adherent to a fold of pia mater, named the telochoroidea of the third ventricle, from the undersurface of which a pair of vascular fringe processes, the choroid plexuses of the third ventricle, project downward, one on either side of the middle line, and invaginate the epithelial roof into the ventricular cavity. The floor slopes downward and forward and is formed mainly by the structures which constitute the hypothalamus. From before backwards these are the optic chiasma, the tuber cinereum and infundibulum, and the corpus mammillaria. Behind the last, the floor is formed by the interpeduncular fossa and the tegmenta of the cerebral peduncles. The ventricle is prolonged downward as the funnel-shaped recess, the recessus infundibuli, into the infundibulum, and to the apex of the latter, the hypophysis, is attached. The anterior boundary is constituted below by the lamina terminalis, a thin layer of gray substance stretching from the upper surface of the optic chiasma to the rostrum of the corpus callosum, above by the columns of the fornix and the anterior commissure. At the junction of the floor and anterior wall, immediately above the optic chiasma, the ventricle presents a small angular recess or diverticulum, the optic recess. Between the columns of the fornix and just above the anterior commissure is a second recess termed the vulva. At the junction of the roof and anterior wall of the ventricle, and situated between the thalami behind and the columns of the fornix in front, is the interventricular foramen, foramen of Monro, through which the third communicates with the lateral ventricles. The posterior boundary is constituted by the pineal body, the posterior commissure, and the cerebral aqueduct. A small recess, the recessus pinealis, projects into the stalk of the pineal body, while in front of and above the pineal body is a second recess, the recess suprapinealis, consisting of a diverticulum of the epithelium which forms the ventricular roof. Each lateral wall consists of an upper portion formed by the medial surface of the anterior two-thirds of the thalamus, and a lower consisting of an upward continuation of gray substance of the ventricular floor. These two parts correspond to the alar and basal laminae, respectively, of the lateral wall of the forebrain, vesicle, and are separated from each other by a furrow, the sulcus of Monro, which extends from the interventricular foramen to the cerebral aqueduct. The lateral wall is limited above by the taenia thalami. The columns of the fornix curve downward in front of the interventricular foramen and then run in the lateral walls of the ventricle, where, at first, they form distinct prominences, but subsequently are lost to sight. The lateral walls are joined to each other across the cavity of the ventricle by a band of gray substance, the massa intermedia. Interpeduncular fossa. This is a somewhat lozenge-shaped area of the base of the brain, limited in front by the optic chiasma, behind by the anterior superior surface of the pons, anterolaterally by the converging optic tracts, and posterior laterally by the diverging cerebral peduncles. The structures contained in it have already been described. From behind forward, they are the posterior perforated substance, corpora mammillaria, tuber cinereum, infundibulum, and hypophysis. End of section 13. Section 14 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. M.L. Cohen, Anatomy of the Human Body, Part 4 by Henry Gray. The Forebrain, Part 2. The telencephalon. The telencephalon includes 1. The cerebral hemispheres with their cavities, the lateral ventricles, and 2. The pars optica hypothalami, and the anterior portion of the third ventricle, already described under the diencephalon. As previously stated, each cerebral hemisphere may be divided into 3 fundamental parts, that is, the rhinencephalon, the corpus striatum, and the neopallium. The rhinencephalon, associated with the sense of smell, is the oldest part of the telencephalon and forms almost the whole of the hemisphere in some of the lower animals, that is, fishes, amphibians, and reptiles. In man, it is rudimentary, whereas the neopallium undergoes great development and forms the chief part of the hemisphere. The cerebral hemispheres. The cerebral hemispheres constitute the largest part of the brain and, when viewed together from above, assume the form of an ovoid mass broader behind than in front, the greatest transverse diameter corresponding with the line connecting the 2 parietal eminences. The hemispheres are separated medially by a deep cleft termed a longitudinal cerebral fissure, and each possesses a central cavity, the lateral ventricle. The longitudinal cerebral fissure, Fissura cerebri longitudinalis, great longitudinal fissure, contains a sickle-shaped process of Dura Mater, the falx cerebri. In front and behind, the fissure extends from the upper to the under surfaces of the hemispheres and completely separates them, but its middle portion separates them for only about 1/2 of their vertical extent, for at this part they are connected across the middle line by a great central white commissure, the corpus callosum. In a median sagittal section, the cut corpus callosum presents the appearance of a broad arched band. Its thick posterior end, termed a splenium, overlaps the midbrain but is separated from it by the telechoroidea of the third ventricle and the pineal body. Its anterior curved end, termed the genu, gradually tapers into a thinner portion, the rostrum, which is continued downward and backward in front of the anterior commissure to join the lamina terminalis. Arching backward from immediately behind the anterior commissure to the undersurface of the splenium is a second white band named the fornix. Between this and the corpus callosum are the laminae and cavity of the septum pellucidum. Surfaces of the cerebral hemispheres. Each hemisphere presents 3 surfaces, lateral, medial, and inferior. The lateral surface is convex in adaptation to the concavity of the corresponding half of the vault of the cranium. The medial surface is flat and vertical, and is separated from that of the opposite hemisphere by the great longitudinal fissure and the falx cerebri. The inferior surface is of an irregular form, and may be divided into 3 areas, anterior, middle, and posterior. The anterior area, formed by the orbital surface of the frontal lobe, is concave and rests on the roof of the orbit and nose. The middle area is convex and consists of the undersurface of the temporal lobe. It is adapted to the corresponding half of the middle cranial fossa. The posterior area is concave, directed medialward as well as downward, and is termed the tentorial surface, since it rests upon the tentorium cerebelli, which intervenes between it and the upper surface of the cerebellum. These 3 surfaces are separated from each other by the following borders: A. Superior medial between the lateral and medial surfaces. B. Inferior lateral between the lateral and inferior surfaces. The anterior part of this border separating the lateral from the orbital surface is known as the superciliary border. C. Medial occipital, separating the medial and tentorial surfaces, and D. Medial orbital, separating the orbital from the medial surface. The anterior end of the hemisphere is named the frontal pole, the posterior the occipital pole, and the anterior end of the temporal lobe the temporal pole. About 5 centimeters in front of the occipital pole on the inferior lateral border is an indentation or notch named the preoccipital notch. The surfaces of the hemispheres are molded into a number of irregular eminences named gyri or convolutions, and separated by furrows termed fissures and sulci. The furrows are of two kinds, complete and incomplete. The former appear earlier in fetal life, are few in number, and are produced by infoldings of the entire thickness of the brain wall and give rise to the corresponding elevations in the interior of the ventricle. They comprise the hippocampal fissure and parts of the calcarine and collateral fissures. The incomplete furrows are very numerous and only indent the subjacent white substance without producing any corresponding elevations in the ventricular cavity. The gyri and their intervening fissures and the sulci are fairly constant in their arrangement. At the same time, they vary within certain limits, not only in different individuals, but on the two hemispheres of the same brain. The convoluted condition of the surface permits a great expansion of the gray matter without the sacrifice of much additional space. The number and extent of the gyri, as well as the depth of the intervening furrows, appear to bear a direct relation to the intellectual powers of the individual. Certain of the fissures and sulci are utilized for the purpose of dividing the hemisphere into lobes, and are therefore termed interlobular. Included under this category are the lateral cerebral, parietal occipital, calcarine, and collateral fissures, the central and cingulate sulci, and the sulcus circularis. The lateral cerebral fissure, fissura cerebri lateralis Sylvii, fissure of Sylvius, is a well-marked cleft on the inferior and lateral surfaces of the hemisphere and consists of a short stem which divides into 3 rami. The stem is situated at the base of the brain and commences in a depression at the lateral angle of the anterior perforated substance. From this point, it extends between the anterior part of the temporal lobe and the orbital surface of the frontal lobe and reaches the lateral surface of the hemisphere. Here it divides into 3 rami, an anterior horizontal, an anterior ascending, and a posterior. The anterior horizontal ramus passes forward about 2.5 centimeters into the inferior frontal gyrus, while the anterior ascending ramus extends upward into the same convolution for about an equal distance. The posterior ramus is the longest. It runs backward and slightly upward for about 7 centimeters and ends by an upward inflection in the parietal lobe. The central sulcus, sulcus centralis Rolandi, fissure of Rolando, central fissure, is situated about the middle of the lateral surface of the hemisphere and begins in or near the longitudinal cerebral fissure, a little behind its midpoint. It runs sinuously downward and forward and ends a little above the posterior ramus of the lateral fissure, and about 2.5 centimeters behind the anterior ascending ramus of the same fissure. It described 2 chief curves, a superior genu with its concavity directed forward, and an inferior genu with its concavity directed backward. The central sulcus forms an angle opening forward of about 70 degrees with the median plane. The parieto-occipital fissure, fissura parietal occipitalis. Only a small part of this fissure is seen on the lateral surface of the hemisphere, its chief part being on the medial surface. The lateral part of the parietal occipital fissure is situated about 5 centimeters in front of the occipital pole of the hemisphere and measures about 1.25 centimeters in length. The medial part of the parietal occipital fissure runs downward and forward as a deep cleft on the medial surface of the hemisphere and joins the calcarine fissure below and behind the posterior end of the corpus callosum. In most cases, it contains a submerged gyrus. The calcarine fissure, fissura calcarina, is on the medial surface of the hemisphere. It begins near the occipital pole in 2 converging rami and runs forward to a point a little below the splenium of the corpus callosum, where it is joined at an acute angle by the medial part of the parietal occipital fissure. The anterior part of this fissure gives rise to the prominence of the calcar avus and the posterior cornule of the lateral ventricle. The cingulate sulcus, sulcus cinguli, callosomarginal fissure, is on the medial surface of the hemisphere. It begins below the anterior end of the corpus callosum and runs upward and forward nearly parallel to the rostrum of this body and, curving in front of the genu, is continued backward above the corpus callosum and finally ascends to the superior medial border of the hemisphere, a short distance behind the upper end of the central sulcus. It separates the superior frontal from the cingulate gyrus. The collateral fissure, fissura collateralis, is on the tentorial surface of the hemisphere and extends from near the occipital pole to within a short distance of the temporal pole. Behind, it lies below and lateral to the calcarine fissure, from which it is separated by the lingual gyrus. In front, it is situated between the hippocampal gyrus and the anterior part of the fusiform gyrus. The sulcus circularis, circuminsular fissure, is on the lower and lateral surfaces of the hemisphere. It surrounds the insula and separates it from the frontal, parietal, and temporal lobes. The lobes of the hemispheres. By means of these fissures and sulci, assisted by certain arbitrary lines, each hemisphere is divided into the following lobes: the frontal, the parietal, the temporal, the occipital, the limbic, and the insula. Frontal lobe, lobus frontalis. On the lateral surface of the hemisphere, this lobe extends from the frontal pole to the central sulcus, the latter separating it from the parietal lobe. Below, it is limited by the posterior ramus of the lateral fissure, which intervenes between it and the central lobe. On the medial surface, it is separated from the cingulate gyrus by the cingulate sulcus, and on the inferior surface, it is bounded behind by the stem of the lateral fissure. The lateral surface of the frontal lobe is traversed by three sulci which divide it into four gyri. The sulci are named the precentral and the superior and inferior frontal. The gyri are the anterior central and the superior, middle, and inferior frontal. The precentral sulcus runs parallel to the central sulcus and is usually divided into an upper and a lower part. Between it and the central sulcus is the anterior central gyrus. From the precentral sulcus, the superior and inferior frontal sulci run forward and downward and divide the remainder of the lateral surface of the lobe into three parallel gyri, named respectively the superior, middle, and inferior frontal gyri. The anterior central gyrus, gyrus centralis anterior, ascending frontal convolution, precentral gyri, is bounded in front by the precentral sulcus, behind by the central sulcus. It extends from the superior medial border of the hemisphere to the posterior ramus of the lateral fissure. The superior frontal gyrus, gyrus frontalis superior, superior frontal gyrus, is situated above the superior frontal sulcus and is continued on to the medial surface of the hemisphere. The portion on the lateral surface of the hemisphere is usually more or less completely subdivided into an upper and a lower part by the anterior-posterior sulcus, the paramedian sulcus, which, however, is frequently interrupted by bridging gyri. The middle frontal gyrus, gyrus frontalis medius, medial frontal gyre, between the superior and inferior frontal sulci, is continuous with the anterior orbital gyrus on the inferior surface of the hemisphere. It is frequently subdivided into two by a horizontal sulcus, the medial frontal sulcus of Eberstaller, which ends anteriorly in a wide bifurcation. The inferior frontal gyrus, gyrus frontalis inferior, subfrontal gyre, lies below the inferior frontal sulcus and extends forward from the lower part of the precentral sulcus. It is continuous with the lateral and posterior orbital gyri on the undersurface of the lobe. It is subdivided by the anterior horizontal and ascending rami of the lateral fissure into three parts. 1. The orbital part, below the anterior horizontal ramus of the fissure. 2. The triangular part, cap of Broca, between the ascending and horizontal rami. And 3. The basilar part, behind the anterior ascending ramus. The left inferior frontal gyrus is, as a rule, more highly developed than the right, and is named the gyrus of Broca, from the fact that Broca described it as the center for articulate speech. The inferior or orbital surface of the frontal lobe is concave and rests on the orbital plate of the frontal bone. It is divided into four orbital gyri by a well-marked H-shaped orbital sulcus. These are named from their position the medial, anterior, lateral, and posterior orbital gyri. The medial orbital gyrus presents a well-marked anterior-posterior sulcus, the olfactory sulcus, for the olfactory tract. The portion medial to this is termed a straight gyrus and is continuous with the superior frontal gyrus on the medial surface. The medial surface of the frontal lobe is occupied by the medial part of the superior frontal gyrus, marginal gyrus. It lies between the cingulate sulcus and the superior medial margin of the hemisphere. The posterior part of this gyrus is sometimes marked off by a vertical sulcus and is distinguished as the paracentral lobule because it is continuous with the anterior and posterior central gyri. Parietal lobe, lobus parietalis. The parietal lobe is separated from the frontal lobe by the central sulcus, but its boundaries below and behind are not so definite. Posteriorly, it is limited by the parietal occipital fissure and by a line carried across the hemisphere from the end of the fissure towards the preoccipital notch. Below, it is separated from the temporal lobe by the posterior ramus of the lateral fissure and by a line carried backward from it to meet the line passing downward to the preoccipital notch. The lateral surface of the parietal lobe is cleft by a well-marked furrow, the intraparietal sulcus of Turner, which consists of an oblique and a horizontal portion. The oblique part is named the postcentral sulcus and commences below about midway between the lower end of the central sulcus and the upturned end of the lateral fissure. It runs upward and backward parallel to the central sulcus and is sometimes divided into an upper and a lower ramus. It forms the hinder limit of the posterior central gyrus. From about the middle of the postcentral sulcus, or from the upper end of its inferior ramus, the horizontal portion of the intraparietal sulcus is carried backward and slightly upward on the parietal lobe and is prolonged under the name of the occipital ramus onto the occipital lobe, where it divides into two parts, which form nearly a right angle with the main stem and constitute the transverse occipital sulcus. The part of the parietal lobe above the horizontal portion of the intraparietal sulcus is named the superior parietal lobule, the part below, the inferior parietal lobule. The posterior central gyrus, gyrus centralis posterior, ascending parietal convolution, postcentral gyre, extends from the longitudinal fissure above to the posterior ramus of the lateral fissure below. It lies parallel with the anterior central gyrus with which it is connected below and also sometimes above the central sulcus. The superior parietal lobule, lobulus parietalis superior, is bounded in front by the upper part of the postcentral sulcus, but is usually connected with the posterior central gyrus above the end of the sulcus. Behind it is the lateral part of the parietal occipital fissure, around the end of which it is joined to the occipital lobe by a curved gyrus, the arcus parietal occipitalis. Below, it is separated from the inferior parietal lobule by the horizontal portion of the intraparietal sulcus. The inferior parietal lobule, lobulus parietalis inferior, subparietal district or lobule, lies below the horizontal portion of the intraparietal sulcus and behind the lower part of the postcentral sulcus. It is divided from before backward into two gyri. 1. The supramarginal arches over the upturned end of the lateral fissure. It is continuous in front with the postcentral gyrus and behind with the superior temporal gyrus. The second, the angular, arches over the posterior end of the superior temporal sulcus, behind which it is continuous with the middle temporal gyrus. The medial surface of the parietal lobe is bounded behind the medial part of the parietal occipital fissure. In front, by the posterior end of the cingulate sulcus, and below, it is separated from the cingulate gyrus by the subparietal sulcus. It is of small size and consists of a square-shaped convolution, which is termed the precuneus or quadrate lobe. Lobus occipitalis. The occipital lobe is small and pyramidal in shape. It presents three surfaces: lateral, medial, and tentorial. The lateral surface is limited in front by the lateral part of the parietal occipital fissure and by a line carried from the end of this fissure to the preoccipital notch. It is traversed by the transverse occipital and lateral occipital sulci. The transverse occipital sulcus is continuous with the posterior end of the occipital ramus of the intraparietal sulcus and runs across the upper part of the lobe a short distance behind the parietal occipital fissure. The lateral occipital sulcus extends from behind forward and divides the lateral surface of the occipital lobe into a superior and inferior gyrus, which are continuous in front with the parietal and temporal lobes. The medial surface of the occipital lobe is bounded in front by the medial part of the parietal occipital fissure and is traversed by the calcarine fissure, which subdivides it into the cuneus and the lingual gyrus. The cuneus is a wedge-shaped area between the calcarine fissure and the medial part of the parietal occipital fissure. The lingual gyrus lies between the calcarine fissure and the posterior part of the collateral fissure. Behind, it reaches the occipital pole. In front, it is continued on to the tentorial surface of the temporal lobe and joins the hippocampal gyrus. The tentorial surface of the occipital lobe is limited in front by an imaginary transverse line through the preoccipital notch and consists of the posterior part of the fusiform gyrus, occipitotemporal convolution, and the lower part of the lingual gyrus, which are separated from each other by the posterior segment of the collateral fissure. End of section 14. Section 15 of Gray's Anatomy Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 3. Temporal lobe, lobus temporalis. The temporal lobe presents superior, lateral, and inferior surfaces. The superior surface forms the lower limit of the lateral fissure and overlaps the insula. On opening out the lateral fissure, 3 or 4 gyri will be seen springing from the depths of the hinder end of the fissure and running obliquely forward and outward on the posterior part of the upper surface of the superior temporal gyrus. These are named the transverse temporal gyri. Heschel. The lateral surface is bounded above by the posterior ramus of the lateral fissure and by the imaginary line continued backward from it. Below, it is limited by the inferior lateral border of the hemisphere. It is divided into superior, middle, and inferior gyri by the superior and middle temporal sulci. The superior temporal sulcus runs from before backwards across the temporal lobe and some little distance below, but parallel with the posterior ramus of the lateral fissure, and hence it is often termed the parallel sulcus. The middle temporal sulcus takes the same direction as the superior, but is situated at a lower level and is usually subdivided into 2 or more parts. The superior temporal gyrus lies between the posterior ramus of the lateral fissure and the superior temporal sulcus and is continuous behind with the supramarginal and angular gyri. The middle temporal gyrus is placed between the superior and middle temporal sulci and is joined posteriorly with the angular gyrus. The inferior temporal gyrus is placed below the middle temporal sulcus and is connected behind with the inferior occipital gyrus. It also extends around the inferior lateral border onto the inferior surface of the temporal lobe, where it is limited by the inferior sulcus. The inferior surface is concave and is continuous posteriorly with the tentorial surface of the occipital lobe. It is traversed by the inferior temporal sulcus, which extends from near the occipital pole behind to within a short distance of the temporal pole in front, but is frequently subdivided by bridging gyri. Lateral to this fissure is the narrow tentorial part of the inferior temporal gyrus, and medial to it the fusiform gyrus, which extends from the occipital to the temporal pole. This gyrus is limited medially by the collateral fissure, which separates it from the lingual gyrus behind and from the hippocampal gyrus in front. The insula, island of Ryle's central lobe, lies deeply in the lateral or sibilant fissure, and can only be seen when the lips of that fissure are widely separated, since it is overlapped and hidden by the gyri which bound the fissure. These gyri are termed the opercula of the insula. They are separated from each other by the 3 rami of the lateral fissure, and are named the orbital frontal, frontotemporal, and temporal opercula. The orbital operculum lies below the anterior horizontal ramus of the fissure, the frontal between this and the anterior ascending ramus, the parietal between the anterior ascending ramus and the upturned end of the posterior ramus, and the temporal below the posterior ramus. The frontal operculum is of small size in those cases where the anterior horizontal and ascending rami of the lateral fissures arise from a common stem. The insula is surrounded by a deep circular sulcus, which separates it from the frontal, parietal, and temporal lobes. When the opercula have been removed, the insula is seen as a triangular eminence, the apex of which is directed toward the anterior perforated substance. It is divided into a larger anterior and a smaller posterior part by a deep sulcus, which runs backward and upward from the apex of the insula. The anterior part is subdivided by a shallow sulci into 3 or 4 short gyri, while the posterior part is formed by 1 long gyrus, which is often bifurcated at its upper end. The cortical gray substance of the insula is continuous with that of the different opercula, while its deep surface corresponds with the lentiform nucleus of the corpus striatum. Limbic lobe. The term limbic lobe was introduced by Broca and under it he included the cingulate and hippocampal gyri, which together arch around the corpus callosum in the hippocampal fissure. These he separated on the morphological ground that they are well developed in animals possessing a keen sense of smell, osmotic animals, such as the dog and the fox. They were thus regarded as part of the rhinencephalon, but it is now recognized that they belong to the neopallium. The cingulate gyrus is therefore sometimes described as part of the frontal lobe, and the hippocampal as part of the temporal lobe. The cingulate gyrus, gyrus cinguli, callosal convolution, is an arch-shaped convolution lying in close relation to the superior surface of the corpus callosum, from which it is separated by a slit-like fissure, the callosal fissure. It commences below the rostrum of the corpus callosum, curves around in front of the genu, extends along the upper surface of the body, and finally turns downward behind the splenium, where it is connected by a narrow isthmus with the hippocampal gyrus. It is separated from the medial part of the superior frontal gyrus by the cingulate sulcus, and from the precuneus by the subparietal sulcus. The hippocampal gyrus, gyrus hippocampi, is bounded above by the hippocampal fissure, and below by the anterior part of the collateral fissure. Behind, it is continuous superiorly through the isthmus with the cingulate gyri and inferiorly with the lingual gyrus. Running in the substance of the cingulate and hippocampal gyri and connecting them together is a tract of arched fibers named the cingulum. The anterior extremity of the hippocampal gyrus is recurved in the form of a hook, uncus, which is separated from the apex of the temporal lobe by a slight fissure, the incisura temporalis. Although superficially continuous with the hippocampal gyrus, the uncus forms morphologically a part of the rhinencephalon. The hippocampal fissure, fissura hippocampi, dentate fissure, begins immediately behind the splenium of the corpus callosum and runs forward between the hippocampal and dentate gyri to end in the uncus. It is a complete fissure and gives rise to the prominence of the hippocampus in the inferior cornu of the lateral ventricle. Rhinencephalon. The rhinencephalon comprises the olfactory lobe, the uncus, the subcallosal and supracallosal gyri, the fascia dentata hippocampus, the septum pellucidum, the fornix, and the hippocampus. 1. The olfactory lobe, lobus olfactorius, is situated under the inferior or orbital surface of the frontal lobe. In many vertebrates, it constitutes a well-marked portion of the hemisphere and contains an extension of the lateral ventricle. But in man, and some other mammals, it is rudimentary. It consists of the olfactory bulb and tract, the olfactory trigone, the para-olfactory area of Broca, and the anterior perforated substance. A. The olfactory bulb, bulbus olfactorius, is an oval, reddish-gray mass which rests on the cribriform plate of the ethmoid and forms the anterior expanded extremity of the olfactory tract. Its undersurface receives the olfactory nerves which pass upward through the cribriform plate from the olfactory region of the nasal cavity. Its minute structure is described later. B. The olfactory tract, Tractus olfactorius, is a narrow white band triangular on coronal section, the apex being directed upward. It lies in the olfactory sulcus on the inferior surface of the frontal lobe and divides posteriorly into two striae, a medial and a lateral. The lateral stria is directed across the lateral part of the anterior perforated substance and then bends abruptly medialward towards the uncus of the hippocampal gyrus. The medial stria turns medialward behind the pallor olfactory area and ends in the subcallosal gyrus. In some cases, a small intermediate stria is seen running backward to the anterior perforated substance. C. The olfactory trigone, Trigonum olfactorium, is a small triangular area in front of the anterior perforated substance. Its apex, directed forward, occupies the posterior part of the olfactory sulcus and is brought into view by throwing back the olfactory tract. D. The para-olfactory area of Broca, Area paraolfactoria, is a small triangular field on the medial surface of the hemisphere in front of the subcallosal gyrus, from which it is separated by the posterior para-olfactory sulcus. It is continuous below with the olfactory trigone and above and in front with the cingulate gyrus. It is limited anteriorly by the anterior para-olfactory sulcus. E. The anterior perforated substance, Substantia perforata anterior, is an irregularly quadrilateral area in front of the optic tract and behind the olfactory trigone, from which it is separated by the fissura primea. Medially and in front, it is continuous with the subcallosal gyrus. Laterally, it is bounded by the lateral stria of the olfactory tract and is continued into the uncus. Its gray substance is confluent above with that of the corpus striatum and is perforated anteriorly by numerous small blood vessels. 2. The uncus has already been described as the recurved, hook-like portion of the hippocampal gyrus. 3. The subcallosal, supracallosal, and dentate gyri form a rudimentary arch-shaped lamina of gray substance extending over the corpus callosum and above the hippocampal gyrus from the anterior perforated substance to the uncus. A. The subcallosal gyrus, gyrus subcallosus, pedicome of the corpus callosum, is a narrow lamina on the medial surface of the hemisphere in front of the lamina terminalis, behind the paraolfactory area, and below the rostrum of the corpus callosum. It is continuous around the genu of the corpus callosum with the supracallosal gyrus. B. The supracallosal gyrus, indusium griseum, gyrus epicallosus, consists of a thin layer of gray substance in contact with the upper surface of the corpus callosum and continuously laterally with the gray substance of the cingulate gyrus. It contains two longitudinally directed strands of fibers termed respectively the medial and lateral longitudinal striae. The supracallosal gyrus is prolonged around the splenium of the corpus callosum as a delicate lamina, the fasciola cinerea, which is continuous below with the fascia dentata hippocampi. C. The fascia dentata hippocampi, gyrus dentatus, is a narrow band extending downward and forward above the hippocampal gyrus, but separated from it by the hippocampal fissure. Its free margin is notched and overlapped by the fimbria, the fimbrio-dentate fissure, intervening. Anteriorly, it is continued into the notch of the uncus, where it forms a sharp bend and is then prolonged as a delicate band, the band of Giacomini, over the uncus on the lateral surface of which it is lost. The remaining parts of the rhinencephalon, that is the septum pellucidum, fornix, and hippocampus, will be described in connection with the lateral ventricle. Interior of the cerebral hemispheres. If the upper part of either hemisphere be removed at a level about 1.25 centimeters above the corpus callosum, the central white substance will be exposed as an oval-shaped area, the centrum ovale minus, surrounded by a narrow convoluted margin of gray substance and studded with numerous minute red dots, puncta vasculosa, produced by the escape of blood from divided blood vessels. If the remaining portion of the hemisphere will be slightly drawn apart, a broad band of white substance, the corpus callosum will be observed connecting them at the bottom of the longitudinal fissure. The margins of the hemispheres which overlap the corpus callosum are called the labia cerebri. Each labrium is part of the cingulate gyrus already described, and the slit-like interval between it and the upper surface of the corpus callosum is termed the callosal fissure. If the hemispheres be sliced off to a level with the upper surface of the corpus callosum, the white substance of that structure will be seen connecting the two hemispheres. The large expanse of medullary matter now exposed, surrounded by a convoluted margin of gray substance, is called the centrum ovale majus. The corpus callosum is the great transverse commissure which unites the cerebral hemispheres and roofs the lateral ventricles. A good conception of its position and size is obtained by examining a medial sagittal section of the brain. When it is seen to form an arch-straight structure about 10 centimeters long, its anterior end is about 4 centimeters from the frontal pole, and its posterior end about 6 centimeters from the occipital pole of the hemisphere. The anterior end is named a genu, and is bent downward and backward in front of the septum pellucidum. Diminishing rapidly in thickness, it is prolonged backwards under the name of the rostrum, which is connected below with the lamina terminalis. The anterior cerebral arteries are in contact with the undersurface of the rostrum. They then arch over the front of the genu and are carried backwards above the body of the corpus callosum. The posterior end is termed the splenium and constitutes the thickest part of the corpus callosum. It overlays the thylakoidea of the third ventricle and the midbrain and ends in a thick convex free border. A sagittal section of the splenium shows that the posterior end of the corpus callosum is actually bent forward, the upper and lower parts being applied to each other. The superior surface is convex from before backward and is about 2.5 centimeters wide. Its medial part forms the bottom of the longitudinal fissure and is in contact posteriorly with the lower border of the falx cerebri. Laterally, it is overlapped by the cingulate gyrus, but is separated from it by the slit-like callosal fissure. It is traversed by numerous transverse ridges and furrows and is covered by a thin layer of gray matter, the supracallosal gyrus, which exhibits on either side of the middle line the medial and lateral longitudinal striae already described. The inferior surface is concave and forms on either side of the middle line of the roof of the lateral ventricle. Medially, this surface is attached in front to the septum pellucidum. Behind this, it is fused with the upper surface of the body of the fornix, while the splenium is in contact with the telechoroidea. On either side, the fibers of the corpus callosum radiate in a white substance and pass to the various parts of the cerebral cortex. Those curving forward from the genu to the frontal lobe constitute the forceps anterior, and those curving backward into the occipital lobe, the forceps posterior. Between these two parts is the main body of the fibers which constitute the tapetum and extends laterally on either side into the temporal lobe and cover in the central part of the lateral ventricle. The lateral ventricles. Ventriculus lateralis. The two lateral ventricles are irregular cavities situated in the lower medial parts of the cerebral hemisphere, one on either side of the middle line. They are separated from each other by a median vertical partition, the septum pellucidum, but communicate with the third ventricle and indirectly with each other through the interventricular foramen. They are lined by a thin diaphanous membrane, the ependema, covered by ciliated epithelium, and contain cerebrospinal fluid, which, even in health, may be secreted in considerable amount. Each lateral ventricle consists of a central part or body and three prolongations from it termed cornua. The central part, pars centralis ventricula lateralis, cella, of the lateral ventricle extends from the interventricular foramen to the splenium of the corpus callosum. It is an irregularly curved cavity, triangular on transverse section, with a roof, a floor, and a medial wall. The roof is formed by the undersurface of the corpus callosum, the floor by the following parts enumerated in order of their position from before backward: the caudate nucleus of the corpus striatum, the stria terminalis and the terminal vein, the lateral portion of the upper surface of the thalamus, the choroid plexus, and the lateral part of the fornix. The medial wall is the posterior part of the septum pellucidum, which separates it from the opposite ventricle. The anterior cornu, cornu anterius, anterior horn, precornu, passes forward and lateralward with a slight inclination downward from the interventricular foramen into the frontal lobe, curving around the anterior end of the caudate nucleus. Its floor is formed by the upper surface of the reflected portion of the corpus callosum, the rostrum. It is bounded medially by the anterior portion of the septum pellucidum and laterally by the head of the caudate nucleus. Its apex reaches the posterior surface of the genu of the corpus callosum. The posterior cornu, cornu posterius post cornu, passes into the occipital lobe, its direction being backward and lateralward and then medialward. Its roof is formed by the fibers of the corpus callosum passing to the temporal and occipital lobes. On its medial wall is a longitudinal eminence, the calcar avris, hippocampus minor, which is an involution of the ventricular wall produced by the calcarine fissure. Above this, the forceps posterior of the corpus callosum sweeping around to enter the occipital lobe causes another projection, termed the bulb of the posterior cornu. The calcar avus and bulb of the posterior cornu are extremely variable in their degree of development. In some cases they are ill-defined, in others prominent. The inferior cornu, cornu inferior, descending horn, middle horn, medial cornu, is the largest of the three, transverses the temporal lobe of the brain, forming in its course a curve around and posterior end of the thalamus. It passes at first backward, laterward, and downward, and then curves forward to within 2.5 centimeters of the apex of the temporal lobe, its direction being fairly well indicated on the surface of the brain by that of the superior temporal sulcus. Its roof is formed chiefly by the inferior surface of the tapetum of the corpus callosum, but the tail of the caudate nucleus and the stria terminalis also extend forward in the roof of the inferior cornu to its extremity, where they end in a massive gray substance, the nucleus amygdalae. Its floor presents the following parts: the hippocampus, the fimbria hippocampi, the collateral eminence, and the choroid plexus. When the choroid plexus is removed, a cleft-like opening is left along the medial wall of the inferior cornu. This cleft constitutes the lower part of the choroidal fissure. End of section 15. Section 16 of Gray's Anatomy Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 4. The hippocampus major, is a curved eminence about 5 centimeters long, which extends throughout the entire length of the floor of the inferior cornu. Its lower end is enlarged and presents 2 or 3 rounded elevations or digitations, which give it a paw-like appearance, and hence it is named a pes hippocampi. If a transverse section be made through the hippocampus, it will be seen that this eminence is produced by the folding of the wall of the hemisphere to form the hippocampal fissure. The main mass of the hippocampus consists of gray substance, but on its ventricular surface is a thin white layer, the alveus, which is continuous with the fimbria hippocampi. The collateral eminence, eminentia collateralis, is an elongated swelling lying lateral and parallel with the hippocampus. It corresponds with the middle part of the collateral fissure, and its size depends on the depth and direction of this fissure. It is continuous behind with the flattened triangular area, the trigonum collaterali, situated between the posterior and inferior cornua. The fimbria hippocampi is a continuation of the crus of the fornix and will be discussed with that body. A description of the choroid plexus will be found later. The corpus striatum has received its name from the striped appearance which a section of its anterior part presents, in consequence of diverging white fibers being mixed with the gray substance which forms its chief mass. A part of the corpus striatum is embedded in the white substance of the hemisphere and is therefore external to the ventricle. It is termed the extraventricular portion, or the lentiform nucleus. The remainder, however, projects into the ventricle and is named the interventricular portion, or the caudate nucleus. The caudate nucleus, nucleus caudatus, caudatum, is a pear-shaped, highly arched gray mass. Its broad extremity or head is directed forward into the anterior cornu of the lateral ventricle, and is continuous with the anterior perforated substance and with the anterior end of the lentiform nucleus. Its narrow end, or tail, is directed backwards on the lateral side of the thalamus, from which it is separated by the stria terminalis and the terminal vein. It is then continued downward into the roof of the inferior cornu and ends in the nucleus amygdalae at the apex of the temporal lobe. It is covered by the lining of the ventricle and crossed by some veins of considerable size. It is separated from the lentiform nucleus in its greater part of its extent by a thick lamina of white substance called the internal capsule, but the 2 portions of the corpus striatum are united in front. The lentiform nucleus. Nucleus lentiformis, lenticular nucleus, lenticula, is lateral to the caudate nucleus and thalamus and is seen only in sections of the hemisphere. When divided horizontally, it exhibits to some extent the appearance of a biconvex lens, while a coronal section of its central part presents a somewhat triangular outline. It is shorter than the caudate nucleus and does not extend as far forward. It is bounded laterally by a lamina of white substance called the external capsule, and lateral to this is a thin layer of gray substance termed the claustrum. Its anterior end is continuous with the lower part of the head of the caudate nucleus and with the anterior perforated substance. In a coronal section through the middle of the lentiform nucleus, 2 medullary laminae are seen dividing it into 3 parts. The lateral and largest part is of a reddish color and is known as the putamen, while the medial and intermediate area of a yellowish tint and together constitute the globus pallidus. All 3 are marked by fine radiating white fibers, which are most distinct in the putamen. The gray substance of the corpus striatum is traversed by nerve fibers, some of which originate in it. The cells are multipolar, both large and small. Those of the lentiform nucleus contain yellow pigment. The caudate and lentiform nuclei are not only directly continuous with each other anteriorly, but are connected to each other by numerous fibers. The corpus striatum is also connected, 1, to the cerebral cortex by what are termed corticostriate fibers, 2, to the thalamus by fibers which pass through the internal capsule and by a strand named ansa lentiformis, 3, to the cerebral peduncle by fibers which leave the lower aspect of the caudate and lentiform nuclei. The claustrum is a thin layer of gray substance situated on the lateral surface of the external capsule. Its transverse section is triangular, with the apex directed upward. Its medial surface, contiguous to the external capsule, is smooth, but its lateral surface presents ridges and furrows corresponding with the gyri and sulci of the insula, with which it is in close relationship. The claustrum is regarded as a detached portion of the gray substance of the insula, from which it is separated by a layer of white fibers, the capsula extrema, band of Ballarger. Its cells are small and spindle-shaped and contain yellowish pigment. They are similar to those of the deepest layer of the cortex. The nucleus amygdalae, amygdala, is an ovoid gray mass situated at the lower end of the roof of the inferior cornu. It is merely a localized thickening of the gray cortex, continuous with that of the uncus. In front, it is continuous with the putamen, behind with the stria terminalis and tail of the caudate nucleus. The internal capsule, capsula interna, is a flattened band of white fibers between the lentiform nucleus on the lateral side and the caudate nucleus and thalamus on the medial side. In horizontal section, it is seen to be somewhat abruptly curved with its convexity inward. The prominence of the curve is called the genu and projects between the caudate nucleus and the thalamus. The portion in front of the genu is termed the frontal part and separates the lentiform from the caudate nucleus. The portion behind the genu is the occipital part and separates the lentiform nucleus from the thalamus. The front part of the internal capsule contains 1. Fibers running from the thalamus to the frontal lobe, 2. Fibers connected to lentiform and caudate nuclei, 3. Fibers connecting the cortex with the corpus striatum, and 4. Fibers passing from the frontal lobe through the medial fifth of the base of the cerebral peduncle to the nucleus pontus. The fibers in the region of the genu are named the geniculate fibers. They originate in the motor part of the cerebral cortex, and after passing downward through the base of the cerebral peduncle with the cerebrospinal fibers, undergo decussation and end in the motor nuclei of the cranial nerves of the opposite side. The anterior two-thirds of the occipital part of the internal capsule contain the cerebrospinal fibers, which arise in the motor area of the cerebral cortex, and, passing downward through the middle 3/5 of the base of the cerebral peduncle, are continued into the pyramids of the medulla oblongata. The posterior third of the occipital part contains 1. Sensory fibers largely derived from the thalamus, though some may be continued upward from the medial lemniscus. 2. The fibers of the optic radiation, from the lower visual centers to the cortex of the occipital lobe. 3. Acoustic fibers, from the lateral lemniscus to the temporal lobe. And 4. Fibers which pass from the occipital and temporal lobes to the nuclei pontus. The fibers of the internal capsule radiate widely as they pass to and from the various parts of the cerebral cortex, forming the corona radiata, and intermingling with the fibers of the corpus callosum. The external capsule, capsula externa, is a lamina of white substance situated lateral to the lentiform nucleus, between it and the claustrum, and continuous with the internal capsule below and behind the lentiform nucleus. It probably contained fibers derived from the thalamus, the anterior commissure, and the subthalamic region. The substantia innominata of Meinert is a stratum consisting partly of gray and partly of white substance, which lies below the anterior part of the thalamus and lentiform nucleus. It consists of 3 layers, superior, middle, and inferior. The superior layer is named the ansa lentiformis, and its fibers, derived from the medullary lamina of the lentiform nucleus, pass medially to end in the thalamus and subthalamic region, while others are said to end in the tegmentum and red nucleus. The middle layer consists of nerve cells and nerve fibers. Fibers enter it from the parietal lobe through the external capsule, while others are said to connect it with the medial longitudinal fasciculus. The inferior layer forms the main part of the inferior stalk of the thalamus and connects this body with the temporal lobe and insula. The stria terminalis, Taenia semicircularis, is a narrow band of white substance situated in the depression between the caudate nucleus and the thalamus. Anteriorly, its fibers are partly continued into the column of the fornix. Some, however, pass over the anterior commissure to the gray substance between the caudate nucleus and the septum pellucidum, while others are said to enter the caudate nucleus. Posteriorly, it is continued into the roof of the inferior cornua of the lateral ventricle, at the extremity of which it enters the nucleus amygdalae. Superficial to it is a large vein, the terminal vein, vein of the corpus striatum, which receives numerous tributaries from the corpus striatum and thalamus. It runs forward to the interventricular foramen and there joins with the vein of the choroid plexus to form the corresponding internal cerebral vein. On the surface of the terminal vein is a narrow white band named the lamina affixa. The fornix is a longitudinal arch-shaped lamella of white substance situated below the corpus callosum and continuous with it behind, but separated from it in front by the septum pellucidum. It may be described as consisting of 2 symmetrical bands, one for either hemisphere. The 2 portions are not united to each other in front and behind, but their central parts are joined together in the middle line. The anterior parts are called the columns of the fornix, the intermediate united portions the body, and the posterior parts the crura. The body, corpus fornicus, of the fornix is triangular, narrow in front and broad behind. The medial part of its upper surface is connected to the septum pellucidum in front, and to the corpus callosum behind. The lateral portion of this surface forms part of the floor of the lateral ventricle and is covered by the ventricular epithelium. Its lateral edge overlaps the choroid plexus and is continuous with the epithelium covering of the structure. The undersurface rests upon the telochoroidea of the third ventricle, which separates it from the epithelial roof of that cavity and from the medial portions of the upper surfaces of the thalami. Below, the lateral portions of the body of the fornix are joined by a thin triangular lamina named a solitarium, lyra. This lamina contains some transverse fibers which connect the two hippocampi across the middle line and constitutes a hippocampal commissure. Between the solitarium and the corpus callosum is a horizontal cleft, the so-called ventricle of the fornix. Ventricle of Verga is sometimes found. The columns, columna fornicus, anterior pillars, fornicolumns, of the fornix arch downward in front of the interventricular foramen and behind the anterior commissure, and each descends through the gray substance in the lateral wall of the third ventricle to the base of the brain, where it ends in the corpus mammillare. From the cells of the corpus mammillare, the thalamo-mammillary fasciculus, bundle of Vic d'Azur, takes origin and is prolonged into the anterior nucleus of the thalamus. The columns of the fornix and the thalamo-mammillary fasciculus together form a loop resembling the figure 8, but the continuity of the loop is broken in the corpus mammillare. The column of the fornix is drawn by the stria medullaris of the pineal body and by the superficial fibers of the stria terminalis, and is said to receive also fibers from the septum pellucidum. Zuckerkandl describes an olfactory fasciculus which becomes detached from the main portion of the columns of the fornix and passes downward in front of the anterior commissure to the base of the brain, where it divides into two bundles, one joining the medial stria of the olfactory tract, the other joins the subcallosal gyrus, and through it reaches the hippocampal gyrus. The crura, crus fornicus, posterior pillars, of the fornix are prolonged backward from the body. They are flattened bands, and at their commencement are intimately connected with the undersurface of the corpus callosum. Diverging from one another, each curves around the posterior ends of the thalamus and passes downward and forward into the inferior cornu of the lateral ventricle. Here it lies along the concavity of the hippocampus, on the surface of which some of its fibers are spread out to form the alveus, while the remainder are continued as a narrow white band, the fimbria hippocampi, which is prolonged into the uncus of the hippocampal gyrus. The inner edge of the fimbria overlaps the fascia dentata hippocampi, dentate gyrus, from which it is separated by the fimbrio-dentate fissure. From its lateral margin, which is thin and ragged, the ventricular epithelium is reflected over the choroid plexus as the lateral projects to the choroidal fissure. Interventricular foramen, foramen of Monro. Between the columns of the fornix and the anterior ends of the thalami, an ovar aperture is present on either side. This is the interventricular foramen, and through it the lateral ventricles communicate with the third ventricle. Behind the epithelial lining of the foramen, the choroid plexus of the lateral ventricles are joined across the middle line. The anterior commissure, precommissure, is a bundle of white fibers connecting the two cerebral hemispheres across the middle line and placed in front of the columns of the fornix. On sagittal section it is oval in shape, its long diameter being vertical and measuring about 5 millimeters. Its fibers can be traced lateralward and backward on either side beneath the corpus striatum into the substance of the temporal lobe. It serves in this way to connect the two temporal lobes, but it also contains decussating fibers from the olfactory tracts. The septum pellucidum. Septum lucidum is a thin, vertically placed partition consisting of two laminae separating, in the greater part of their extent by a narrow chunk or interval, the cavity of the septum pellucidum. It is attached above to the undersurface of the corpus callosum, below to the anterior part of the fornix behind, and the reflected part of the corpus callosum in front. It is triangular in form, broad in front and narrow behind. Its inferior angle corresponds with the upper part of the anterior commissure. The lateral surface of each lamina is directed towards the body and anterior cornu of the lateral ventricle, and is covered by the ependema of that cavity. The cavity of the septum pellucidum, cavum septum pellucidii, pseudocele, fifth ventricle, is generally regarded as part of the longitudinal cerebral fissure, which has become shut off by the union of the hemispheres in the formation of the corpus callosum above and the fornix below. Each half of the septum therefore forms part of the medial wall of the hemisphere, and consists of a medial layer of gray substance derived from that of the cortex, and a lateral layer of white substance continuous with that of the cerebral hemisphere. This cavity is not developed from the cavity of the cerebral vesicles and never communicates with the ventricles of the brain. The choroid plexus of the lateral ventricle. Plexus choroideus ventriculus lateralis, paraplexus, is a highly vascular, fringe-like process of Pia mater which projects into the ventricular cavity. The plexus, however, is everywhere covered by a layer of epithelium continuous with the epithelial lining of the ventricle. It extends from the interventricular foramen, where it is joined with the plexus of the opposite ventricle, to the end of the inferior cornu. The part in relation to the body of the ventricle forms the vascular fringed margin of a triangular process of Pia mater, named the telechoroidea of the third ventricle, and projects from under cover the lateral edge of the fornix. It lies upon the upper surface of the thalamus, from which the epithelium is reflected over the plexus onto the edge of the fornix. This portion, in relation to the inferior cornu, lies in the concavity of the hippocampus and overlaps the fimbria hippocampi. From the lateral ends of the fimbria, the epithelium is reflected over the plexus onto the roof of the cornu. It consists of minute and highly vascular villous processes, each with an afferent and an efferent vessel. The arteries of the plexus are A. the anterior choroidal, a branch of the internal carotid which enters the plexus at the end of the inferior cornu, and B. the posterior choroidal, one or two small branches of the posterior cerebral which passes forward under the splenium. The veins of the choroid plexus unite to form a tortuous vein which courses from behind forward to the interventricular foramen and there joins with the terminal vein to form the corresponding intercerebral vein. When the choroid plexus is pulled away, the continuity between its epithelial covering and the epithelial lining of the ventricle is severed, and a cleft-like space is produced. This is named the choroidal fissure. Like the plexus, it extends from the interventricular foramen to the end of the inferior cornu. The upper part of the fissure, that is the part nearest the interventricular foramen, is situated between the lateral edge of the fornix and the upper surface of the thalamus. Farther back, at the beginning of the inferior cornu, is between the commencement of the fimbria hippocampi and the posterior end of the thalamus, while in the inferior cornu it lies between the fimbria in the floor and the stria terminalis in the roof of the cornu. The telechoroidea of the third ventricle. Telechoroidea ventriculus tertii, velum interpositum, is a double fold of Pia mater, triangular in shape, which lies beneath the fornix. The lateral portions of its lower surface rest upon the thalami, while its medial portion is in contact with the epithelial roof of the third ventricle. Its apex is situated at the interventricular foramen. Its base corresponds with the splenium of the corpus callosum, and occupies the interval between that structure above and the corpora quadrigemini and pineal body below. This interval, together with the lower portions of the choroidal fissures, is sometimes spoken of as the transverse fissure of the brain. At its base, the two layers of the velum separate from each other and are continuous with the Pia mater investing the brain in this region. Its lateral margins are modified to form the highly vascular choroid plexuses of the lateral ventricles. It is supplied by the anterior and posterior choroidal arteries already described. The veins of the telechoroidea are named the internal cerebral veins, venae galenae. They are 2 in number and run backward between its layers, each being formed at the interventricular foramen by the union of the terminal vein with the choroidal vein. The internal cerebral veins unite posteriorly in a single trunk, the great cerebral vein, vena magna galenae, which passes backward beneath the splenium and ends in the straight sinus. End of section 16. Section 17 of Gray's Anatomy Part 4. This is Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by M.L. Cohen. Anatomy of the Human Body Part 4 by Henry Gray. The Forebrain Part 5. Structure of the cerebral hemispheres. The cerebral hemispheres are composed of gray and white substance. The former covers their surface and is termed the cortex. The latter occupies the interior of the hemispheres. The white substance consists of medullated fibers varying in size and arranged in bundles separated by neuroglia. They may be divided according to their course and connections into 3 distinct systems. 1. Projection fibers connect the hemispheres with the lower parts of the brain and with the medulla spinalis. 2. Transverse or commissural fibers unite the 2 hemispheres. 3. Association fibers connect different structures in the same hemisphere, These are, in many instances, collateral branches of the projection fibers, but others are the axons of independent cells. 1. The projection fibers consist of efferent and afferent fibers uniting the cortex with the lower part of the brain and with the medulla spinalis. The principal efferent strands are 1. The motor tract, occupying the genu and the anterior thirds of the occipital part of the internal capsule, and consisting of a. The geniculate fibers, which decussate and end in the motor nuclei of the cranial nerves of the opposite side, and b. The cerebrospinal fibers, which are prolonged through the pyramid of the medulla oblongata into the medulla spinalis. 2. The corticopontine fibers, ending in the nuclei pontis. The chief afferent fibers are 1. Those of the lemniscus, which are not interrupted in the thalamus. 2. Those of the superior cerebellar peduncle, which are not interrupted in the red nucleus and thalamus. 3. Numerous fibers arising within the thalamus and passing through its stalks to the different parts of the cortex. 4. Optic and acoustic fibers, the former passing to the occipital, the latter to the temporal lobe. 2. The transverse or commissural fibers connect the two hemispheres. They include a. The transverse fibers of the corpus callosum, b. The anterior commissure, c. The posterior commissure, and d. The lyra or hippocampal commissure. They have already been described. 3. The association fibers unite different parts of the same hemisphere and are of two kinds. Those connecting adjacent gyri, short association fibers. 2. Those passing between more distant parts, long association fibers. The short association fibers lie immediately beneath the gray substance of the cortex of the hemispheres and connect together adjacent gyri. The long association fibers include the following: a. The uncinate fasciculus, b. The cingulum, c. The superior longitudinal fasciculus, d. The inferior longitudinal fasciculus, e. The perpendicular fasciculus, f. The occipital-frontal fasciculus, and g. The fornix. A. The uncinate fasciculus passes across the bottom of the lateral fissure and unites the gyri of the frontal lobe with the anterior end of the temporal lobe. B. The cingulum is a band of white matter contained within the cingulate gyrus. Beginning in front at the anterior perforated substance, it passes forward and upward parallel with the rostrum, winds around the genu, runs backward above the corpus callosum, turns around the splenium, and ends in the hippocampal gyrus. C. The superior longitudinal fasciculus passes backwards from the front lobe above the lentiform nucleus and insula. Some of its fibers end in the occipital lobe and others curve downward and forward into the temporal lobe. D. The inferior longitudinal fasciculus connects the temporal and occipital lobes, running along the lateral walls of the inferior and posterior cornues of the lateral ventricle. E. The perpendicular fasciculus runs vertically through the front part of the occipital lobe and connects the inferior parietal lobule with the fusiform gyrus. F. The occipital frontal fasciculus passes backward from the frontal lobe along the lateral border of the caudate nucleus and on the mesial aspect of the corona radiata. Its fibers radiate in a fan-like manner and pass into the occipital and temporal lobes lateral to the posterior and inferior cornua. Desjardins regards the fibers of the tapetum as being derived from this fasciculus and not from the corpus callosum. G. The fornix connects the hippocampal gyrus with the corpus mammillare, and, by means of the thalamo-mammillary fasciculus, with the thalamus. Through the fibers of the hippocampal commissure, it probably also unites the opposite hippocampal gyri. The gray substance of the hemisphere is divided into 1. That of the cerebral cortex, and 2. That of the caudate nucleus, the lentiform nucleus, the claustrum, and the nucleus amygdalae. Structure of the cerebral cortex. The cerebral cortex differs in thickness and structure in different parts of the hemisphere. It is thinner in the occipital region than in the anterior and posterior central gyri, and is also much thinner at the bottom of the sulci than on the top of the gyri. Again, the minute structure of the anterior central differs from that of the posterior central gyrus, and areas possessing a special type of cortex can be mapped out in the occipital lobe. On examining a section of the cortex with a lens, it is seen to consist of alternating white and gray layers thus disposed from the surface inward: 1. A thin layer of white substance, 2. A layer of gray substance, 3. A second white layer, outer band of Ballinger or band of Generi, 4. A second gray layer, 5. A third white layer, inner band of Ballinger, 6. A third gray layer, which rests on the medullary substance of the gyrus. The cortex is made up of nerve cells of varying size and shape, and of nerve fibers which are either medullated or naked axis cylinders embedded in a matrix of neuroglia. Nerve cells. According to Cahal, the nerve cells are arranged in 4 layers, named from the surface inward as follows. 1. The molecular layer. 2. The layer of small pyramidal cells. 3. The layer of large pyramidal cells. 4. The layer of polymorphous cells. The molecular layer. In this layer, the cells are polygonal, triangular, or fusiform in shape. Each polygonal cell gives off some 4 or 5 dendrites, while its axon may arise directly from the cell or from one of its dendrites. Each triangular cell gives off 2 or 3 dendrites, from one of which the axon arises. The fusiform cells are placed with the long axis parallel to the surface and are mostly bipolar, each pole being prolonged into a dendrite which runs horizontal for some distance and furnishes ascending branches. Their axons, 2 or 3 in number, arise from the dendrites and, like them, take a horizontal course, giving off numerous ascending collaterals. The distribution of the axons and dendrites of all 3 sets of cells is limited to the molecular layer, the layer of small, and the layer of large pyramidal cells. The cells in these 2 layers may be studied together, since, with the exception of the differentiation in size and the more superficial position of the smaller cells, they resemble each other. The average length in the small cells is from 10 to 15 microns, that of the large cells from 20 to 30 microns. The body of each cell is pyramidal in shape, its base being directed to the deeper parts and its apex towards the surface. It contains granular pigment and stains deeply with ordinary reagents. The nucleus is of large size and round or oval in shape. The base of the cell gives off the axis cylinder, and this runs into the central white substance, giving off collaterals in its course, and is distributed as a projection, commissural, or association fiber. The apical and basal parts of the cell give off dendrites. The apical dendrite is directed towards the surface and ends in the molecular layer by dividing into numerous branches, all of which may be seen, when prepared by the silver or methylene blue method, to be studied with projection bristle-like processes. The largest pyramidal cells are found in the upper part of the anterior central gyrus and in the paracentral lobule. They are often arranged in groups or nests from 3 to 5 and are named the giant cells of Betz. In the former situation, they may exceed 50 microns in length and 40 microns in breadth, while in the paracentral lobule, they may attain a length of 65 microns. Layer of polymorphous cells. The cells of this layer, as their name implies, are very irregular in contour. They may be fusiform, oval, triangular, or star-shaped. Their dendrites are directed outward, but do not reach so far as the molecular layer. Their axons pass into the subjacent white matter. There are 2 other kinds of cells in the cerebral cortex. They are A. The cells of Golgi, the axons of which divide immediately after their origins into large number of branches, which are directed towards the surface of the cortex. B. The cells of Martinati, which are chiefly found in the polymorphous layer. Their dendrites are short and may have an ascending or descending course, while their axons pass out into the molecular layer and form an extensive horizontal arborization. Nerve fibers. These fill up a large part of the intervals between the cells and may be medullated or non-medullated, the latter comprising the axons of the smallest pyramidal cells and the cells of Golgi. In their direction, the fibers may be either tangential or radial. The tangential fibers run parallel to the surface of the hemispheres, intersecting the radial fibers at right angles. They constitute several striata, of which the following are the more important. 1. A striatum of white fibers covering the superficial aspect of the molecular layer, plexus of Exner. 2. The band of Bechtereau in the outer part of the layer of small pyramidal cells. 3. The band of Jenneri, or external band of Ballinger, running through the latter of large pyramidal cells. 4. The interior band of Ballinger, between the layers of the large pyramidal cells and the polymorphous layer. 5. The deep tangential fibers in the lower part of the polymorphous layer. The tangential fibers consist of A. The collateral of the pyramidal and polymorphous cells and the cells of Martinati. B. The branching axons of Golgi's cells. C. The collaterals and terminal arborizations of the projection, commissural, and association fibers. The radial fibers. Some of these, that is, the axons of the pyramidal and polymorphous cells, descend into the central white matter, while others, the terminations of the projection, commissural or association fibers, ascend to end in the cortex. The axons of the cells of Martinati are also ascending fibers. Special types of cerebral cortex. It has already been pointed out that the minute structure of the cortex differs in different regions of the hemisphere, and A.W. Campbell has endeavored to prove, as the result of an exhaustive examination of a series of humans and anthropoid brains, quote, that there exists a direct correlation between the physiologic function and histologic structure, end quote. The principal regions where the typical structures departed from will now be referred to. 1. In the calcarine fissure and the gyri bounding it, the internal line of Ballinger is absent, while the band of generi is of considerable thickness and forms a characteristic feature of this region of the cortex. If a section be examined microscopically, an additional layer of cells is seen to be interpolated between the molecular layer and the layer of small pyramidal cells. This extra layer consists of 2 or 3 strata of fusiform cells, the long axis of which are at right angles to the surface. Each cell gives off 2 dendrites, external and internal, from the latter of which the axon arises and passes into the white central substance. In the layer of small pyramidal cells, fusiform cells identical with the above are seen, as well as ovoid or star-like cells with ascending axons, cells of Martinati. This is the visual area of the cortex, and it has been shown by J.S. Bolton that in old-standing cases of optic atrophy, the thickness of Generi's band is reduced by nearly 50%. A.W. Campbell says, quote, histologically, 2 distinct types of cortex can be made out in the occipital lobe. The first of these coats the walls and bounding convolutions of the calcarine fissure and is distinguished by the well-known line of Generi or Vic d'Azur. The second area forms an investing zone a centimeter or more broad around the first and is characterized by a remarkable wealth of fibers, as well as by a curious piriform cells of large size richly stocked with chromophobe elements, cells which seem to have escaped the observation of Ramani Cajal, Bolton, and others who have worked at this region. As to the functions of these 2 regions, there is abundant evidence, anatomical, embryological, and pathological, to show that the first, or calcarine area, is that to which visual sensations primarily pass, and we are gradually obtaining proof to the effect that the second investing area is constituted for the interpretation and further elaboration of these sensations. These areas therefore deserve the name visio-sensory and visio-psychic, end quote. 2. The anterior central gyrus is characterized by the presence of the giant cells of Betz and by, quote, a wealth of nerve fibers immeasurably superior to that of any other part, end quote, Campbell, and in these respects differs from the posterior central gyrus. These 2 gyri, together with the paracentral lobule, were long regarded as constituting the, quote, motor areas, end quote, of the hemispheres. But Sherrington and Grunbaum have shown that in the chimpanzee the motor area never extends onto the free face of the posterior central gyrus, but occupies the entire length of the anterior central gyrus, and in most cases the greater part or whole of its width. It extends into the depth of the central sulcus, occupying the anterior wall, and in some places the floor, and in some extending even into the deeper part of the posterior wall of the sulcus. 3. In the hippocampus, the molecular layer is very thick and contains a large number of Golgi cells. It has been divided into 3 strata. A. S. convolution, or S. granulosum, containing many tangential fibers. B. S. lacunosum, presenting numerous vascular spaces. C. S. radiatum, exhibiting a rich plexus of fibrils. The 2 layers of pyramidal cells are condensed into one, and the cells are mostly of large size. The axons of the cells of the polymorphous layer may run in an ascending, a descending, or a horizontal direction. Between the polymorphous layer and the ventricular ependema is the white substance of the alveus. 4. In the fascia dentata hippocampi, or dentate gyrus, the molecular layer contains some pyramidal cells, while the layer of pyramidal cells is almost entirely represented by small ovoid cells. 5. The olfactory bulb. In many of the lower animals, this contains a cavity which communicates through the olfactory tract with the lateral ventricle. In man, the original cavity is filled up by neuroglia, and its wall becomes thickened, but more and more so on the ventral than on the dorsal aspect. Its dorsal part contains a small amount of gray and white substance, but it is scanty and ill-defined. A section through the ventral part shows it to consist of the following layers from without inward. 1. A layer of olfactory nerve fibers, which are the non-medullated axons prolonged from the olfactory cells of the nasal cavity and reach the bulb by passing through the cribriform plate of the ethmoid bone. At first they cover the bulb, and then penetrate at the end by forming synapses with the dendrites of the mitral cells, presently to be described. 2. Glomerular layer. This layer contains numerous spheroidal reticulated enlargements termed glomeruli, produced by the branching and arborizations of the processes of the olfactory nerve fibers with the descending dendrites of the mitral cells. 3. Molecular layer. This is formed of a matrix of neuroglia embedded in which are the mitral cells. These cells are pyramidal in shape, and the basal part of each gives off a thick dendrite which descends into the glomerular layer where it arborizes as indicated above, and others which interlace with similar dendrites of neighboring mitral cells. The axons pass through the next layer into the white matter of the bulb, and after becoming bent on themselves at right angle, are continued into the olfactory tract. 4. Nerve fiber layer. This lies next to the central core of neuroglia, and its fibers consist of the axons or afferent processes of the mitral cells passing into the brain. Some efferent fibers are, however, also present and end in the molecular layer, but nothing is known as to their exact origin. Weight of the encephalon. The average weight of the brain in the adult male is about 1,380 grams, that of the female about 1,250 grams. In the male, the maximum weight out of 278 cases was 1,840 grams, and the minimum weight 964 grams. The maximum weight of the adult female brain out of 191 cases was 1,585 grams, and the minimum weight 879 grams. The brain increases rapidly during the first 4 years of life and reaches its maximum weight by about the 20th year. As age advances, the brain decreases slowly in weight. In old age, the decrease takes place more rapidly, to the extent of about 28 grams per year. The human brain is heavier than that of any of the lower animals except the elephant and whale. The brain of the former weighs from 3.5 to 5.4 kilograms, and that of the whale, in a specimen 19 meters long, weighed rather more than 6.7 kilograms. Cerebral localization. Physiological and pathological research have now gone far to prove that a considerable part of the surface of the brain may be mapped out into a series of more or less definite areas, each of which is intimately connected with some well-defined function. Motor areas. The motor area occupies the anterior central and frontal gyri and the paracentral lobule. The centers for the lower limb are located on the uppermost part of the anterior central gyrus and its continuation onto the paracentral lobule. Those for the trunk are on the upper portion, and those for the upper limb on the middle portion of the anterior central gyrus. The facial centers are situated on the lower part of the anterior central gyrus, those for the tongue, larynx, muscles of mastication, and pharynx on the frontal operculum, while those for the head and neck occupy the posterior end of the middle frontal gyrus. Sensory areas. Tactile and temperature senses are located on the posterior central gyrus, while the sense of form and solidity is on the superior parietal lobule and precuneus. With regard to the special senses, the area for the sense of taste is probably related to the uncus and hippocampal gyrus. The auditory area occupies the middle third of the superior temporal gyrus and adjacent gyri in the lateral fissure. The visual area, the calcarine fissure and cuneus. The olfactory area, the rhinencephalon. As special centers of much importance may be noted, the emissive center for speech on the left inferior frontal and anterior central gyri, Broca, the auditory receptive center on the transverse and superior temporal gyri, and the visual receptive center on the lingual gyrus and cuneus. End of section 17.
Section 18 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Veronica Jenkins. Anatomy of the Human Body, Part 4, by Henry Gray. Composition and central connections of the spinal nerves. The typical spinal nerve consists of at least 4 types of fibers: the somatic sensory, sympathetic afferent or sensory, somatic motor, and sympathetic efferent or preganglionic. The somatic sensory fibers, afferent fibers, arise from cells in the spinal ganglia and are found in all the spinal nerves, except occasionally the first cervical, and conduct impulses of pain, touch, and temperature from the surface of the body through the posterior roots to the spinal cord, and impulses of muscle sense, tendon sense, and joint sense from the deeper structures. The sympathetic afferent fibers conduct sensory impulses from the viscera through the rami communicantes and posterior roots to the spinal cord. They are probably limited to the white rami connected with the spinal nerves in 2 groups, that is, the first thoracic to the second lumbar and the second sacral to the fourth sacral nerves. The somatic motor fibers, afferent fibers, arise from cells in the anterior column of the spinal cord and pass out through the anterior roots to the voluntary muscles. The sympathetic afferent fibers probably arise from cells in the lateral column or the base of the anterior column and emerge through the anterior roots and white rami communicantes. These are preganglionic fibers which end in various sympathetic ganglia, from which postganglionic fibers conduct the motor impulses to the smooth muscles of the viscera and vessels and secretory impulses to the glands. These fibers are also limited to 2 regions, the first thoracic to the second lumbar and the second sacral to the fourth sacral nerves. The afferent fibers, which pass into the spinal cord, establish various types of connections, some within the cord itself for spinal reflexes, others for reflexes connected with higher centers in the brain, while still others conduct impulses of conscious sensation by a series of neurons to the cerebral cortex. The intrinsic spinal reflex paths. The collaterals and terminals of the ascending and descending branches of the posterior root fibers, which leave the fasciculus cuneatus to enter the gray matter of the spinal cord, end in various ways. Many end in the dorsal column, some near its apex, others in the substance of Rolando, others in the intermediate region between the dorsal and ventral columns. Others traverse the whole thickness of the gray matter to reach the ventral column, others end in the dorsal nucleus, and others pass through the gray commissure to the dorsal column of the opposite side. All of these collaterals and terminals end in connection with cells or dendrites of cells in the gray columns. The axons of these cells have various destinations. Some pass out into the lateral and ventral funiculi and turn upward to reach the brain. Those concerned with the intrinsic spinal reflexes come into relation either directly or indirectly with motor cells in the anterior column. It is very unlikely that either the terminals or collaterals of the dorsal root fibers affect simple direct connections with the motor cells of the ventral column. There is at least one, if not several, intercalated neurons in the path. These intercalated or correlation neurons may have short axons that do not pass out of the gray matter, or the axons may pass out into the proper fasciculi and extend for varying distances up and down or in both directions, giving off collaterals and finally terminating in the gray matter of the same or the opposite side. The shortest fibers of the proper fasciculi lie close to the gray matter. The longest ones are nearer the periphery of the proper fasciculi and are more or less intermingled with the long ascending and descending fasciculi, which occupy the more marginal regions of the spinal cord. Each sensory neuron with its ascending and descending branches, giving off as it does many collaterals into the gray matter, each one of which may form a synapse with one or several correlation neurons, is thus brought into relation with many correlation neurons, and each one of these in turn, with its ascending and descending branches and their numerous collaterals, is brought into relation either directly or through the intercalation of additional correlation neurons with great numbers of motor cells in the anterior column. The great complexity of these so-called simple reflex mechanisms in the least complex portion of the nervous system, the spinal cord, renders them extremely difficult of exact analysis. The association or correlation neurons are concerned not only with the reflex mechanism of the spinal cord, but play an equally important role in the transmission of impulses from the higher centers in the brain to the motor neurons of the spinal cord. The complex mechanisms just described are probably concerned not so much in the contraction of individual muscles as in the complicated action of groups of muscles concerned in the enormous number of movements which the limbs and trunk exhibit in the course of our daily life. Sensory pathways from the spinal cord to the brain. The posterior root fibers conducting the impulses of conscious muscle sense, tendon sense, and joint sense, those impulses which have to do with the coordination and adjustment of muscular movements, ascend in the fasciculus gracilis and fasciculus cuneatus to the nucleus gracilis and the nucleus cuneatus in the medulla oblongata. In the nucleus gracilis and nucleus cuneatus, synaptic relations are found with neurons whose cell bodies are located in these nuclei and whose axons pass by way of the internal arcuate fibers, cross in the raphe to the opposite side in the region between the olives, and turn abruptly upward to form the medial lemniscus or medial fillet. The medial fillet passes upward in the ventral part of the formatio reticularis through the medulla oblongata, pons, and midbrain to the principal sensory nucleus of the ventrolateral region of the thalamus. Here the terminals form synapses with neurons of the third order, whose axons pass through the internal capsule and corona radiata to the somatic sensory area of the cortex in the postcentral gyrus. Fibers conducting the impulses of unconscious muscle sense pass to the cerebellum partly by way of the fasciculus gracilis and fasciculus cuneatus to the nucleus gracilis and nucleus cuneatus. Thence, neurons of the second order convey the impulses either via the dorsal external arcuate fibers directly into the inferior peduncle of the cerebellum, or via the ventral external arcuate fibers, which are continued from the internal arcuate fibers through the ventral part of the raphe, and, after crossing the midline, emerge on the surface of the medulla in the ventral sulcus between the pyramids, or in the groove between the pyramid and the olive. They pass over the lateral surface of the medulla and olive to reach the inferior peduncle, through which they pass to the cerebellum. Other fibers conducting impulses of unconscious muscle sense pass upward in the dorsal spinocerebellar fasciculus, which arises from cells in the nucleus dorsalis. The posterior root fibers conducting these impulses pass into the fasciculus cuneatus, and the collaterals from them to the nucleus dorsalis are said to come almost exclusively from the middle area of the fasciculus cuneatus. They form by their multiple divisions baskets about the individual cells of the nucleus dorsalis, each fiber coming in relation with the bodies and dendrites of several cells. The axons of the second order pass into the dorso-spinocerebellar fasciculus of the same side, and ascend along the lateral surface of the spinal cord and medulla oblongata until they arrive at the level of the olive. They then curve backward beneath the external arcuate fibers into the inferior peduncle and pass into the cerebellum. Here they give off collaterals to the dentate nucleus and finally terminate in the cortex of the dorsal and superior portion of the vermis, partly on the same side, but to a great extent by way of a large commissure to the opposite side. The fibers lose their myelin sheaths as they enter the gray substance, and terminate by end ramifications along the nerve cells and their processes. Some of the fibers are said to end in the nucleus dentatus and the roof nuclei of the cerebellum, the nucleus globosus, nucleus emboliformis, and nucleus fastigius, and others pass through them to terminate in the inferior vermis. A few fibers of the dorsal spinocerebellar fasciculus are said not to enter the inferior peduncle, but to pass with the ventro-spinocerebellar fasciculus. The cerebellar reflex arc is supposed to be completed by the fibers of the superior peduncle, which pass from the cerebellum to the red nucleus of the midbrain, where some of their terminals and collaterals form synapses with neurons whose axons descend to the spinal cord in the cerebrospinal fasciculus. The terminal and collaterals of this fasciculus end either directly or indirectly about the motor cells in the anterior column. The ventral-spinocerebellar fasciculus, since most of its fibers pass to the cerebellum, is also supposed to be concerned in the conduction of unconscious muscle sense. The location of its cells of origin is uncertain. They are probably in or near the dorsal nucleus of the same and the opposite side. Various other locations are given: the dorsal column, the intermediate zone of the gray matter, and the central portion of the anterior column. The neurons of the first order, whose central fibers enter the fasciculus cuneatus from the dorsal roots, send collaterals and terminals to form synapses with these cells. The fibers which come from the opposite gray columns cross some in the white and some in the gray commissure, and pass with fibers from the same side through the lateral funiculus to the marginal region ventral to the dorsal spinocerebellar fasciculus. The fasciculus begins about the level of the third lumbar nerve and continues upward on the lateral surface of the spinal cord and medulla oblongata until it passes under cover of the external arcuate fibers. It passes just dorsal to the olive, and above this joins the lateral edge of the lateral lemniscus along which it runs, ventral to the roots of the trigeminal nerve, almost to the level of the superior colliculus. It then crosses over the superior peduncle, turns abruptly backward along its medial border, enters the cerebellum with it, and ends in the vermis of the same and the opposite side. Some of its fibers are said to join the dorsal spinocerebellar fasciculus in the medulla oblongata and enter the cerebellum through the inferior peduncle. A number of fibers are said to continue upward in the dorsolateral part of the tegmentum as far as the superior colliculus, and a few pass to the thalamus. They probably form part of the sensory or higher reflex path. The posterior root fibers conducting impulses of pain and temperature probably terminate in the posterior column or the intermediate region of the gray matter soon after they enter the spinal cord. The neurons of the second order are supposed to pass through the anterior commissure to the superficial anterolateral fasciculus, tract of Gowers, and pass upward in that portion of it known as the lateral spinothalamic fasciculus. This fasciculus lies along the medial side of the ventro-spinocerebellar fasciculus. It is stated by some authors that the pain fibers pass upward in the anterolateral ground bundles. In some of the lower mammals, this pathway carries the pain fibers upward by a series of neurons, some of which cross to the opposite side, so that in part there is a double path. In man, however, the lateral spinothalamic fasciculus is probably the most important pathway. On reaching the medulla, these fibers continue upward through the formatio reticularis in the neighborhood of the median fillet to the thalamus, probably its ventrolateral region. Whether higher neurons convey the pain impulses to the cortex through the internal capsule is uncertain. The pathway is probably more complex, and Head is of the opinion that our sensations of pain are essentially thalamic. The pain and temperature pathways in the lateral spinothalamic fasciculus are not so closely intermingled but that one can be destroyed without injury to the other. Ransom suggests that the non-medullated fibers of the posterior roots, which turn into Lesor's tract and ascend or descend for short distances, not exceeding 1 or 2 segments, and finally end in the substantia gelatinosa, are in part at least pain fibers, and that the fasciculus of Lasore and the substantia gelatinosa represent part of the mechanism for reflexes associated with pain conduction and reception, while the fibers to the higher centers pass up in the spinothalamic tract. The fibers of tactile discrimination, according to Head and Thompson, pass up in the fasciculus cuneatus and fasciculus gracilis of the same side and follow the path of the muscle sense fibers. The axons of the second order arising in the nucleus cuneatus and gracilis cross with the internal arcuate fibers and ascend to the thalamus with the medial lemniscus. Thence, by neurons of higher order, the impulses are carried to the somatic sensory area of the cortex through the internal capsule. The other touch fibers, shortly after entering the spinal cord, terminate in the dorsal column or intermediate gray matter. Neurons of the second order send their axons through the anterior commissure to pass upward in the anterolateral funiculus, probably in the ventral spinothalamic fasciculus. In the medulla they join or pass upward in the neighborhood of the medial lemniscus to the thalamus, and thence by neurons of higher order to the somatic sensory area of the cortex. The remaining ascending fasciculi form a part of the complex known as the superficial anterolateral fasciculus tract of Gowers. The spinotectal fasciculus, as its name indicates, is supposed to have its origin in the gray matter of the cord and terminations in the superior and inferior colliculi of the midbrain, serving for reflexes between the cord and the visceral and auditory centers of the midbrain. The spino-olivary fasciculus, olivospinal, bulbospinal, Hellwig's bundle, is likewise of unknown constitution and function. There is uncertainty even in regard to the direction of its fibers. The sympathetic afferent fibers, visceral afferent, viscerosensory, splanchnic afferent, enter the spinal cord by the posterior roots of the thoracic and first 2 or 3 lumbar nerves, and the second to the 4th sacral nerves. The fibers pass to these nerves from the peripheral sympathetic system through the white rami communicantes. Some of the cell bodies of these afferent fibers are located in the spinal ganglia, and others are in the sympathetic ganglia. Some of the afferent sympathetic fibers end about the cell bodies of sensory neurons, and visceral impulses are thus transmitted to these neurons, which conduct them as well as their own special impulses to the spinal cord. Other sympathetic afferent neurons, whose cell bodies are located in the spinal ganglia, send collaterals to neighboring cells of somatic sensory neurons, and thus have a double path of transmission to the spinal cord. Such an arrangement provides a mechanism for some of the referred pains. These sympathetic afferent fibers presumably divide on entering the spinal cord into ascending and descending branches. Their distribution and termination within the spinal cord are unknown. Some of them probably eventually came into relation with the sympathetic afferent fibers whose cell bodies are located in the lateral column. Our knowledge concerning both the termination and origin of these fibers is very unsatisfactory. The sympathetic afferent fibers, splanchnic motor, visceromotor, preganglionic fibers, are supposed to arise from cells in the intermediate zone between the dorsal and ventral gray columns and in the intermediolateral column at the margin of the lateral column. These preganglionic sympathetic fibers are not distributed throughout the entire series of spinal nerves, but are confined to 2 groups, the thoracolumbar from the 1st thoracic to the 2nd or 3rd lumbar nerves, and the sacral group from the 2nd to the 4th sacral nerves. They pass out with the anterior root fibers and through the rami communicantes to end in sympathetic ganglia. The impulses are distributed from cells in these ganglia through postganglionic fibers to the smooth muscles and glands. The thoracolumbar outflow and the sacral outflow form 2 distinct functional groups which are considered more fully under the sympathetic system. End of section 18. Recorded by Veronica Jenkins in Ottawa, Illinois.
Section 19 of Gray's Anatomy. Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 1. 4e. Composition and Central Connections of the Spinal Nerves. The cranial nerves are more varied in their composition than the spinal nerves. Some, for example, contain somatic motor fibers only. Others contain the various types of fibers found in the spinal nerves, namely, somatic motor, sympathetic efferent, somatic sensory, and sympathetic sensory. In addition, there are included the nerves of the special senses, namely, the nerves of smell, sight, hearing, equilibration, and taste. The hypoglossal nerve, 12th cranial, consists of somatic motor fibers only and supplies the muscles of the tongue. Its axons arise from cells in the hypoglossal nucleus and pass forward between the white reticular formation and the grey reticular formation to emerge from the anterolateral sulcus of the medulla. The hypoglossal nuclei of the two sides are connected by many commissural fibers and also by dendrites of motor cells which extend across the midline to the opposite nucleus. The hypoglossal nucleus receives either directly or indirectly numerous collaterals and terminals from the opposite pyramidal tract, corticobulbar or cerebrobulbar fibers, which convey voluntary motor impulses from the cerebral cortex. Many reflex collaterals enter the nucleus from the secondary sensory paths of the trigeminal and vagus, and probably also from the nervus intermedius and the glossopharyngeal. Collaterals from the posterior longitudinal bundle and the ventral longitudinal bundle are said to pass to the nucleus. The accessory nerve, 11th cranial, contains somatic motor fibers. The spinal part arises from lateral cell groups in the anterior column near its dorsolateral margin in the upper 5 or 6 segments of the cord. Its roots pass through the lateral funiculus to the lateral surface of the cord. It supplies the trapezius and sternocleidomastoideus. The cranial part arises from the nucleus ambiguus, the continuation in the medulla oblongata of the lateral cell groups of the anterior column of the spinal cord from which the spinal part has origin. The upper part of the nucleus ambiguus gives motor fibers to the vagus and glossopharyngeal nerves. The cranial part sends its fibers through the vagus to the laryngeal nerves to supply the muscles of the larynx. The root fibers of the cranial part of the accessory nerve pass anterior to the spinal tract of the trigeminal, while those of the vagus pass through or dorsal to the trigeminal root, and emerge in the line of the posterolateral sulcus. The nucleus of origin of the spinal part undoubtedly receives either directly or indirectly terminals and collaterals controlling voluntary movements from the pyramidal tracts. It is probable that terminals and collaterals reach the nucleus either directly or indirectly from the rubrospinal and vestibulospinal tracts. It is also connected indirectly with the spinal somatic sensory nerves by association fibers of the proper fasciculi. The cranial part receives indirectly or directly terminals and collaterals from the opposite pyramidal tract and form the terminal sensory nuclei of the cranial nerves. A few fibers of the cranial part are said to arise in the dorsal nucleus of the vagus, and are thus sympathetic efferent. They are said to join the vagus nerve. The vagus nerve, 10th cranial, contains somatic sensory, sympathetic afferent, somatic motor, sympathetic efferent, and taste fibers. The afferent fibers, somatic sensory, sympathetic, and taste, have their cells of origin in the jugular ganglion and in the nodosal ganglion, ganglion of the trunk, and entering the medulla divide into ascending and descending branches as do the sensory fibers of the posterior roots of the spinal nerves after they enter the spinal cord. 1. The somatic sensory fibers are few in number, convey impulses from a limited area of the skin on the back of the ear and posterior part of the external auditory meatus, and probably join the spinal tract of the trigeminal nerve to terminate in its nucleus. Connections are probably established to the central path of the trigeminal with the thalamus and somatic sensory area of the cortex for the conscious recognition of impulses. The descending fibers in the spinal tract of the trigeminal, terminating in the nucleus of the tract, probably establish relations through connecting neurons with motor nuclei in the anterior column of the spinal cord and with motor nuclei of the medulla. 2. The sympathetic afferent fibers are usually described as terminating in the dorsal nucleus of the vagus and glossopharyngeal. Some authors, however, believe they join the Tractus Solitarius and terminate in its nucleus. These afferent fibers convey impulses from the heart, the pancreas, and probably from the stomach, esophagus, and respiratory tract. Their terminals in the dorsal nucleus come into relation with neurons whose axons probably descend into the spinal cord, conveying impulses to the motor nuclei supplying fibers to the muscles of respiration, i.e. the phrenic nerve and the nerves to the intercostal and levatorius costarum muscles. Other axons probably convey vasomotor impulses to certain sympathetic efferent neurons throughout the spinal cord. The dorsal nucleus of the ala cinerea, and the posterior continuation of it into the commissural nucleus of the ala cinerea constitute probably the so-called respiratory and vasomotor center of the medulla. The shorter reflex neurons of the dorsal nucleus probably effect connections, either directly or indirectly, with motor cells of the vagus itself and other cranial nerves. 3. Taste fibers conducting impulses from the epiglottis and larynx are supposed to pass in the vagus and join the Tractus Solitarius, finally terminating in the nucleus of the Tractus Solitarius. It is not certain that this nucleus represents the primary terminal center for taste, and some authors maintain that the taste fibers terminate in the dorsal nucleus. The secondary ascending pathways from the primary gustatory nucleus to the cortex, as well as the location of the cortical center for taste, are unknown. A gustatory center has been described near the anterior end of the temporal lobe. The nucleus of the Tractus Solitarius is connected with motor centers of the pons, medulla, and spinal cord for the reactions of mastication and swallowing. 4. Somatic motor fibers to the cross-striated muscles of the pharynx and larynx arise in the nucleus ambiguus. This nucleus undoubtedly receives either directly or indirectly collaterals or terminals from the opposite pyramidal tract controlling the voluntary movements of the pharynx and larynx. The reflex pathways conveying impulses from the terminal sensory nuclei are unknown, but probably form part of the intricate maze of fibers constituting the reticular formation. 5. Sympathetic efferent fibers arise from cells in the dorsal nucleus of the ala cinerea. These are preganglionic fibers of the sympathetic system, and all terminate in sympathetic ganglia from which postganglionic fibers are distributed to various organs, i.e. motor fibers to the esophagus, stomach and small intestine, gallbladder, and to the lungs, inhibitory fibers to the heart, secretory fibers to the stomach and pancreas. The dorsal nucleus not only receives terminals of sympathetic afferent fibers for reflexes, but undoubtedly receives terminals and collaterals from many other sources, but the exact pathways are at present unknown. The glossopharyngeal nerve, 9th cranial, is similar to the vagus nerve as regards its central connections, and is usually described with it. It contains somatic sensory, sympathetic afferent, taste, somatic motor, and sympathetic efferent fibers. The afferent sensory fibers arise from cells in the superior ganglion and in the petrosal ganglion. The same uncertainty exists concerning the nuclei of termination and nuclei of origin of the various components as for the vagus. 1. The somatic sensory fibers are few in number. Some are distributed with the auricular branch of the vagus to the external ear, others probably pass to the pharynx and fauces. They are supposed to join the spinal tract of the trigeminal and terminate in its nucleus. The connections are similar to those of the somatic sensory fibers of the vagus. 2. Sympathetic afferent fibers from the pharynx and middle ear are supposed to terminate in the dorsal nucleus. Connections are probably established with motor nuclei concerned in chewing and swallowing. Very little is known, however, about the connections with other parts of the brain. 3. Taste fibers from the tongue probably terminate in the nucleus of the Tractus Solitarius. These fibers, together with similar fibers from the facial, nervus intermedius, and the vagus are supposed to form the Tractus Solitarius and terminate in its nucleus. The central connections have been considered under the vagus. 4. Somatic motor fibers to the silopharyngeus muscle arise in the upper end of the nucleus ambiguus. The existence of these fibers in the roots of the glossopharyngeal is uncertain, as there are other paths by which such fibers might reach in the glossopharyngeal from the vagus. The sources of impulses passing to the nucleus ambiguus are considered under the vagus. 5. Sympathetic efferent fibers, motor and secretory fibers, arise from the nucleus dorsalis. Some authors believe that the secretory fibers to the parotid gland arise from a distinct nucleus, the inferior salivatory nucleus, situated near the dorsal nucleus. The preganglionic fibers from this nucleus terminate in the otic ganglion. The postganglionic fibers from the otic ganglion pass to the parotid gland. The acoustic nerve, 8th cranial, consists of two distinct nerves, the cochlear nerve, this nerve of hearing, and the vestibular nerve, the nerve of equilibration. End of Part 19. Section 20 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 2. The cochlear nerve arises from bipolar cells in the spiral ganglion of the cochlea. The peripheral fibers end in the organ of Corti. The central fibers bifurcate as they enter the cochlear nucleus. The short ascending branches end in the ventral portion of the nucleus. The longer descending branches terminate in the dorsal portion of the nucleus. From the dorsal portion of the cochlear nucleus axons arise which pass across the dorsal aspect of the inferior peduncle and the floor of the fourth ventricle, the striae medullaris, to the median sulcus. Here they dip into the substance of the pons, cross the median plane, and join the lateral lemniscus. Some of the fibers terminate in the superior olivary nucleus. The fibers of the stria medullaris are not always visible on the floor of the rhomboid fossa. From the ventral portion of the cochlear nucleus, axons pass into the trapezoid body. Here some of them end in the superior olivary nucleus of the same size. Others cross the midline and end in the superior olivary nucleus of the opposite side, or pass by these nuclei, giving off collaterals to them, and join the lateral lemniscus. Other fibers either terminate in, or give off collaterals to the nucleus of the trapezoid body of the same or the opposite side. Other fibers from the ventral portion of the cochlear nucleus pass dorsal to the inferior peduncle and then dip into the substance of the pons to join the trapezoid body or the superior olivary nucleus of the same side. From the superior olivary nucleus of the same and opposite sides, axons join in the lateral lemniscus. Collaterals and probably terminals also pass from the lateral lemniscus to other nuclei in its path, and receive in turn axons from these nuclei. These are the accessory nucleus, the medial preolivary nucleus, the lateral preolivary or semilunar nucleus, and the nucleus of the lateral lemniscus. The trapezoid body consists of horizontal fibers in the ventral part of the formatio reticularis of the lower part of the pons, behind its deep transverse fibers and the pyramid bundles. The axons come from the dorsal and ventral portions of the cochlear nucleus. After crossing the raphe, where they decussate with those from the opposite side, they turn upward to form the lateral lemniscus. Fibers from the striae medullaris contribute to the trapezoid body. In addition, it sends terminals or collaterals to, and receive axons from, the superior olivary nucleus, the nucleus of the trapezoid body, the lateral preolivary or semilunar nucleus, and the mesial preolivary nucleus. The cochlear nucleus, the terminal nucleus for the nerve of hearing, is usually described as consisting of a larger dorsal nucleus on the dorsal and lateral aspect of the inferior peduncle, forming a prominent projection, the acoustic tubercle, and a ventral or accessory cochlear nucleus more ventral to the inferior peduncle. The two nuclei are continuous and are merely portions of one large nucleus. The axons from cells of the spiral ganglion of the cochlear nerve on reaching the nucleus divide into ascending and descending branches which enter the ventral and dorsal nuclei respectively. Axons from the large fusiform cells of the dorsal nucleus pass partly by way of the stria medullaris to the trapezoid body and lateral lemniscus and the nuclei associated with the former, and partly transversely beneath the inferior peduncle and spinal tract of the trigeminal to the trapezoid body. Axons from the ventral cochlear nucleus pass partly by the stria medullaris, but for the most part horizontally to the trapezoid body. The superior olivary nucleus is a small mass of grey matter situated on the dorsal surface of the lateral part of the trapezoid body. Some of its axons pass backward to the adjacent nucleus. This bundle is known as the peduncle of the superior olivary nucleus. Other fibers from the nucleus join the posterior longitudinal bundle and terminate in the nuclei of the trochlear and oculomotor nerves. The majority of axons, after giving off collaterals to the nuclei itself, join the lateral lemniscus of the same side. Other axons pass in the trapezoid body toward the ventral portion of the cochlear nucleus. The nucleus of the trapezoid body lies between the root fibers of the adjacent nerve and the superior olivary nucleus. Its cells lie among the fibers of the trapezoid body. In it terminate fibers and collaterals of the trapezoid body, which come from the cochlear nucleus of the opposite and probably the same side, and from the opposite trapezoid nucleus. They terminate in the nucleus of the trapezoid body in diffuse arborizations and peculiar end plaques or acoustic calyces of yellowish color which fuse with the cell bodies. Its cells are round and of medium size. Their axons pass into the trapezoid body, cross the median line, and probably join the lateral fillet. The lateral preolivary or semilunar nucleus lies ventral to the superior olivary nucleus. In it, end terminals and collaterals of the trapezoid body and probably fibers of the opposite cochlear nucleus. Its axons mingle with the trapezoid body and join the lateral fillet. The mesial preolivary nucleus is in contact with the ventral side of the nucleus of the trapezoid body. It receives many collaterals from the trapezoid body. Its cells are smaller than those of the trapezoid nucleus. Their axons join the lateral fillet. The lateral lemniscus, lateral fillet, the continuation upward of the central path of hearing, consists of fibers which come from the cochlear nuclei of the same and opposite side by way of the trapezoid body and from the preolivary nuclei. It lies in the ventral or ventrolateral part of the reticular formation of the pons, at first ventral, then lateral to the median fillet. Above the pons these ascending fibers come to the surface at the side of the reticular formation in the trigonum lemnisci and are covered by a layer of ependyma. This part of the lateral lemniscus is known as the fillet of Rayleigh. On reaching the level of the inferior colliculus, the dorsal fibers which overlie the superior peduncle decussate in the velum medullare anterius with similar fibers of the opposite side. Numerous small masses of cells are scattered along the path of the lateral lemniscus above the superior olivary nucleus, and constitute lower and upper nuclei of the lateral lemniscus. They are supplied with many collaterals and possibly terminals from the fibers of the lemniscus. The axons of the lower nucleus of the lateral lemniscus, which arise from the larger stellate or spindle-shaped cells, with long, smooth, much-branched dendrites, are said by some authors to join the lateral lemniscus. But according to Cahal, they pass medially toward the raphe. Their termination is unknown. The cells of the upper nucleus of the lateral lemniscus are more scattered. The same uncertainty exists in regard to their termination. The fibers of the lateral lemniscus end by terminals or collaterals in the inferior colliculus and the medial geniculate body. A few of the fibers are said to pass by the inferior colliculus to terminate in the middle portion of the stratum griseum of the superior colliculus, and are probably concerned with reflex movements of the eyes depending on acoustic stimuli. The inferior colliculi, lower or posterior quadrigeminal bodies, are important auditory reflex centres. Each consists of a compact nucleus of grey matter covered by a superficial white layer and separated from the central grey matter about by the aqueduct by a thin, deep white layer. Many of the axons which appear in the superficial white layer ascend through the inferior brachium to the medial geniculate body. Others, mainly from large cells in the dorso-mesial part of the nucleus, pass through the deep white layer into the tegmentum of the same and the opposite side and descend. Their termination is unknown, but they probably constitute an auditory reflex path to the lower motor centres, perhaps descending into the spinal cord with the Tectospinal Fasciculus. Other axons are said to descend in the lateral lemniscus to the various nuclei in the auditory path, held, and probably to motor nuclei of the medulla and spinal cord. The medial geniculate body receives terminals and collaterals from the lateral lemniscus, the central auditory path, and also large numbers of axons from the inferior colliculus of the same side and a few from the opposite side. It is thus a station in the central auditory path. A large proportion of its axons pass forward beneath the optic tract to join the corona radiata and then sweep backward and lateralward as the auditory radiation to terminate in the cortex of the superior temporal gyrus. V. Monaco holds that Golgi cells type 2 are interpolated between the terminations of the incoming fibers to the medial geniculate body, and the cells located there which give rise to the fibers of the auditory radiation. The medial geniculate bodies are united by the long, slender commissure of Gooden. These fibers join the optic tract as it passes over the edge of the medial geniculate and passes through the posterior part of the optic chiasma. It is probably a commissure connected with the auditory system. The vestibular nerve, vestibular root, 8th cranial, arise from the bipolar cells in the vestibular ganglion, Scarpa's ganglion. The peripheral fibers end in the semicircular canals, the saccule and the utricle, the end organs concerned with the mechanism for the maintenance of bodily equilibrium. The central fibers enter the medulla oblongata and pass between the inferior peduncle and the spinal tract of the trigeminal. They bifurcate into ascending and descending branches, as do the dorsal root fibers of all the spinal nerves and all afferent cranial nerves. The descending branches terminate in the dorsal, medial, vestibular nucleus, the principal nucleus of the vestibular nerve. This nucleus is prolonged downward into a descending portion in which end terminals and collaterals of the descending branch. The ascending branches pass to Dieter's nucleus, to Bechterer's nucleus, and through the inferior peduncle of the cerebellum to the nucleus tecti of the opposite side. The dorsal vestibular nucleus, medial or principal nucleus, is a large mass of small cells in the floor of the fourth ventricle under the area acoustica, located partly in the medulla and partly in the pons. The stria medullaris cross the upper part of it. It is separated from the median plane by the nucleus intercalatus. Its axons pass into the posterior longitudinal bundle of the same and the opposite side, and ascend to terminate in the nucleus abducens of the same side, and in the trochlear nucleus and the oculomotor nucleus of the opposite side, and to the motor nuclei of the trigeminal on both sides. The descending portion, the nucleus of the descending tract, extends downward as far as the upper end of the nucleus gracilis, and the decussation of the medial lemniscus. It is sometimes called the inferior vestibular nucleus. Many of its axons cross the midline, and probably ascend with the medial lemniscus to the ventrolateral region of the thalamus. The lateral vestibular nucleus, Dieter's nucleus, is the continuation upward and lateralward of the principal nucleus, and in it terminate many of the ascending branches of the vestibular nerve. It consists of very large multipolar cells whose axons form an important part of the posterior longitudinal bundle of the same and the opposite side. The axons bifurcate as they enter the posterior longitudinal bundle. The ascending branches send terminals and collaterals to the motor nuclei of the abducens, trochlear and oculomotor nerves, and are concerned in coordinating the movements of the eyes with alterations in the position of the head. The descending branches pass down in the posterior longitudinal bundle into the anterior funiculus of the spinal cord as the Vestibulospinal Fasciculus, anterior marginal bundle, and are distributed to motor nuclei of the anterior column by terminals and collaterals. Other fibers are said to pass directly to the Vestibulospinal Fasciculus without passing into the posterior longitudinal bundle. The fibers which pass into the Vestibulospinal Fasciculus are intimately concerned with equilibratory reflexes. Other axons from Dieter's nucleus are supposed to cross and descend in the opposite medial lemniscus to the ventrolateral nuclei of the thalamus. Still other fibers pass into the cerebellum with the inferior peduncle and are distributed to the cortex of the vermis and the roof nuclei of the cerebellum. According to Cajal, they merely pass through the nucleus fasticii on their way to the cortex of the vermis and the hemisphere. The superior vestibular nucleus, Becher's nucleus, is the dorsolateral part of the vestibular nucleus and receives collaterals and terminals from the ascending branches of the vestibular nerve. Its axons terminate in much the same manner as do those from the lateral nucleus. The facial nerve, seventh cranial, consists of somatic sensory, sympathetic afferent, taste, somatic motor, and sympathetic efferent fibers. The afferent or sensory fibers arise from cells in the geniculate ganglion. This portion of the nerve is often described as the nervus intermedius. 1. The somatic sensory fibers are few in number and convey sensory impulses from the middle ear region. Their existence has not been fully confirmed. Their central termination is likewise uncertain. It is possible that they join in the spinal tract of the trigeminal, as do the somatic sensory fibers of the vagus and glossopharyngeal. 2. The sympathetic afferent fibers are likewise few in number and of unknown termination. 3. Taste fibers convey impulses from the anterior two-thirds of the tongue via the chorda tympani. They are supposed to join the Tractus Solitarius and terminate in its nucleus. The central connections of this nucleus have already been considered. 4. Somatic motor fibers, supplying the muscles derived from the hyoid arch, arise from the large multipolar cells of the nucleus of the facial nerve. This nucleus is serially homologous with the nucleus ambiguous and lateral part of the anterior column of the spinal cord. Voluntary impulses from the cerebral cortex are conveyed by terminals and collaterals of the pyramidal tract of the opposite side, indirectly, that is, with the interpolation of a connecting neuron to the facial nucleus. This nucleus undoubtedly receives many reflex fibers from the various sources, i.e. from the superior colliculus via the ventral longitudinal bundle, Tectospinal Fasciculus, for optic reflexes, from the inferior colliculus via the auditory reflex path, and indirectly from the terminal sensory nuclei of the brainstem. Through the posterior longitudinal bundle it is intimately connected with other motor nuclei of the brainstem. 5. Sympathetic efferent fibers, preganglionic fibers, arise according to some authors from the small cells of the facial nucleus, or according to others from a special nucleus of cells scattered in the reticular formation, dorsomedial to the facial nucleus. This is sometimes called the superior salivatory nucleus. These preganglionic fibers are distributed partly via the chorda tympani and lingual nerves to the submaxillary ganglion, thence by postganglionic vasodilator fibers to the submaxillary and sublingual glands. Some of the preganglionic fibers pass to the sphenopalatine ganglion via the great superficial petrosal nerve. The abducens nerve, sixth cranial, contains somatic motor fibers only, which supply the lateral rectus muscle of the eye. The fibers arise from the nucleus of the abducens nerve and pass ventrally through the formatio reticularis of the pons to emerge in the transverse groove between the caudal edge of the pons and the pyramid. The nucleus is serially homologous with the nuclei of the trochlea and oculomotor above, and with the hypoglossal and medial part of the anterior column of the spinal cord below. It is situated close to the floor of the fourth ventricle, just above the level of the stria medullaris. Voluntary impulses from the cerebral cortex are conducted by the pyramidal tract fibers, corticopontine fibers. These fibers probably terminate in relation with association neurons which control the coordinated action of all the eye muscles. This association and coordination mechanism is interposed between the terminals and collaterals of the voluntary fibers and the neurons within the nuclei of origin of the motor fibers to the eye muscles. The fibers of the posterior longitudinal bundle are supposed to play an important role in the coordination of the movements of the eyeball. Whether it is concerned only with coordinations between the vestibular apparatus and the eye, or with more extensive coordinations, is unknown. Many fibers of the posterior longitudinal bundle have their origin in the terminal nuclei of the vestibular nerve, and from the posterior longitudinal bundle many collaterals and terminals are given off to the abducens nucleus as well as to the trochlear and oculomotor nuclei. The abducens nucleus probably receives collaterals and terminals from the ventral longitudinal bundle, Tectospinal Fasciculus, fibers which have their origin in the superior colliculus, the primary visual centre, and are concerned with visual reflexes. Others probably come from the reflex auditory centre in the inferior colliculus and from other sensory nuclei of the brain stem. End of Part 20. Section 21 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. Composition and Connections of Cranial Nerves, Part 3. The trigeminal nerve, fifth cranial, contains somatic motor and somatic sensory fibers. The motor fibers arise in the motor nucleus of the trigeminal and pass ventrolaterally through the pons to supply the muscles of mastication. The sensory fibers arise from the unipolar cells of the semilunar ganglion. The peripheral branches of the T-shaped fibers are distributed to the face and anterior 2/3 of the head. The central fibers pass into the pons with the motor root, and bifurcate into ascending and descending branches, which terminate in the sensory nuclei of the trigeminal. The motor nucleus of the trigeminal is situated in the upper part of the pons, beneath the lateral angle of the fourth ventricle. It is serially homologous with the facial nucleus and the nucleus ambiguus, motor nucleus of the vagus and glossopharyngeal, which belong to the motor nuclei of the lateral somatic group. The axons arise from large pigmented multipolar cells. The motor nucleus receives reflex collaterals and terminals, one from the trigeminal nucleus of the trigeminal of the same, and a few from the opposite side, via the central sensory tract, trigeminothalamic tract. 2. From the mesencephalic root of the trigeminal. 3. From the posterior longitudinal bundle. 4. And probably from fibers in the formatio reticularis. It also receives collaterals and terminals from the opposite pyramidal tract, corticopontine fibers, for voluntary movements. There is probably a connecting or association neuron interposed between these fibers and the motor nucleus. The terminal sensory nucleus consists of an enlarged upper end, the main sensory nucleus, and a long, more slender descending portion which passes down through the pons and medulla to become continuous with the dorsal part of the posterior column of the grey matter, especially the substantia gelatinosa of the spinal cord. This descending portion consists mainly of substantia gelatinosa, and is called the nucleus of the spinal tract of the trigeminal nerve. The main sensory nucleus lies lateral to the motor nucleus beneath the superior peduncle. It receives the short ascending branches of the sensory root. The descending branches, which form the Tractus Spinalis, pass down through the pons and the medulla on the lateral side of the nucleus of the Tractus Spinalis, in which they end by collaterals and terminals, into the spinal cord on the level of the second cervical segment. It decreases rapidly in size as it descends. At first it is located between the emergent part of the facial nerve and the vestibular nerve, then between the nucleus of the facial nerve and the inferior peduncle. Lower down in the upper part of the medulla, it lies beneath the inferior peduncle and is broken up into bundles by the olivocerebellar fibers and the roots of the 9th and 10th cranial nerves. Finally, it comes to the surface of the medulla under the tubercle of Rolando, and continues in this position lateral to the fasciculus cuneatus as far as the upper part of the cervical region, where it disappears. The cells of the sensory nucleus are of large and medium size, and send their axons into the formatio reticularis, where they form a distinct bundle, the central part of the trigeminothalamic tract, which passes upward through the formatio reticularis and tegmentum to the ventrolateral part of the thalamus. Most of the fibers cross to the trigeminothalamic tract of the opposite side. This tract lies dorsal to the medial fillet, approaches close to it in the tegmentum, and terminates in a distant part of the thalamus. From the thalamus, impulses are conveyed to the somatic sensory area of the cortex by axons of cells in the thalamus through the internal capsule and corona radiata. Many collaterals are given off in the medulla and pass from the trigeminothalamic tract to the motor nuclei, especially to the nucleus ambiguus, the facial nucleus, and the motor nucleus of the trigeminal. The somatic sensory fibers of the vagus, the glossopharyngeal, and the facial nerves probably end in the nucleus of the descending tract of the trigeminal, and their cortical impulses are probably carried up in the central sensory path of the trigeminal. The mesencephalic root, descending root of the trigeminal, arises from unipolar cells arranged in scattered groups in a column at the lateral edge of the central grey matter surrounding the upper end of the fourth ventricle and the cerebral aqueduct. They have usually been considered as motor fibers that join the motor root, but Johnston claims that they join the sensory root of the trigeminal, that they develop in the alar, not the basal lamina, and that the pear-shaped unipolar cells are sensory in type. The trochlear nerve, fourth cranial, contains somatic motor fibers only. It supplies the superior oblique muscle of the eye. Its nucleus of origin, trochlear nucleus, is a small oval mass situated in the ventral part of the central grey matter of the cerebral aqueduct at the level of the upper part of the inferior colliculus. The axons from the nucleus pass downward in the tegmentum toward the pons, but turn abruptly downward before reaching it, and pass into the superior medullary velum, in which they cross horizontally to decussate with the nerve of the opposite side, and emerges from the surface of the velum, immediately behind the inferior colliculus. The cells of the trochlear nucleus are large, irregular, and yellowish in color. The nuclei of the two sides are separated by the raphe through which dendrites extend from one nucleus to the other. They receive many collaterals and terminals from the posterior longitudinal bundle, which lies on the ventral side of the nucleus. There are no branches from the fibers of the pyramidal tracts to these nuclei. The volitional pathway must be an indirect one, as is the case with the other motor nuclei. The oculomotor nerve, third cranial, contains somatic motor fibers to the obliquus inferior, rectus superior, levator palpebrae superioris, and rectus medialis muscles, and sympathetic efferent fibers, preganglionic fibers, to the ciliary ganglion. The postganglionic fibers connected with these supply the ciliary muscle and the sphincter of the iris. The axons arise from the nucleus of the oculomotor nerve, and pass in bundles through the posterior longitudinal bundle, the tegmentum, the red nucleus, and the medial margin of the substantia nigra in a series of curves, and finally emerge from the oculomotor sulcus on the medial side of the cerebral peduncle. The oculomotor nucleus lies in the grey substance of the floor of the cerebral aqueduct, subjacent to the superior colliculus, and extends in front of the aqueduct a short distance into the floor of the third ventricle. The inferior end is continuous with the trochlear nucleus. It is from 6 to 10 mm in length. It is intimately related to the posterior longitudinal bundle, which lies against its ventrolateral aspect, and many of its cells lie among the fibers of the posterior longitudinal bundle. The nucleus of the oculomotor nerve contains several distinct groups of cells which differ in size and appearance from each other, and are supposed to send their axons each to a separate muscle. Much uncertainty still exists as to which group supplies which muscle. There are 7 of these groups or nuclei on either side of the midline, and 1 medial nucleus. The cells of the anterior nuclei are smaller, and are supposed to give off the sympathetic efferent axons. The majority of fibers arise from the nucleus of the same side. Some, however, cross from the opposite side and are supposed to supply the rectus medialis muscle. Since oculomotor and abducens nuclei are intimately connected by the posterior longitudinal bundle, this decussation of fibers to the medial rectus may facilitate the conjugate movements of the eyes, in which the medial and lateral recti are especially involved. Many collaterals and terminals are given off to the oculomotor nucleus from the posterior longitudinal bundle, and thus connect it with the vestibular nucleus, the trochlear and abducens nuclei, and probably with other cranial nuclei. Fibers from the visual reflex center in the superior colliculus pass to the nucleus. It is also connected with the cortex of the occipital lobe of the cerebrum by fibers which pass through the optic radiation. The pathway for voluntary motor impulses is probably similar to that for the abducent nerve. The optic nerve, or nerve of sight, second cranial, consists chiefly of coarse fibers which arise from the ganglionic layer of the retina. They constitute the third neuron in the series composing the visual path, and are supposed to convey only visual impressions. A number of fine fibers also pass in the optic nerve from the retina to the primary centers, and are supposed to be concerned in the pupillary reflexes. There are in addition a few fibers which pass from the brain to the retina. They are supposed to control chemical changes in the retina and the movements of the pigment cells and cones. Each optic nerve has, according to Sulzer, about 500,000 fibers. In the optic chiasma, the nerves from the medial half of each retina cross to enter the opposite optic tract, while the nerves from the lateral half of each retina pass into the optic tract of the same side. The crossed fibers tend to occupy the medial side of each optic nerve, but in the chiasma and in the optic tract they are more intermingled. The optic tract is attached to the tuber cinereum and lamina terminalis, and also to the cerebral peduncle as it crosses obliquely over its under surface. These are not functional connections. A small band of fibers from the medial geniculate body joins the optic tract as the latter passes over it and crosses to the opposite tract and medial geniculate body in the posterior part of the chiasma. This is the commissure of Gooden, and is probably connected with the auditory system. Most of the fibers of the optic tract terminate in the lateral geniculate body. Some pass through the superior brachium to the superior colliculus, and others either pass over or through the lateral geniculate body to the pulvinar of the thalamus. These end stations are often called the primary visual centers. The lateral geniculate body consists of medium-sized pigmented nerve cells arranged in several layers by the penetrating fibers of the optic tract. Their axons pass upward beneath the longer fibers of the optic tract, the taenia semicircularis, the caudate nucleus, and the posterior horn of the lateral ventricle, where they join the optic radiation of Gratiolae. They pass backward and medially to terminate in the visuosensory cortex in the immediate neighborhood of the calcarine fissure of the occipital lobe. This center is connected with the one in the opposite side by commissural fibers which course in the optic radiation and the splenium of the corpus callosum. Association fibers connect it with other regions of the cortex of the same side. The region of the pulvinar in which optic tract fibers terminate resembles in structure the lateral geniculate body. Its axons also have a similar course, though in a somewhat more dorsal plane. The superior colliculus receives fibers from the optic tract through the superior brachium. Some enter by the superficial white layer, stratum zonale. Others appear to dip down into the grey cap, stratum cinereum, while others probably decussate across the midline to the opposite colliculus. Other fibers from the superior brachium pass into the stratum opticum, upper grey-white layer. Some of these turn upward into the grey cap, while others terminate among the cells of this layer. Since the superior colliculi appear to be the central organs concerned in the control of eye muscle movements and eye muscle reflexes, we should expect to find them receiving fibers from other sensory paths. Many fibers pass to the superior colliculus from the medial fillet, as the latter passes through the tegmentum, bringing the superior colliculus into relation with the sensory fibers of the spinal cord. Fibers from the central sensory path of the trigeminal probably pass with these. Part of the ventral spinocerebellar tract, Gowers, is said to pass up through the reticular formation of the pons and midbrain toward the superior colliculus and thalamus. The superior colliculus is intimately connected with the central auditory path, the lateral lemniscus, as part of its fibers pass the inferior colliculus and terminate in the superior colliculus. They are probably concerned with reflex movements of the eye depending on auditory stimuli. The superior colliculus is said to receive fibers from the stria medullaris thalamus of the opposite side, which pass through the commissura habenuli and turn back to the roof of the midbrain, especially to the superior colliculus. By this path, both the primary and cortical olfactory centers are brought into relation with the eye muscle reflex apparatus. The fibers which pass to the nuclei of the eye muscles arise from large cells in the stratum opticum and stratum lemnisci, and pass around the ventral aspect of the central grey matter, where most of them cross the midline in the phantom decussation of Maynard, and then turn downward to form the ventral longitudinal bundle. This bundle runs down partly through the red nucleus in the formatio reticularis, ventral to the posterior longitudinal bundle of the midbrain, pons, and medulla oblongata into the ventral funiculus of the spinal cord, where it is known as the Tectospinal Fasciculus. some of the fibers are said to pass down with the rubrospinal tract in the lateral funiculus. Some fibers do not decussate but pass down in the ventral longitudinal bundle of the same side on which they arise, unless possibly they come from the opposite colliculus over the aqueduct. From the ventral longitudinal bundle, collaterals are given off to the nuclei of the eye muscles, the ocular motor, the trochlea, and the abducens. Many collaterals pass to the red nucleus, and are probably concerned with the reflexes of the rubrospinal tract. The fibers of the tectospinal tract end by collaterals and terminals either directly or indirectly among the motor cells in the anterior column of the spinal cord. The superior colliculus receives fibers from the visual sensory area of the occipital cortex. They pass in the optic radiation. Probably no fibers pass from the superior colliculus to the visual sensory cortex. End of Part 21. Section 22 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4 by Henry Gray. Composition and Connections of Cranial Nerves, Part 4. The olfactory nerves, first cranial, or nerves of smell, arise from spindle-shaped bipolar cells in the surface epithelium of the olfactory region of the nasal cavity. The non-medullated axons pass upward in groups through numerous foramina in the cribriform plate to the olfactory bulb. Here several fibers, each ending in a tuft of terminal filaments, come into relation with the brush-like ends of a single dendrite from a mitral cell. This interlacing gives rise to the olfactory glomeruli of the bulb. The termination of several or many olfactory fibers in a single glomerulus, where they form synapses with the dendrites of one or two mitral cells, provides for the summation of stimuli in the mitral cells, and accounts in part at least for the detection by the olfactory organs of very dilute solutions. Lateral arborizations of the dendrites of the mitral cells, and the connection of neighboring glomeruli by the axons of small cells of the glomeruli, and the return of impulses of the mitral cells by collaterals, either directly or through the interpolation of granule cells to the dendrites of the mitral cells, reinforce the discharge of the mitral cells along their axons. The axons turn abruptly backward in the deep fiber layer of the bulb to form the olfactory tract. The olfactory tract is continued into the olfactory trigone, just in front of the anterior perforated substance. The axons of the mitral cells on reaching the olfactory trigone separate into three bundles: the lateral olfactory stria, the medial olfactory stria, and the less marked intermedial olfactory stria. The lateral olfactory stria curve lateralward. A few of the fibers end in the olfactory trigone and the anterolateral portion of the anterior perforated substance. Most of the fibers, however, pass into the uncus, the anterior end of the hippocampal gyrus, and there end in the complicated cortex of the hippocampal gyri. The lateral striae more or less disappear as they cross the anterolateral region of the anterior perforated substance. The greater mass of the fibers of the olfactory tract pass into the lateral stria. Numerous collaterals are given into the plexiform layer of the subfrontal cortex, over which the striae pass on their way to the uncus, where they intermingle with the apical dendrons of the medium-sized and small pyramidal cells of the pyramidal layer of this subfrontal or frontal olfactory cortex. The axons give rise to projection fibers, which take an antero-posterior direction to the subthalamic region, sending collaterals and terminal branches to the stria medullaris and others toward the thalamus. Some of the fibers extend farther back and are believed to reach the pons and the medulla oblongata. Most of the fibers of the lateral olfactory stria pass to the hippocampal region of the cortex, especially to the gyrus hippocampi, which may be regarded as the main ending place of the secondary olfactory path derived from axons of the mitral cells. The fibers of the medial olfactory striae terminate for the most part in the parolfactory area, Broca's area. A few end in the subcallosal gyrus, and a few in the anterior perforated substance and the adjoining part of the septum pellucidum. Some of the fibers pass into the anterior commissure, pars olfactoria, to the olfactory tract of the opposite side, where they end partly within the granular layer and partly in the neighbourhood of the glomeruli of the olfactory bulb, thus connecting the bulbs of the two sides. The intermediate olfactory striae are as a rule scarcely visible. The fibres terminate in the anterior perforated substance. A few are said to continue to the uncus. The trigonum olfactorium, anterior perforated substance, and the adjoining part of the septum pellucidum are important primary olfactory centres, especially for olfactory reflexes. In these centres terminate many axons from the mitral cells of the olfactory bulb. In addition, the grey substance of the olfactory tract and the gyrus subcallosus receive terminals of the mitral cells. The pathways from these centres to lower centres in the brainstem and spinal cord are only partly known. The most direct path, the Tractus olfactomesencephalicus, basal olfactory bundle of Wallenberg, is supposed to arise from cells in the grey substance of the olfactory tract, the olfactory trigone, the anterior perforated substance, and the adjoining part of the septum pellucidum. The fibres are said to pass direct to the tuber cinereum, to the corpus mammillare, to the brainstem, and the spinal cord. The fibres which enter the mammillary body probably come into relation with cells whose axons give rise to the fasciculus mammilotegmentalis, mammilotegmental bundle of Gooden, which is supposed to end in the grey substance of the tegmentum and of the aqueduct. Some of its fibres are said to join the posterior longitudinal bundle, and others to extend as far as the reticular formation of the pons. Some of the fibres of the medial olfactory stria come into relation with cells in the parolfactory area of Broca and in the anterior perforated substance, whose axons course in the medullary stria of the thalamus. As the axons pass through the lower part of the septum pellucidum, they are joined by other fibres whose cells receive impulses from the mitral cells. These fibres of the medullary stria end for the most part in the habenular nucleus of the same side. Some, however, cross in the habenular commissure, dorsal part of the posterior commissure, to the habenular nucleus of the opposite side. A few fibres of the medullary stria are said to pass by the habenular nucleus to the roof of the midbrain, especially the superior colliculus, while a few others come into relation with the posterior longitudinal bundle and association tracts of the mesencephalon. The ganglion of the habenuli, located in the trigonum habenuli, just in front of the superior colliculus, contain a mesial nucleus with small cells and a lateral nucleus with larger cells. The axons of these cells are grouped together in a bundle, Fasciculus retroflexus of Maynard, which passes ventrally medial to the red nucleus and terminates in a small medial ganglion in the substantia perforata posterior, immediately in front of the pons, called the interpeduncular ganglion. The interpeduncular ganglion has rather large nerve cells whose axons curve backward and downward as the tegmental bundle of Gooden, to end partly in the dorsal tegmental nucleus and surrounding grey substance, where they come into relation with association neurons and the dorsal longitudinal bundle of Schutz. The majority of the axons that arise from the mitral cells of the olfactory bulb, and course in the olfactory tract, course in the lateral olfactory stria to the uncus and hippocampal gyrus, and terminate in the cortex. Other fibres probably pass to the uncus and hippocampal gyrus from the primary olfactory centres in the trigonum and anterior perforated substance. The gyrus hippocampus is continued through the isthmus to the gyrus cinguli, which passes over the corpus callosum to the area parolfactoria. The cortical portions of these gyri are connected together by a thick association bundle, the cingulum, that lies buried in the depth of the gyrus cinguli, extending forward to the parolfactory area and backward into the hippocampal region. The axons from the gyrus cinguli pass into the cingulum, many of them bifurcate. The anterior branches together with the axons which run in that direction are traceable as far forward as the anterior part of the septum pellucidum and the anterior end of the corpus striatum, where some of them are incorporated with projection fibres passing toward the internal capsule. The branches and axons which pass backward terminate partly in the hippocampus, the dentate gyrus, and the hippocampal gyrus. Shorter association fibres connect various sections of the gyrus fornicatus, cingulate gyrus, isthmus, and hippocampal gyrus, and these with other regions of the cortex. These gyri constitute the cortical centre for smell. The dentate gyrus, which may be considered as a modified part of the hippocampus, is partially separated from the gyrus hippocampus by the hippocampal fissure and from the fimbria by the fimbrio-dentate sulcus. It is intimately connected with the hippocampal gyrus and the hippocampus. When followed backward, the dentate gyrus separates from the fimbria at the splenium, loses its incisions and knobs, and as the fasciola cinerea passes over the splenium onto the dorsal surface of the corpus callosum and spreads out into a thin layer of grey substance known as the indusium, which can be traced forward around the genu of the corpus callosum into the gyrus subcallosus. The white matter of the indusium, known as the medial longitudinal striae, nerves of Lancisi, and the lateral longitudinal striae are related to the indusium somewhat as the cingulum is to the gyrus cinguli. Axons from the indusium pass into the longitudinal striae, some running forward and others running backward, while some, after entering the medial longitudinal stria, pierce the corpus callosum to join the fornix. Some of the fibres which pass forward extend around the front of the corpus callosum and the anterior commissure, then curve downward, according to Cahal, enter the corpus striatum where they join the olfactory projection path. Other fibres are said to arise in the parolfactory area, the gyrus subcallosus and the anterior perforated substance, diagonal band of Broca, and course backward in the longitudinal striae to the dentate gyrus and the hippocampal region. The indusium is usually considered as a rudimentary part of the rhinencephalon. The olfactory projection fibres which arise from the pyramidal cells of the uncus and hippocampus and from the polymorphic cells of the dentate gyrus form a dense stratum on the ventricular surface, especially on the hippocampus, called the alveus. These fibres pass over into the fimbria and are continued into the fornix. About 1/4 of all the fibres of the fimbria are large projection fibres. The other 3/4 consist of fine commissural fibres which pass from the hippocampus of one side through the fimbria and hippocampal commissure, ventral psalterium or lyra, to the fimbria and hippocampus of the opposite side, where they penetrate the pyramidal layer and terminate in the stratum radiatum. The fibres which course in the fornix pass forward and downward into the corpora mammillare, where numerous collaterals are given off and a few terminate. Most of the fibres in the fornix, however, pass through the corpora, cross the middle line and turn downward in the reticular formation in which they are said to be traceable as far as the pons and possibly farther. As the fornix passes beneath the corpus callosum, it receives fibres from the longitudinal striae of the indusium and from the cingulum. These are the perforating fibres of the fornix which pass through the corpus callosum and course in the fornix toward the mammillary body. As the fornix passes the anterior end of the thalamus, a few fibres are given off to the stria medullaris of the thalamus, and turn back in the stria to the habenular ganglion of the same and the opposite side, having probably the same relation that the reflex fibres have which arise from the primary centres and course in the stria medullaris of the thalamus. Aside from the fibres of the fornix, which pass through the mammillary body to decussate and descend, as the mamillo-mesencephalic fasciculus, many fibres are said to pass into the bundle of Vix-Zazir, and one bundle of fibres is said to pass from the fornix to the tubus cinereum. The mammillary bodies receive collaterals and terminals, then, from the cortical centres via the fornix, and probably other collaterals and terminals are received directly from the primary centres through the tractus olfacto-mesencephalicus. According to Cahal, fibres also reach the mammillary body through the peduncle of the corpus mammillare, from the arcuate fibres of the tegmentum, and from the main fillet. The fornix probably brings the cortical centres into relation with the reflex path that runs from the primary centres to the mammillary body and the tubus cinereum. The bundle of Vix-Zazir, mammillothalamic fasciculus, arises from cells in both the medial and lateral nuclei of the mammillary body, and by fibres that are directly continued from the fornix. There axons divide within the grey matter. The coarser branches pass into the anterior nucleus of the thalamus as the bundle of Vix-Zazir. The finer branches pass downward as the mammillotegmental bundle of Gooden. The bundle of Vix-Zazir spreads out fan-like as it terminates in the anterior or dorsal nucleus of the thalamus. A few of the fibres pass through the dorsal nucleus to the angular nucleus of the thalamus. The axons from these nuclei are supposed to form part of the thalamocortical system. The mammillotegmental bundle has already been considered under the olfactory reflex paths. The amygdaloid nucleus and the taenia semicircularis, stria terminalis, probably belong to the central olfactory apparatus. The taenia semicircularis extends from the region of the anterior perforated substance to the nucleus amygdalae. Its anterior connections are not clearly understood. Fibers are said to arise from cells in the anterior perforated substance. Some of the fibers pass in front of the anterior commissure, others join the fornix for a short distance as they pass behind the anterior commissure. The two strands ultimately join to form the taenia and pass backward in the groove between the caudate nucleus and the thalamus to the amygdaloid nucleus. Other fibers are said to pass in the opposite direction from the amygdaloid nucleus to the thalamus. End of Part 22.
Section 23 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Anatomy of the Human Body, Part 4, by Henry Gray. Pathways from the brain to the spinal cord. The descending fasciculi, which convey impulses from the higher centers to the spinal cord and located in the lateral and ventral funiculi. The motor tract, conveying voluntary impulses, arises from the pyramid cells situated in the motor area of the cortex, the anterior, central, and the posterior portions of the frontal gyri, and the paracentral lobule. The fibers are at first somewhat widely diffused, but as they descend through the corona radiata, they gradually approach each other and pass between the lentiform nucleus and thalamus in the genu and anterior two-thirds of the occipital part of the internal capsule. Those in the genu are named the geniculate fibers, while the remainder constitute the cerebrospinal fibers. Proceeding downward, they enter the middle 3/5 of the base of the cerebral peduncle. The geniculate fibers cross the middle line and end by arborizing around the cells of the motor nuclei of the cranial nerves. The cerebrospinal fibers are continued downward into the pyramids of the medulla oblongata, and the transit of the fibers from the medulla oblongata is effected by two paths. The fibers nearest to the anterior median fissure cross the middle line, forming the decussation of the pyramids, and descend in the opposite side of medulla spinalis as the lateral cerebrospinal fasciculus. Cross-pyramidal tract. Throughout the length of the medulla spinalis, fibers from this column pass into the gray substance, to terminate either directly or indirectly around the motor cells of the anterior column. The more laterally placed portion of the tract does not decussate in the medulla oblongata, but descends as the anterior cerebrospinal fasciculus, direct pyramidal tract. These fibers, however, end in the anterior gray column of the opposite side of the medulla spinalis by passing across in the anterior white commissure. There is considerable variation in the extent to which decussation takes place in the medulla oblongata. About 2/3 or 3/4 of the fibers usually decussate in the medulla oblongata and the remainder in the medulla spinalis. The axons of the motor cells in the anterior column pass out as the fibers of the anterior roots of the spinal nerves, along which the impulses are conducted to the muscles of the trunk and limbs. From this it will be seen that all the fibers of the motor tract pass to the nuclei of the motor nerves on the opposite side of the brain or medulla spinalis, a fact which explains why a lesion involving the motor area of one side causes paralysis of the muscles of the opposite side of the body. Further, it will be seen that there is a break in the continuity of the motor chain. In the case of the cranial nerves, this break occurs in the nuclei of these nerves, and in the case of the spinal nerves, in the anterior gray column of the medulla spinalis. For clinical purposes, it is convenient to emphasize this break and divide the motor tract into two portions. One, a series of upper motor neurons, which comprises the motor cells in the cortex and their descending fibers down to the nuclei of the motor nerves. 2, a series of lower motor neurons, which includes the cells of the nuclei of the motor cerebral nerves or the cells of the anterior columns of the medulla spinalis and their axis cylinder processes to the periphery. The rubrospinal fasciculus arises from the large cells of the red nucleus. The fibers cross the raphe of the midbrain in the decussation of Forel and descend in the formatio reticularis of the pons and medulla
dorsal to the medial lemniscus, and as they pass into the spinal cord, come to lie in a position ventral to the cross-pyramidal tracts in the lateral funiculus. The rubrospinal fibers end either directly or indirectly by terminals and collaterals about the motor cells in the anterior column on the side opposite from their origin in the red nucleus. A few are said to pass down on the same side. Since the red nucleus is intimately related to the cerebellum by terminals and collaterals of the superior peduncle which arises in the dentate nucleus of the cerebellum, the rubrospinal fasciculus is supposed to be concerned with cerebellar reflexes, complex motor coordinations necessary in locomotion and equilibrium. The afferent paths concerned in these reflexes have already been partly considered, namely, the dorsal and ventral spinocerebellar fasciculi, and probably some of the fibers of the posterior funiculi which reach the cerebellum by the inferior peduncle. The tectospinal fasciculus arises from the superior colliculus of the roof tectum of the midbrain. The axons come from large cells in the stratum opticum and stratum lemnici and sweep ventrally around the central grey matter of the aqueduct, cross the raphe in the fountain decussation of Maynard, and turn downward in the tegmentum in the ventral longitudinal bundle. Some of the fibers do not cross in the raphe, but pass down on the same side. It is uncertain whether they come from the superior colliculus of the same side or arch over the aqueduct from the colliculus of the opposite side. The tectospinal fasciculus, which comprises the major part of the ventral longitudinal bundle, passes down through the tegmentum and reticular formation of the pons and medulla oblongata ventral to the medial longitudinal bundle. In the medulla, the two bundles are more or less intermingled, and the tectospinal portion is continued into the anterolateral funiculus of the spinal cord, ventral to the rubrospinal fasciculus, with which some of its fibers are intermingled. Some of the fibers of the tectospinal fasciculus pass through the red nucleus, giving off collaterals to it. Others are given off to the motor nuclei of the cranial nerves, and in the spinal cord they terminate either directly or indirectly by terminals and collaterals among the nuclei of the anterior column. Since the superior colliculus is an important optic reflex center, this tract is probably concerned in optic reflexes, and possibly also with auditory reflexes, since some of the fibers of the central auditory path, the lateral lemniscus, terminate in the superior colliculus. The Vestibulospinal Fasciculus, part of the anterior marginal fasciculus, or Lowenthal's tract, situated chiefly in the marginal part of the anterior funiculus, is mainly derived from the cells of the terminal nuclei of the vestibular nerve, probably Dieter's and Bechtereau's, and some of its fibers are supposed to come from the nucleus fastigius, roof nucleus of the cerebellum. The latter nucleus is intimately connected with Dieter's and Bechtereau's nuclei. The vestibulospinal fasciculus is concerned with equilibratory reflexes. Its terminals and collaterals end about the motor cells in the anterior column. It extends to the sacral region of the cord. Its fibers are intermingled with the ascending spinothalamic fasciculus, with the anterior proper fasciculus, and laterally with the tectospinal fasciculus. Its fibers are supposed to be both crossed and uncrossed. In the brain stem it is associated with the dorsal longitudinal bundle. The pontospinal fasciculus, Bechtereau, arises from the cells in the reticular formation of the pons from the same and the opposite side, and is associated in the brain stem with the ventral longitudinal bundle. In the cord it is intermingled with the fibers of the vestibulospinal fasciculus in the anterior funiculus. Not much is known about this tract. There are probably other descending fasciculi, such as the thalamospinal, but not much is known about them. End of section 23.
Section 24 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Read by Laurie Anne Walden. Anatomy of the Human Body, Part 4, by Henry Gray. The Meninges of the Brain and Medulla Spinalis, Part 1. The brain and medulla spinalis are enclosed within three membranes. These are named from without inward, the Dura Mater, the arachnoid, and the pia mater. The Dura Mater. The Dura Mater is a thick and dense inelastic membrane. The portion which encloses the brain differs in several essential particulars from that which surrounds the Medulla Spinalis, and therefore it is necessary to describe them separately, but at the same time it must be distinctly understood that the two form one complete membrane, and are continuous with each other at the foramen magnum. The cranial Dura Mater, Dura Mater encephali, dura of the brain, lines the interior of the skull, and serves the twofold purpose of an internal periosteum to the bones, and a membrane for the protection of the brain. It is composed of two layers, an inner or meningeal, and an outer or endosteal, closely connected together, except in certain situations, where, as already described, they separate to form sinuses for the passage of venous blood. Its outer surface is rough and fibrillated, and adheres closely to the inner surfaces of the bones, the adhesions being most marked opposite the sutures and at the base of the skull. Its inner surface is smooth and lined by a layer of endothelium. It sends inward 4 processes which divide the cavity of the skull into a series of freely communicating compartments for the lodgment and protection of the different parts of the brain, and it is prolonged to the outer surface of the skull through the various foramina which exist at the base, and thus becomes continuous with the pericranium. Its fibrous layer forms sheaths for the nerves which pass through these apertures. Around the margin of the foramen magnum it is closely adherent to the bone, and is continuous with the spinal Dura Mater. Processes. The processes of the cranial Dura Mater, which projects into the cavity of the skull, are formed by reduplications of the inner or meningeal layer of the membrane, and are 4 in number: the falx cerebri, the tentorium cerebelli, the falx cerebelli, and the diaphragma celli. The falx cerebri, so named from its sickle-like form, is a strong, arched process which descends vertically in the longitudinal fissure between the cerebral hemispheres. It is narrow in front, where it is attached to the crista galli of the ethmoid, and broad behind, where it is connected with the upper surface of the tentorium cerebelli. Its upper margin is convex and attached to the inner surface of the skull in the middle line, as far back as the internal occipital protuberance. It contains the superior sagittal sinus. Its lower margin is free and concave, and contains the inferior sagittal sinus. The tentorium cerebelli is an arched lamina, elevated in the middle, and inclining downward toward the circumference. It covers the superior surface of the cerebellum, and supports the occipital lobes of the brain. Its anterior border is free and concave, and bounds a large oval opening, the incisura tentorii, for the transmission of the cerebral peduncles. It is attached, behind, by its convex border, to the transverse ridges upon the inner surface of the occipital bone, and there encloses the transverse sinuses; in front, to the superior angle of the petrous part of the temporal bone on either side, enclosing the superior petrosal sinuses. At the apex of the petrous part of the temporal bone the free and attached borders meet, and crossing one another are continued forward to be fixed to the anterior and posterior clinoid processes respectively. To the middle line of its upper surface the posterior border of the falx cerebri is attached, the straight sinus being placed at their line of junction. The falx cerebelli is a small triangular process of Dura Mater, received into the posterior cerebellar notch. Its base is attached, above, to the under and back part of the tentorium, its posterior margin to the lower division of the vertical crest on the inner surface of the occipital bone. As it descends it sometimes divides into 2 smaller folds, which are lost on the sides of the foramen magnum. The diaphragma celli is a small circular horizontal fold, which roofs in the sella turcica and almost completely covers the hypophysis. A small central opening transmits the infundibulum. Structure. The cranial Dura Mater consists of white fibrous tissue and elastic fibers arranged in flattened laminae which are imperfectly separated by lacunar spaces in blood vessels into 2 layers, endosteal and meningeal. The endosteal layer is the internal periosteum for the cranial bones, and contains the blood vessels for their supply. At the margin of the foramen magnum it is continuous with the periosteum lining the vertebral canal. The meningeal or supporting layer is lined on its inner surface by a layer of nucleated flattened mesothelium, similar to that found on serous membranes. The arteries of the Dura Mater are very numerous. Those in the anterior fossa are the anterior meningeal branches of the anterior and posterior ethmoidal and internal carotid, and a branch from the middle meningeal. Those in the middle fossa are the middle and accessory meningeal of the internal maxillary, a branch from the ascending pharyngeal, which enters the skull through the foramen lacerum, branches from the internal carotid, and a recurrent branch from the lacrimal. Those in the posterior fossa are meningeal branches from the occipital, one entering the skull through the jugular foramen, and another through the mastoid foramen, the posterior meningeal from the vertebral, occasional meningeal branches from the ascending pharyngeal, entering the skull through the jugular foramen and hypoglossal canal, and a branch from the middle meningeal. The veins returning the blood from the cranial Dura Mater anastomose with the diploic veins and end in the various sinuses. Many of the meningeal veins do not open directly into the sinuses, but indirectly through a series of ampullae, termed venous lacunae. These are found on either side of the superior sagittal sinus, especially near its middle portion, and are often invaginated by arachnoid granulations. They also exist near the transverse and straight sinuses. They communicate with the underlying cerebral veins, and also with the diploic and emissary veins. The nerves of the cranial Dura Mater are filaments from the semilunar ganglion, from the ophthalmic, maxillary, mandibular, vagus, and hypoglossal nerves, and from the sympathetic. The spinal Dura Mater, Dura Mater spinalis, spinal dura, forms a loose sheath around the Medulla Spinalis, and represents only the inner or meningeal layer of the cranial Dura Mater. The outer or endosteal layer ceases at the foramen magnum, its place being taken by the periosteum lining the vertebral canal. The spinal Dura Mater is separated from the arachnoid by a potential cavity, the subdural cavity. The two membranes are, in fact, in contact with each other, except where they are separated by a minute quantity of fluid which serves to moisten the opposed surfaces. It is separated from the wall of the vertebral canal by a space, the epidural space, which contains a quantity of loose areolar tissue and a plexus of veins. The situation of these veins between the Dura Mater and the periosteum of the vertebrae corresponds, therefore, to that of the cranial sinuses between the meningeal and endosteal layers of the cranial Dura Mater. The spinal Dura Mater is attached to the circumference of the foramen magnum and to the second and third cervical vertebrae. It is also connected to the posterior longitudinal ligament, especially near the lower end of the vertebral canal, by fibrous slips. The subdural cavity ends at the lower border of the second sacral vertebra. Below this level the Dura Mater closely invests the filum terminale and descends to the back of the coccyx, where it blends with the periosteum. The sheath of Dura Mater is much larger than is necessary for the accommodation of its contents, and its size is greater in the cervical and lumbar regions than in the thoracic. On each side may be seen the double openings which transmit the two roots of the corresponding spinal nerve, the Dura Mater being continued in the form of tubular prolongations on them as they pass through the intervertebral foramina. These prolongations are short in the upper part of the vertebral column, but gradually become longer below, forming a number of tubes of fibrous membrane which enclose the lower spinal nerves and are contained in the vertebral canal. Structure. The spinal Dura Mater resembles in structure the meningeal or supporting layer of the cranial Dura Mater, consisting of white fibrous and elastic tissue arranged in bands or lamellae, which, for the most part, are parallel with one another and have a longitudinal arrangement. Its internal surface is smooth and covered by a layer of mesothelium. It is sparingly supplied with blood vessels, and a few nerves have been traced into it. The arachnoid. The arachnoid is a delicate membrane enveloping the brain and medulla spinalis, and lying between the pia mater internally and the Dura Mater externally. It is separated from the pia mater by the subarachnoid cavity, which is filled with cerebrospinal fluid. The cranial part, arachnoidea encephali, of the arachnoid, invests the brain loosely, and does not dip into the sulci between the gyri, nor into the fissures, with the exception of the longitudinal. On the upper surface of the brain the arachnoid is thin and transparent; at the base it is thicker, and slightly opaque toward the central part, where it extends across between the two temporal lobes in front of the pons, so as to leave a considerable interval between it and the brain. The spinal part, Arachnoidea spinalis, of the arachnoid, is a thin, delicate, tubular membrane loosely investing the medulla spinalis. Above it is continuous with the cranial arachnoid. Below it widens out and invests the cauda equina and the nerves proceeding from it. It is separated from the Dura Mater by the subdural space, but here and there this space is traversed by isolated connective tissue trabeculae, which are most numerous on the posterior surface of the medulla spinalis. The arachnoid surrounds the cranial and spinal nerves, and encloses them in loose sheaths as far as their points of exit from the skull and vertebral canal. Structure. The arachnoid consists of bundles of white fibrous and elastic tissue intimately blended together. Its outer surface is covered with a layer of low cuboidal mesothelium. The inner surface and the trabeculae are likewise covered by a somewhat low type of cuboidal mesothelium, which in places are flattened to a pavement type. Vessels of considerable size, but few in number, and, according to Bachtelek, a rich plexus of nerves derived from the motor root of the trigeminal, the facial, and the accessory nerves, are found in the arachnoid. The subarachnoid cavity, cavum subarachnoideale, subarachnoid space, is the interval between the arachnoid and pia mater. It is occupied by a spongy tissue consisting of trabeculae of delicate connective tissue and intercommunicating channels in which the subarachnoid fluid is contained. This cavity is small on the surface of the hemispheres of the brain. On the summit of each gyrus the pia mater and the arachnoid are in close contact, but in the sulci between the gyri triangular spaces are left, in which the subarachnoid trabecular tissue is found, for the pia mater dips into the sulci, whereas the arachnoid bridges across them from gyrus to gyrus. At certain parts of the base of the brain the arachnoid is separated from the pia mater by wide intervals which communicate freely with each other and are named subarachnoid cisternae. In these the subarachnoid tissue is less abundant. End of section 24. Section 25 of Gray's Anatomy, Part 4. This is a Librivox recording. All Librivox recordings are in the public domain. For more information or to volunteer, please visit Librivox.org. Read by Laurie Anne Walden. Anatomy of the Human Body, Part 4 by Henry Gray. The Meninges of the Brain and Medulla Spinalis, Part 2. Subarachnoid cisternae, cisternae subarachnoidales. The cisterna cerebellomedullaris, cisterna magna, is triangular on sagittal section, and results from the arachnoid bridging over the interval between the medulla oblongata and the under surfaces of the hemispheres of the cerebellum. It is continuous with the subarachnoid cavity of the medulla spinalis at the level of the foramen magnum. The cisterna pontis is a considerable space on the ventral aspect of the pons. It contains the basilar artery, and is continuous behind with the subarachnoid cavity of the medulla spinalis, and with the cisterna cerebellomedullaris, and in front of the pons with the cisterna interpeduncularis. The cisterna interpeduncularis, cisterna basalis, is a wide cavity where the arachnoid extends across between the two temporal lobes. It encloses the cerebral peduncles and the structures contained in the interpeduncular fossa, and contains the arterial circle of Willis. In front, the cisterna interpeduncularis extends forward across the optic chiasma, forming the cisterna chiasmatis, and onto the upper surface of the corpus callosum, for the arachnoid stretches across from one cerebral hemisphere to the other immediately beneath the free border of the falx cerebri, and thus leaves a space in which the anterior cerebral arteries are contained. The cisterna fossae cerebri lateralis is formed in front of either temporal lobe by the arachnoid bridging across the lateral fissure. This cavity contains the middle cerebral artery. The cisterna veni magni cerebri occupies the interval between the splenium of the corpus callosum and the superior surface of the cerebellum. It extends between the layers of the tela choroidea of the third ventricle, and contains the great cerebral vein. The subarachnoid cavity communicates with the general ventricular cavity of the brain by 3 openings. One, the foramen of Magendie, is in the middle line at the inferior part of the roof of the fourth ventricle. The other 2 are at the extremities of the lateral recesses of that ventricle, behind the upper roots of the glossopharyngeal nerves, and are known as the foramina of Luschka. It is still somewhat uncertain whether these foramina are actual openings or merely modified areas of the inferior velum which permit the passage of the cerebrospinal fluid from the ventricle into the subarachnoid spaces as through a permeable membrane. The spinal part of the subarachnoid cavity is a very wide interval, and is the largest at the lower part of the vertebral canal, where the arachnoid encloses the nerves which form the cauda equina. Above, it is continuous with the cranial subarachnoid cavity. Below, it ends at the level of the lower border of the second sacral vertebra. It is partially divided by a longitudinal septum, the subarachnoid septum, which connects the arachnoid with the pia mater, opposite the posterior median sulcus of the medulla spinalis, and forms a partition, incomplete and cribriform above, but more perfect in the thoracic region. The spinal subarachnoid cavity is further subdivided by the ligamentum denticulatum, which will be described with the pia mater. The cerebrospinal fluid is a clear, limpid fluid, having a saltish taste, and a slightly alkaline reaction. According to Lassagne, it consists of 98.5 parts of water, the remaining 1.5% being solid matters, animal and saline. It varies in quantity, being most abundant in old persons, and is quickly secreted. The arachnoid villi, granulationes arachnoideales, glandulae pacchioni, pacchionian bodies, are small, fleshy-looking elevations, usually collected into clusters of variable size, which are present upon the outer surface of the dura mater, in the vicinity of the superior sagittal sinus, and in some other situations. Upon laying open the sagittal sinus and the venous lacunae on either side of it, villi will be found protruding into its interior. They are not seen in infancy, and very rarely until the 3rd year. They are usually found after the 7th year, and from this period they increase in number and size as age advances. They are not glandular in structure, but are enlarged normal villi of the arachnoid. As they grow, they push the thinned dura mater before them, and cause absorption of the bone from pressure, and so produce the pits or depressions on the inner wall of the calvarium. Structure. An arachnoidal villus represents an invasion of the dura by the arachnoid membrane. The latter penetrates the dura in such a manner that the arachnoid mesothelial cells come to lie directly beneath the vascular endothelium of the great dural sinuses. It consists of the following parts. 1. In the interior is a core of subarachnoid tissue, continuous with the meshwork of the general subarachnoid tissue through a narrow pedicle by which the villus is attached to the arachnoid. 2. Around this tissue is a layer of arachnoid membrane, limiting and enclosing the subarachnoid tissue. 3. Outside this is the thinned wall of the lacuna, which is separated from the arachnoid by a potential space which corresponds to and is continuous with the subdural cavity. 4. And finally, if the villus projects into the sagittal sinus, it will be covered by the greatly thinned wall of the sinus, which may consist merely of endothelium. It will be seen, therefore, that fluid injected into the subarachnoid cavity will find its way into these villi, and it has been found experimentally that it passes from the villi into the venous sinuses, into which they project. The pia mater. The pia mater is a vascular membrane consisting of a minute plexus of blood vessels, held together by an extremely fine areolar tissue, and covered by a reflection of the mesothelial cells from the arachnoid trabeculae. It is an incomplete membrane, absent probably at the foramen of Magendie and the 2 foramina of Luschka, and perforated in a peculiar manner by all the blood vessels as they enter or leave the nervous system. In the perivascular spaces, the pia apparently enters as a mesothelial lining of the outer surface of the space. A variable distance from the exterior, these cells become unrecognizable, and are apparently lacking, replaced by neuroglia elements. The inner walls of these perivascular spaces seem likewise covered, for a certain distance, by the mesothelial cells, reflected with the vessels from the arachnoid covering of these vascular channels as they traverse the subarachnoid spaces. The cranial pia mater encephali, pia of the brain, invests the entire surface of the brain, dips between the cerebral gyri and cerebellar laminae, and is invaginated to form the tela choroidea of the third ventricle, and the choroid plexuses of the lateral and third ventricles. As it passes over the roof of the fourth ventricle, it forms the tela choroidea and the choroid plexuses of this ventricle. On the cerebellum the membrane is more delicate. The vessels from its deep surface are shorter, and its relations to the cortex are not so intimate. The spinal pia mater spinalis, pia of the cord, is thicker, firmer, and less vascular than the cranial pia mater. This is due to the fact that it consists of two layers, the outer, or additional one, being composed of bundles of connective tissue fibers, arranged, for the most part, longitudinally. Between the layers are cleft-like spaces which communicate with the subarachnoid cavity, and a number of blood vessels which are enclosed in perivascular lymphatic sheaths. The spinal pia mater covers the entire surface of the medulla spinalis, and is very intimately adherent to it. In front, it sends a process backward into the anterior fissure. A longitudinal fibrous band, called the linea splendens, extends along the middle line of the anterior surface, and a somewhat similar band, the ligamentum denticulatum, is situated on either side. Between the conus medullaris, the pia mater is continued as a long, slender filament, filum terminale, which descends through the center of the mass of nerves forming the cauda equina. It blends with the Dura Mater at the level of the lower border of the second sacral vertebra, and extends downward as far as the base of the coccyx, where it fuses with the periosteum. It assists in maintaining the medulla spinalis in its position during the movements of the trunk, and is, from this circumstance, called the central ligament of the medulla spinalis. The pia mater forms sheaths for the cranial and spinal nerves. These sheaths are closely connected with the nerves, and blend with their common membranous investments. The ligamentum denticulatum, dentate ligament, is a narrow fibrous band situated on either side of the medulla spinalis throughout its entire length, and separating the anterior from the posterior nerve roots. Its medial border is continuous with the pia mater at the side of the medulla spinalis. Its lateral border presents a series of triangular, tooth-like processes, the points of which are fixed at intervals to the Dura Mater. These processes are 21 in number, on either side, the first being attached to the Dura Mater, opposite the margin of the foramen magnum, between the vertebral artery and the hypoglossal nerve, and the last near the lower end of the medulla spinalis. End of the meninges of the brain and medulla spinalis. The cerebrospinal fluid. The cerebrospinal fluid, for the most part elaborated by the choroid plexuses, is poured into the cerebral ventricles which are lined by smooth ependema. That portion of the fluid formed in the lateral ventricles escapes by the foramen of Monro into the third ventricle, and thence by the aqueduct into the fourth ventricle. Likewise an ascending current of fluid apparently occurs in the central canal of the spinal cord. This, representing a possible product of the ependema, may be added to the intraventricular supply. From the fourth ventricle, the fluid is poured into the subarachnoid spaces through the medial foramen of Magendie and the two lateral foramina of Luschka. There is no evidence that functional communications between the cerebral ventricles and the subarachnoid spaces exist in any region except from the fourth ventricle. In addition to the elaboration of the cerebrospinal fluid by the choroid plexuses, there seems fairly well established a second source of the fluid from the nervous system itself. The blood vessels that enter and leave the brain are surrounded by perivascular channels. It seems most likely that the outer wall of these channels is lined by a continuation inward of the pial mesothelium, while the inner wall is probably derived from the mesothelial covering of the vessels, which are thus protected throughout the subarachnoid spaces. These mesothelial cells continue inward only a short distance, neuroglia cells probably replacing, on the outer surface, the mesothelial elements. Through these perivascular channels there is probably a small amount of fluid flowing from nerve cell to subarachnoid space. The chemical differences between the subarachnoid fluid, product of choroid plexuses and perivascular system, and the ventricular fluid, product of choroid plexuses alone, indicate that the products of nerve metabolism are poured into the subarachnoid space. The absorption of the cerebrospinal fluid is a dual process, being chiefly a rapid drainage through the arachnoid villi into the great dural sinuses, and, in small part, a slow escape into the true lymphatic vessels, by way of an abundant but indirect perineural course. In general, the arachnoid channels are equipped as fluid retainers with unquestionable powers of diffusion or absorption in regard to certain elements in the normal cerebrospinal fluid, deriving in this way a cellular nutrition. The subdural space, between arachnoid and dura, is usually considered to be a part of the cerebrospinal channels. It is a very small space, the two limiting surfaces being separated by merely a capillary layer of fluid. Whether this fluid is exactly similar to the cerebrospinal fluid is very difficult to ascertain. Likewise, our knowledge of the connections between the subdural and subarachnoid spaces is hardly definite. In some ways, the subdural space may be likened to a serous cavity. The inner surface of the dura is covered by flattened polygonal mesothelial cells, but the outer surface of the arachnoid is covered by somewhat cuboidal mesothelium. The fluid of the subdural space has probably a local origin from the cells lining it. End of section 25.